Therapeutic Development Branch, and it's my pleasure to introduce Dr. Joni Rudder, NCATS Director, for her opening welcome remarks. Good morning, Joni.
All right, thank you so much, Liz. Hello, everyone. It's great to be here. It's great to see you. And we're coming together today under sort of under NCATS to.
We're preschoolers that talk about future.
Oh, I'm hearing some feedback.
You're muted.
Yeah, Joni got muted.
Okay, how about that? There we go. Everybody, can you hear me now? All right, okay. Thank you. Okay, so welcome everybody. We're coming today to come together under the invitation of Liz and Mark and various other folks here at NCATS to address, really those common bottlenecks and common issues that have been slowing down translational science for a long time. And today we're really focused on the idea of recognizing and addressing the pressing challenge of rare diseases specifically. And as many of you know, there are over 10,000 rare diseases, yet we only have treatments for a mere 5% of them. And that leaves a substantial unmet medical need, and one that we are committed in NCATS to tackle head on. And we do have an ambitious goal. We aim to see the number of those 10,000 rare diseases covered within the experimental pipeline. At least we want to raise that 5% to something like 25% over the next 10 years. And so, yes, 25% is a significant leap. But I'd like to emphasize that I'd rather aim high and fall short at 25% than settle for a modest progress at 6%. So we have a lot of work to do, but I believe that with the tools and technologies that we have today, with the focus on data science and other key areas of science, we have a real opportunity to do that. So NCATS has always focused on rare diseases, but recently we've been elevating our game. We're elevating our efforts in this crucial space. And so how do we plan on addressing this sort of audacious goal efficiently? Well, we're not looking at this challenge at a sort of one disease at a time pace. We're focusing on therapeutic development approaches that can address multiple diseases simultaneously. And so by studying the genes and the pathways involved in disease manifestations, we can identify those overlaps and opportunities for broader therapeutic strategies. And I think you'll hear this theme throughout the day today, but certainly within our next keynote speaker in David Fagenbaum. I'm just delighted to see he's here today. So by studying the genes and pathways involved in those diseases, we can move the needle on the various therapeutics that could benefit a wide range of diseases, all within the same treatment approaches. And furthermore, that combined with the idea that the FDA Modernization Act 2.0 has really opened doors for non-animal alternative approaches, removing the requirement for animal models. And so we see this as a golden opportunity for non-animal model approaches to flourish and for really modeling different diseases effectively within what we're trying to do here. And NCATS is fully committed to supporting the development, validation, and the use of non-animal models, including initiatives like the NCATS tissue chip for drug screening program, as well as our 3D bioprinting programs. And we have experts here today from NCATS and all over really to help talk about some of those promises within using those technologies too. And last but not least, partnerships are at the core of our mission. The biomedical ecosystem is vast, it's complex, and we can't do any one thing alone. And so we believe in leveraging the strengths of the different sectors within that biomedical ecosystem so that we can really attack these challenges head on. And today's workshop is a shining example of how we engage with the biomedical community to assess the landscape for using non-animal models to fast-track therapies for rare diseases. So I'm looking forward to really diving right in and coming at it today with a lot of thinking about innovation, coming at it with the attitude of grit and determination, and a shared commitment to making a significant impact on the realm of rare diseases. And so I really appreciate you all being here today. So let's just jump in, and this promises to be a day of valuable insights and progress, and I look forward to certainly hearing a lot more. So thank you so much, Liz, and I'll turn it back over to you.
Thank you. Thank you so much, Joni. And now I have the pleasure of introducing our keynote speaker, Dr. David Feigenbaum, a physician scientist at the University of Pennsylvania, co-founder of Every Cure, and the national best-selling author of Chasing My Cure, A Doctor's Race to Turn Hope into Action. Before Dr. Feigenbaum begins, I'd also want to let the audience know that they can post questions in the Q&A that you should see the bottom button at the bottom. And if we have some time left, about 5 minutes, we'll try to get some questions in for Dr. Feigenbaum. So thank you so much, and I'm going to turn it over to you.
Liz, thank you so much for the introduction. And Joni, thank you so much for the opportunity to be with you and to be with the rest of this amazing NCATS community, and to have the opportunity to share about the work that we're doing, looking at computational drug discovery and drug repurposing, and how critical all of you are that are part of the non-animal model community so that we can actually validate these promising hits. So I'm going to begin by sharing my screen, but really just want to thank you all for the work that you do. And of course, thank Joni and Liz for the opportunity to share about the work that I do. Great. So today I'll be sharing about the potential for unlocking FDA-approved drugs and the treatment and the additional disease that each FDA-approved drug can potentially address. This is a very personal mission for me. When I was a third-year medical student at the University of Pennsylvania, I became critically ill with a rare and deadly disease called Castleman disease. I spent months and months in ICU, and was even so sick at one point with this horrible disease that my doctors told my family to say their final goodbyes, and a priest read me my last rites. Now, just a few years before, I was a healthy college quarterback. I was a medical student, as I shared, and I was hoping to become an oncologist in memory of my mom. She had passed away from brain cancer, and that's what drove me to want to dedicate my life and my career towards medicine. And I was marching along in that journey, and I was in my third year of medical school when I became so critically ill with this horrible disease. At the very last moment, I was diagnosed. The disease is called idiopathic multicentric Castleman disease, a hyperinflammatory disorder where basically your immune system attacks and shuts down your vital organs for an unknown cause. Unfortunately, it's quite deadly, with about a third of patients dying within 5 years and another third dying within 10 years of diagnosis. I was given a combination of 7 chemotherapies in a last-ditch effort to hopefully save my life, and somehow I survived. And of course, so thankful for this treatment. Of course, this was the first exposure that I had to this concept that a drug that wasn't made for my disease could actually save my life. These 7 chemotherapies, adriamycin, Cytoxan, etoposide, Velcade, Dex, thalidomide, Rituxan, they were not developed for Castleman disease. They were not approved for Castleman disease. They were developed and approved for a number of hematologic malignancies and other cancers, but had never been used, or sorry, had been used for Castleman's, but certainly were not approved for Castleman's. And this sort of concept, wait a minute, these drugs aren't made for my disease, but they just saved my life, certainly stuck with me. After spending about 6 months hospitalized, battling my illness and nearly dying 3 times during that period, I finally was well enough to be discharged and to go home. And you can see how big my belly was because of the liver and the kidney failure that comes with this hyperinflammatory cytokine storm that you see with Castleman disease. And thankfully, the chemotherapy saved my life. I was able to return to medical school at Penn, continue on that dream of unlocking treatments for cancer in memory of my mom, when I relapsed and I nearly died now for the 4th time. What was so difficult about this relapse is that I was on the only drug in development for my disease, the only drug that's ever undergone a randomized controlled trial, and it didn't work for me. So now I was in a position where not only were there no FDA-approved drugs, like Joni mentioned earlier, for the vast majority of rare diseases, but the only one in clinical development didn't work for me. And so my doctor came in my room and I asked him, I said, what other drugs are in development? What else is coming down the pipeline? And he explained to me, David, there are no more promising leads. There are no more drugs in development. This is it. We're going to give you chemotherapy. We're going to hope that it works. And eventually it's going to stop working. And there are no more drugs coming down the pipeline. And so when my doctor explained that to me, and after he left the room, I turned to my family and I said I would dedicate my life, however long that may be, trying to find treatments for me and for other patients like me. And now, given that I was relapsing every just several months, I knew I didn't have a lot of time. I didn't have 10 years and $1 billion to develop a new drug from scratch. If I wanted to survive, I would need to find something in the next several months that could maybe save my life. So by necessity, I thought back to those chemotherapy drugs that weren't made for my disease. And I thought, well, maybe there's a drug out there that could be repurposed that could save my life. Of course, chemotherapy is being repurposed and saving my life. Maybe there's some other immunomodulator that could potentially treat my disease. So in order to do that, I had to start digging into the disease itself and understand what are the underlying mechanisms of the disease. And for many years, we understood that Castleman's involved this hyperinflammatory syndrome and this particularly unusual appearance under the microscope of the lymph nodes. And that was well established, but it wasn't until the late 80s, early 90s that a researcher in Japan discovered that interleukin-6 was elevated in a large portion of patients and was likely responsible for the cytokine storm in a large number of patients. So though the entire immune system is really activated, interleukin-6 seems to be playing a really critical role. But even back then and still today, there's really been a black box. Well, why is the interleukin-6 elevated in the first place? What are the signaling pathways, the cell types, the stimuli that lead to the elevated interleukin-6? Are there autoimmune mechanisms? Is this driven by some sort of pathogen that hasn't been discovered yet? Are there neoplastic mechanisms? Should this actually be considered a lymphoma? We don't know the answers to those, though we are certainly honing in on the right answer to these questions. But, understanding the underlying mechanisms isn't really all that important if you can block interleukin-6 and patients just get better. And it's pretty amazing. About 1/3 of patients do get better. And that experimental drug I mentioned is called cetuximab. It did not work for me. So what we've subsequently learned is about 1/3 of patients will respond to cetuximab or IL-6 blockade, and about 2/3 of patients don't respond to IL-6 blockade. And for those of us that don't respond to IL-6 blockade, now we really don't have a clue about what to go after to target drugs against. And so for those patients, we use multi-agent chemotherapy. We just basically obliterate the immune system. If you don't know what to target, just knock out the whole immune system. And fortunately, it works for some of us. Unfortunately, it's not a durable response. So I went on this journey to try to understand, are there underlying mechanisms that we can uncover that could lead us to identify a drug that could be repurposed for this disease? And so I started conducting research in the lab and started an organization called the Castleman Disease Collaborative Network. And I began banking my blood samples every few weeks in the freezer with the belief that if I were to relapse, that I would need to have those samples so I could figure out what was happening in my disease. And so I stored peripheral blood mononuclear cells and also serum as well as lymph node tissue, and unfortunately relapsed in the midst of doing this research, in the midst of banking these samples. But fortunately, now I had samples that I could go back to, because thankfully chemotherapy, those same 7 agents saved my life for a 5th time. But I knew that it was unlikely that I could survive another one. So as soon as I got out of the hospital after spending about a month in critical condition, I went back to those samples. And what I discovered first by doing some really simple cytokine panels where we measured 13 cytokines is that nearly all of the cytokines we measured were through the roof when I was sick. And in this particular visual, the gray bar represents when I was sick. And all of my cytokines go through the roof. But what was really interesting is that 2 particular cytokines, vascular endothelial growth factor and soluble IL-2 receptor, began to rise in the weeks leading up to relapse. And so to me, that suggested that maybe VEGF, this angiogenic factor, and soluble IL-2 receptor, which is a marker of T cell activation, maybe they are playing a pathogenic role in my disease. If they are paralleling and almost leading, preceding disease activity, then maybe they're playing a role in pathogenesis. So I went to those PBMCs that I had been storing, and I ran flow cytometry on them, and I confirmed, in fact, there were increased proportions of activated both CD4 and CD8 T cells in my case. And then also what I'm showing here is that as we expanded this out to additional patients, first a total of 3 and then a total of 10 other patients, we find repeatedly increased CD4 and CD8 T cells. And again, we hypothesized that because of the soluble IL-2 receptor. And so now we were starting to develop a bit of a model for how we were thinking about maybe targeting this disease. So there were clearly a number of elevated cytokines and growth factors like VEGF, and notably some really strong T cell activation. So the question became, do we target one of these things with a drug? And of course, there are VEGF inhibitors like bevacizumab. There are targeted cytokine inhibitors, and of course, there's ways to deplete your T cells. But we wondered, could there be some sort of final common mechanism that's involved in all of the features that we're picking up in my particular samples? And so I went back to the serum samples and ran serum proteomics, measured over 1,000 analytes in my blood for multiple time points. And we confirmed the T cell activation markers, but we also got a potential signal for mTOR signaling. So there was an enrichment of the proteins that are expressed when the mTOR signaling pathway is turned on. mTOR, of course, is a critical pathway for cellular proliferation and also for T cell activation, as well as VEGF production. So we began to think maybe mTOR could be that common pathway that's involved in the cytokine production, the VEGF production, and then also the T cell hyperactivation based on the enrichment of this pathway from mixed work. And so we hypothesized it could be mTOR. I went back to that lymph node that I'd had resected during my last relapse. And first, I studied a number of normal lymph nodes, and we were looking to see if mTOR was activated. So we used a really good marker that's very stable and reliable called phospho-S6, and looked first at a bunch of normal lymph nodes to see just how much phospho-S6 or mTOR activation is present in a typical lymph node. And this is a representative image of a typical lymph node where there are certainly some immune cells in the lymph node that do have mTOR activation, but the vast majority of cells stain negative for mTOR activation. They're blue in the image. And then I stained my lymph node, and I was just blown away by the tremendous amount of mTOR activation. And so we hypothesized that mTOR would be active based on the serum proteomic signature, and now we really were seeing right in front of us that there was just tremendous mTOR activation. So basically, so based on this information, I thought that maybe using the mTOR inhibitor like sirolimus, or it's also referred to as rapamycin when used in the lab, that maybe that could be an effective treatment for my disease. And so I began testing on myself over 9.5 years ago. It's been 116.19 months that I've been in remission. And I say 116.19 months because I know that I can't round up. I don't know how long this drug is going to work for me, but I also refuse to round down. I'm so thankful for every fraction of a week, a month, a day, a year of remission that I've had, and of course, so thankful for the other patients that are benefiting from this treatment. So the New York Times called this "Doctor Cure Thyself," which I think is a bit of an overstatement. I think it should be doctor helping himself a little bit right now, and hopefully for a lot longer. I've, as I mentioned, been in remission for 9.5 years, where I joined the faculty at the University of Pennsylvania, and we set up a center called the Center for Cytokine Storm Treatment and Laboratory, or the CASTLE, where we do this deep immune profiling of hyperinflammatory diseases and seek out existing drugs that could potentially be repurposed to treat patients. I also wrote a book about my journey called Chasing My Cure. And during this remission, we began testing sirolimus, this mTOR inhibitor, and other Castleman's patients. The first 3 patients that we treated had really remarkable responses. We published that in the Journal of Clinical Investigation, and that led us to launch a clinical trial that we currently have underway that's NIH funded of this mTOR inhibitor in treatment refractory IMCD patients. So I shared this for a couple of reasons. One, to highlight an example where this sort of deep mechanistic work can result in therapeutics and treatments for patients in a matter of weeks. I made the discovery around mTOR being increased in Castleman's within about 2 weeks of being on an mTOR inhibitor, and of course having my life saved. And I really want to emphasize throughout this talk just how critical it is to do the laboratory validation work that all of you do to move these sorts of early insights into full clinical trials and really life-saving treatments for lots of patients. But just to give another sort of framework, we know that sirolimus inhibits mTOR. We know that mTOR is a good prophylaxis. It's actually FDA approved to prevent kidney transplant rejection. That's why it was available. That's why I could get it prescribed to me. We also know that it can be repurposed for my disease, Castleman's, as well as lymphangioliomatosis. It's also used for kidney cancer, highlighting that hitting one target, that target can often be involved in many diseases. So I'm sure many of you are wondering, what about other Castleman's patients? And we've been able to definitively demonstrate that in a number of different cohorts that mTOR signaling is increased quite uniformly across IMCD patients, regardless of what readout we use or regardless of the cohort or even the methodological approach. We find repeated increased mTOR activation. So this really has solidified that mTOR is a critical therapeutic target and mediator of IMCD. And of course, we know that sirolimus inhibits mTOR. And so this is the kind of triplet that we like to see in the work that we do. Disease, target, drug. And that's really so important to be able to help patients as quickly as possible establishing these links and of course doing the work with all of you to validate them. So I'm sure many of you are wondering, well, maybe mTOR signaling is increased across all diseases that involve enlarged lymph nodes and lymphoproliferation. We actually looked into that and demonstrated that mTOR activation is increased in IMCD compared to all of the other disorders involving large lymph nodes that we studied, except for autoimmune lymphoproliferative syndrome, which is actually treated with sirolimus. So the only other disease with mTOR activation to this level is actually also treated with an mTOR inhibitor. We also did some functional work in the lab where we took peripheral blood mononuclear cells from Castleman's disease patients and stimulated them with two different cytokines, one IL-6 and then later on interferon beta. And what we found is that there was increased mTOR activation with equivalent doses of cytokines, suggesting that there's this hypersensitivity to mTOR activation upon stimulation with cytokine. And again, both in IL-6 and also the type 1 interferon, we saw this basically hyper mTOR activation phenotype. So I shared a bit earlier about our center at Penn where we do this kind of work repeatedly. It's now been a total of 16 times that we've taken a drug that's developed for one thing and found another use for it, thanks to funding from the NIH and also from the FDA and a really amazing group of collaborators where we bring in both computational approaches but also laboratory studies to take drugs directly into patients. Each of the 8 patients photographed here are all alive today because of a drug that wasn't made for their disease. It was made for some other disease, but we discovered it could be repurposed for their disease. And they're alive and well, and there are many, many more that are also benefiting from this repurposing of drugs. So I wanted to share one example, one of my favorite examples, and that is around angiosarcoma. So back in 2013, there was a paper that looked at PD-1 or PD-L1 expression in a number of sarcomas, and buried within that paper is a table that demonstrates that the few patients with angiosarcoma that had PD-L1 expression measured had increased PD-L1 expression. I should share that angiosarcoma is a horrible form of cancer, particularly when it's metastatic. It's uniformly fatal within 6 to 12 months when it's metastatic. And so in 2016, we had a patient come to our center who was failing to respond to existing approved therapies. And all we did was look back at the world's biomedical knowledge and uncover this link that had already been made by researchers, that there was increased PD-L1 expression in the few patients that had been studied. And so we stained for PD-L1 in this patient now 3 years later in 2016, and we found blazingly positive PD-L1 expression. And so we were able to obtain a PD-1 inhibitor, pembrolizumab, and treat this patient for the first time ever that we're aware of with a PD-1 inhibitor. And he's been in remission for over 7 years now. And really importantly, based on this anecdotal improvement, a number of other patients have been treated with pembrolizumab, and now it's actually recommended as standard of care by the NCCN, and there are a number of clinical trials underway. But this goes to show that uncovering the mechanistic work that was done previously, confirming a patient, getting it into a human with a horrible disease, demonstrating its benefit, has actually been able to completely change clinical practice for this rare cancer without actually having a single clinical trial completed, because the natural history is so poor and so bad, that when you do turn things around, it's really, really, really obvious. So my center pad also leaned quite heavily into COVID. Early on, it became clear that there were a lot of drugs being utilized or attempted for COVID-19. We were shocked that within the first 3 months of the pandemic, over 100 different treatments were either given to patients off-label or studied in clinical trials. So we thought, man, if there are going to be over 100 drugs tried for this virus, why don't we have a central way to track which ones look promising, which ones look less promising, and let's not just rely on basically scientists to have to read 27,000 papers around all the treatments being utilized for COVID-19. What if we could utilize artificial intelligence to pull out which drugs were working, which drugs were not working, the clinical trials that needed a human to actually review them versus the clinical trials that could be fully automatically reviewed. And so we did this thanks to a grant from the Parker Institute for Cancer Immunotherapy, and in a partnership working closely with our friends at NIH, we built this central resource that truly served as a real-time tracker of all the drugs being utilized, and we applied a grading system so that every single drug got grades for how either promising it looked, meaning that more trials are needed, or how effective it is based on the trials that were out there. And it was really exciting to be a part of it, and it was great to be able to have it so vastly utilized. Let's see. So one of my favorites in terms of repurposing for COVID was a drug called baricitinib. It was identified from utilizing biomedical knowledge graphs and applying artificial intelligence to these biomedical knowledge graphs. Many of you all will be very familiar with biomedical knowledge graphs, but you can imagine every drug, disease, target, gene, protein, all being in one two-dimensional space. And each one of those only listed once. So IL-6 is only on the knowledge graph in one place. But every disease that involves interleukin-6, every drug that modulates IL-6 will have an edge connected to that IL-6. So it's a way to look for connections across diseases, drugs, targets, mechanisms. And through utilizing AI on top of these biomedical knowledge graphs, it was discovered that baricitinib appeared to hit multiple targets that appeared to be playing a role in SARS-CoV-2. And many of you all will be familiar with the fact that baricitinib went on to demonstrate efficacy across multiple randomized controlled trials, and then became approved for the treatment of COVID patients that were quite sick. This is a case where we're highlighting that the same drug can actually hit multiple mechanisms, and through multiple mechanisms can treat multiple diseases. So this begs the question, how many cures are sitting on the pharmacy shelf? Excuse me. Like sirolimus was just waiting to be uncovered for Castleman's, like baricitinib was waiting to be uncovered for COVID. Particularly when the data that can link these drugs and diseases really hide in plain sight. They're available in the world's biomedical knowledge. In many cases, they're actually in these biomedical knowledge graphs. So just to give you a sense for to frame this, so there are many drugs already have data supporting their use in other diseases, but they really aren't being pursued due to systemic barriers. So there are about 3,000 FDA-approved drugs that are approved for about 3,000 diseases. And there's another 9,000 diseases that don't have a single FDA-approved therapy. And so, of course, many of those 9,000 diseases are either being treated off-label with an FDA-approved drug, or there's some sort of mechanistic link that would make one believe that a drug like sirolimus might work for a disease like Castleman disease. But unfortunately, the work hasn't been done to clearly establish it as a treatment. And that's really for a few reasons. One, that there's never been a central database that tracks all of the drugs and all the diseases that they're likely to be able to treat. Secondly, there are insufficient incentives for drug companies to repurpose drugs. And importantly, over 80% of FDA-approved drugs are already generic, which means by definition that over 80% of drugs have close to zero R&D or research being spent on those drugs. So 80% of the drugs that are at your CVS that have the potential to be utilized today or tomorrow to save lives are not being studied because there is no financial incentive for companies to pursue them. And finally, no organization has been responsible for ensuring that FDA-approved drugs are utilized for every disease possible. The FDA has the opportunity to say that they'll approve it or not approve it based on the diseases that are presented to them by the manufacturer, but there's never been an entity that's had responsibility for making sure the drugs are fully utilized. About a year ago, to really try to address these major gaps, I launched a nonprofit organization called Every Cure, along with my co-founders Grant Mitchell and Tracy Sikora, which is a nonprofit on a mission to unlock the full potential of each and every drug to treat each and every disease possible. So not just the first few diseases that a drug is developed for, but all of the diseases that can potentially be impacted by a single drug, and really taking end-to-end ownership from, okay, if the drug's approved for one thing, let's help to do the work to make sure that it gets utilized for all the other things. That's utilizing artificial intelligence to make the connections. It's working with communities like all of you to do the validation work to say, I really believe this drug works in this disease area. And then it's getting it into patients rapidly to make sure that we help patients with the cures that are within reach. So we combined together NIH-funded knowledge graphs from the translator program, along with proprietary data, and applied these AI algorithms to come up with a single grade for the likelihood of every drug to treat every disease. Now, there are about 3,000 FDA-approved drugs, and there are about 12,000 human diseases, which is why there's 36 million scores. So if you were to try every drug to treat every single disease, you'd have 36 million shots on goal. So really the underlying framework are these biomedical knowledge graphs, which were actually funded by NCATS. Layered into that is data from companies like Elsevier that have donated data towards us that we integrate into the biomedical knowledge graph, along with real-world evidence from companies like Atropo, again, integrated into the biomedical knowledge graph. And the last piece, something we haven't done yet, but we're really excited about the potential to work with pharmaceutical companies to understand from them what were the additional uses of their drugs that are now generic that they didn't pursue, but that there were clear insights that could suggest it could work. And so the output is this 36 million cell heat map, where the drug disease links that are strongest are in this particular case, you can see our darkest. This is actually our first version of the heat map. That actually is all 36 million scores when we ran it for the first time ever in March. We utilize the scores, and we utilize what we consider ground truth, which is whether a drug actually worked in a trial or not, to help to tune the algorithm and to validate the grading system. But you can imagine one particular drug disease link has a score of 0.93. That's a really high score. We score from 0 to 1. So that really suggests mechanistically there's reason to believe that drug might work for that disease. We plan to make all the data publicly available. And then, of course, that's where we get into the next step, which is where you all are so important, and that's evaluating the most promising hits and making sure that those drug disease pairs that we think will work, we actually evaluate in the laboratory, and then we move forward the most promising ones into clinical trials and make sure that they can be optimally accessed, both that there's treatment coverage, but also the physicians utilize them. And in some cases, we will go for an FDA approval, but not in all cases, because the drugs we're talking about are all already FDA approved for one thing, which means that they can be used for other diseases, whether or not the label has been changed. 20% of all prescriptions every day are for drugs that are being utilized off-label, so for diseases they're not approved for. As long as there's a very robust clinical trial demonstrating that the drug works, that's typically sufficient to change guidelines and to ensure coverage, especially if the drug's cheap and generic. And so this is a call to action for all of you. We are very good at making these computational predictions, but we really need to partner with groups like all of you to make sure that the drugs that look most promising to save lives in particular diseases can be modeled in systems with all of you. And we would love to create a system so that these top priority hits can be funneled to you all to do the model testing, and then we can rapidly get funds in place to perform clinical trials and really change clinical practice in a matter of years as opposed to decades. So I don't have tons of time to go into the underlying machine learning algorithms that we utilize, but basically we have 5 different AI algorithms, and one of my favorite ones was developed by a collaborator at Penn State named Dave Kozlicki, and that's where the algorithm looks for patterns across the biomedical knowledge graph, and we train the algorithm to say, this pattern of this drug to this disease This is a case where the drug actually works in that disease. It's a known positive. Find patterns across the rest of the biomedical knowledge graph, like the known drug X treats disease Y, and then give us a score from 0 to 1 to tell us how similar every other drug disease link is to the known positives. And then we also utilize a similar or a slightly different algorithm to predict the exact mechanism for why a drug may have gotten a high score. So we ran our first ever query back in March. I mentioned with our query collaborator, Dave Kozlicki. We got the 36 million scores. We were really happy with the distribution of scores. The top hits were drugs that we know work for those diseases, so we were happy with that. And we're really excited. We did some targeted looking into Castleman disease, as you can imagine, and we were interested to see adalimumab, which is a TNF inhibitor, was the #1 predicted drug for Castleman's. It had never been used before for Castleman's until right around that time when we used proteomics data to identify TNF as a potential target, and actually had just treated a patient for the first time ever with adalimumab, and he's been in remission for over 8 months now. And so this for us was really exciting to see the algorithm predict adalimumab as the #1 drug, and for a patient to receive this drug for the first time ever and to now be in remission for over 8 months. So this was really, really exciting for us. These are a couple of pictures of Alan with his young daughter. Alan was getting ready to go into hospice right before we gave him adalimumab. So what we're able to do with these algorithms is we can get away from the old world of looking for the lowest hanging fruit in a single tree, where you pick a single disease and you say, I want to repurpose a drug for it, to really looking for the lowest hanging fruit across the whole forest. You can say, I'm going to come up with a score for every drug versus every disease, and I'm not just going to focus on one disease. I'm going to look across the whole forest to find the lowest hanging fruit drug, and I'm going to go after that drug, because we're disease agnostic, we're drug agnostic. We just want to make the greatest impact possible. We've got amazing partners, our foundational funding partners at the top of this screen, but a number of other partners all throughout this particular slide. So along the way, we received a lot of support from folks from across healthcare and even beyond healthcare, including President Clinton, who read my book a few years ago and has become an amazing supporter for the work that we're doing. He's helped to elevate what we're doing and really generate a lot of awareness, which has led to a lot of families and patients reaching out to us over the last year about, well, what about a drug for my child, or what about a drug for me? And this is really what drives us, and it gives us the sense of urgency for why I'm so excited to chat with all of you, because there's so much unmet medical need. And at the same time that there's all these patients suffering like those on the screen, there's also all these drugs at your neighborhood CVS that could potentially help them. And in many cases, the data linking them already exists. We just need to confirm those links, and we need to work with you all to validate them in the lab, and then we can treat patients like Baker, Lisa, and Raj. So my challenge to you all is how can we efficiently advance these treatments into model systems so that we can determine which of these look most promising. And bottom line, I've already gone through this, lots of suffering. We can rapidly identify candidates, but validating them with all of you in model systems is critical to de-risking clinical trials. So how can 3D cellular models help to accelerate the evaluation of repurposing candidates? What systems can we put in place to connect our top AI-generated hits with leaders in your field to make sure that we can rapidly get them in cellular model systems? And are there other gaps that are getting in the way of rapid advancement that I'm not thinking of, that we haven't been thinking of? And hopefully this workshop today will help to do that. Of course, you all, your model systems are so important for new drug development, and I hope you continue to work on new drug development, but I really hope you'll work with us on utilizing old drugs in new ways. These are some of our team members and our advisors. And of course, I want to close with a special thank you to our scientific partners, including those on the top of the screen, and all of those that have been a part of this effort. And I really hope you all will join this effort with us. And you can go to everycure.org to learn more. And please do email me if any of this seems interesting to you. Thank you all so much.
Thank you so much. Thank you so much for your inspiring talk. So we had a few questions, and so maybe we could take one. One of them was, in general, how can rare disease organizations engage with Every Cure?
Sure. So one way is that we can set up a process for you to donate your data towards our biomedical knowledge graph. As we shared earlier, the foundation of the knowledge graph has been funded by NCATS, but we're layering in significant additional data. And so if you have registry data on drugs being utilized off-label, if you have mechanistic, proteomic, transcriptomic, genomic data, you can work with us and we can integrate that into this new instance of the biomedical knowledge graph. So it's particularly good at predicting scores for your disease. And then we do plan to make all these scores publicly available.
Great. There was a couple questions related to your AI model. For one was, are you planning to build in genetic susceptibility? And the other one was, are the algorithms you developed freely available, and how would we be able to use them to search for potential drugs for a specific disease?
Yeah, one of the things I'm so excited about the knowledge graphs that we're utilizing is that they are sort of data agnostic, meaning that if there's data that could link together a drug to a protein and a protein to a disease, then it can be embedded into the knowledge graph. So certainly, there's a lot of genomic data that's already in the knowledge graph, and we're eager to partner with others that have non-public data that we can integrate in. The knowledge graphs that exist right now are completely sourced from public data, and what we're excited about is integrating in private data from companies like Elsevier, but also from researchers and disease advocacy groups, so that way we're really supercharging the biomedical knowledge graph. So yes, genomic data will be in there. And then in terms of access to these algorithms, the 5 algorithms that we're utilizing right now were all funded through the NCATS translator program. We're building sort of, I would say, like a super algorithm that integrates the strengths of each of those 5. We plan to make that super algorithm publicly available. And we also likely will make like a super algorithm V2 that's utilizing the most cutting edge new approaches as well. And so, yes, our roadmap and our game plan is to make all these algorithms available.
Okay, great. And then there were a couple kind of related actually to your mTOR talk. So they were wondering if you know how the mTOR activation actually induces the cytokine release. And then there was a question on if you had tested other mTOR inhibitors and what they were.
Yeah, we think about mTOR more as a mediator of hyperinflammation, and so it's not that the mTOR is driving the cytokine production. It's that it's that when there is cytokine production, it mediates hyperinflammation afterwards and lymphoproliferation afterwards. So you can imagine a lot of things can stimulate some cytokine production. If you have equivalent cytokine production in one person or another patient, then you have this overexuberant response to it. We think mTOR mediates the overexuberant response as opposed to being the driver of the initial cytokine production. And then in terms of other mTOR inhibitors, we have not utilized other mTOR inhibitors. Just an important comment to make is that when we first made the discovery with sirolimus for me, I received a lot of external pressure to try to study an mTOR inhibitor that wasn't yet approved, or at least a few that weren't approved, or ones that were still on patent, because drug companies had money to fund that kind of work. And I think that would have been reasonable to do that. But I was very fortunate to get NIH funding to do the trial and to do further studies. And I was really excited to use an old generic drug that I knew has been around for decades. So I knew its safety. It's an inexpensive drug, so I knew it would be accessible. And it was the one that had actually worked already in patients, as opposed to utilizing a different molecule that's likely going to be expensive if it ever gets an approval, where you don't really know the safety profile. But I do recognize most of us researchers usually have to go that route, where we work with the company because they actually have money to fund the work. But I was so thrilled I could get NIH funding and I could go with the old cheap generic drug, which has turned out to be effective. I should also mention it's effective in about 1/3 of patients. So we still have about 1/3 of patients that don't respond to either IL-6 blockade or to mTOR inhibition, which is why we keep pushing all day, every day.
Great. So I think we will end there. There's so many questions. There are more for you, David. Everyone was very inspired by your talk and everything that you're doing, and really to kick off this of how non-animal models, right? What are the ways that we can actually work together using non-animal models to advance this, faster for patients also. So thank you so much.
Oh, thank you so much, Liz. Have a great day, everyone. Thank you for all that you do.
Thank you. Bye. Okay, so let me share my screen. Can everyone see my screen? Yes. Yes. Okay, great. Thank you. So we're going to switch gears a little bit. And again, as I mentioned, my name is Elizabeth Ottinger, and I'm the acting director of our therapeutic development branch. And I'm going to provide an overview of NCATS activities that relate to the topics of this workshop on non-animal model approaches for rare diseases. So as Joni mentioned in her opening remarks, the current mission of NCATS is to re-engineer the translational pipeline to remove those long-standing bottlenecks in both the preclinical and the clinical stages of the process. As you know, the majority of drugs fail in development, as you can see on the left-hand side, due to the lack of efficacy or safety issues, and that's in spite of the large investments in discovery and development. So this highlights that the lack of clinical predictability of the current tools being used, such as 2D cell culture and animal models, may not be as predictable as we would want. And so one approach to improve the predictability of the drug discovery and development pipeline is to utilize the more physiological, relevant non-animal models that we hope to develop. So recently in the US, the FDA Modernization Act 2.0 was signed into law at the end of 2022, and it allows for alternative to animal testing for the purposes of drug and biological product applications, which can include cell-based assays, microphysiological systems, bioprinted experimental models, or computer models. And there are different types of non-cellular based models, ranging from 2D spheroids on the left, which are cell aggregates, to 3D organoids, which are made from iPS cells or embryonic stem cells by a process of differentiation and have tissue-like morphologies. Then we have 3D scaffold systems that are usually assembled using tissue engineering methods. And as you go more in complexity, you can see you can increase physiological complexity and increase the complexity and the features of these as you go from the spheroids to the multi-organ chips. So there are non-animal models that are already being used in different stages of drug discovery and development, and this slide outlines some of the ways that they can be used to support the different stages. For example, in early discovery, they can be used for compound screening and lead optimization and the selection of the best candidates for preclinical development. And I think, as David mentioned, that they could also be used in early discovery for the selection of those best candidates and then their validation of what could move into the clinic, especially for repurposed molecules. Okay. So we've been exploring how these models are being used to accelerate the treatments for rare diseases by doing a landscape analysis of the literature. What we found so far is that we were able to identify about 220 articles since 2010 using PubMed and Google Scholar. But again, these are for rare diseases, but this work is still in progress. And for now, I just want to let everybody know that we have excluded rare cancers. And as you see here, the preliminary analysis shows that on the left are the organ-specific microphysiological systems for each organ type and that have been studied. And as you can see, the nervous system has the largest numbers of studies, followed by respiratory and gastrointestinal. In addition, as you can see from the literature, organoids are the most widely used microphysiological system, and even across organ types, as seen on the right. We also asked, what are the most studied rare diseases using these models? And the top 15 diseases are shown here, and you can see that the disease that has the most studies is cystic fibrosis, and we're very lucky to have Professor Beekman in session 1, who will tell us more about this work on cystic fibrosis. And many of these rare diseases have clinical manifestations in different organs and tissues, and multiple models have been developed for more than one organ in one disease. On the left is a representation of cystic fibrosis, with the green ribbon showing studies in several different organ types, such as the GI, lung, kidney, liver, and pancreas. And on the right, you can see, at least from the literature studies that we did a landscape on, that other diseases, the models for different organs are listed. So there are other diseases with models across different organ systems that are listed on the right. And so I'm going to now discuss NCATS' approach to advancing translational science through collaborations by forming partnerships with both public and private organizations to leverage technology platforms so that we can address the gaps and bottlenecks to enable more therapies for more diseases quicker. So again, we take this approach, as Joni mentioned also, that we're trying to advance translational science and to see how we can actually work with the community to do this. Intramurally, NCATS has a scientific and operational infrastructure that spans from target discovery all the way through to IND enabling. And as I mentioned, NCATS forms partnerships directly through different external programs, such as the Office of Special Initiatives, the Division of Rare Disease Research Innovation, and the Office of Drug Development and Partnership Programs with the biomedical community. And I'll briefly highlight some of these programs in the next few slides. At the heart of our intramural labs is the Research Services Core Group, led by Dr. Sam Michael, which enables unique capabilities for lab automation, automated cell culture capabilities, and compound management, including the automated protocols for iPS cell differentiation and scale-up production of iPS-derived cells. We also have our stem cell translational laboratory led by Dr. Carlos Tristan, who is dedicated to develop innovative stem cell technologies, including developing efficient protocols for cell differentiation and characterization. Our 3D tissue bioprinting lab is a dedicated lab to the biofabrication of 3D models for drug screening, including assessing their clinical predictability. We also have a therapeutic development branch that enables INDs and first-in-human proof of concept studies through team and technology-based strategies and platforms. And that includes biology and chemistry for drug repurposing and lead optimization, and has a strong focus on rare and neglected diseases. Our informatics group has three main areas of work. One is building standards and knowledge sources. The second is the translational data analytics, and three is scientific computing and research. Two of the main areas of focus that you can see on the right are metabolomics and multi-omics, from data collection all the way to interpretation. And two, also working on rare diseases and knowledge graphs for drug repurposing and other efforts, such as the Rare Disease Alert System, which alerts people to new information, whether clinical trials or literature articles related to diseases. And we also, as we said, there are extramural programs within NCATS that work with intramural and engage with the biomedical community. So ASPIRE is a new program which stands for a specialized platform for innovative research exploration, which combines automated synthetic chemistry with AI and machine learning to map biologically active chemical space. And this is a collaboration program to facilitate translational research, again, between intramural scientists and the extramural community. And finally, we have the Tissue Chip for Drug Screening program, which aims to develop and use the tissue chip technology for safety and toxicity testing, and personalized medicine applications. disease modeling here, and then personalized medicine applications. This is an external program, so there have been several rounds of funding opportunities, as you can see on the screen here. And as the program has developed and expanded over the years. And we have several speakers here today also that have participated in this. We have Dr. George Trusky and Dr. Clyde Svensson. So we really look forward to them talking in the next sessions and learning more about tissue chips and 3D models and drug screening. So this brings us to the NCATS concept of i3D-Rare and why we're all here today. And with the objectives is to develop and operationalize a platform of validated rare disease 3D models using IPSs to accelerate the development of personalized therapeutics for rare diseases. This workshop is a landscaping exercise on the gaps, bottlenecks, and challenges to determine where NCATS could really play a catalytic role to engage all of you for candid feedback from the community. So we really look forward to everyone providing their both positive and things that we should be looking out for as we embark on trying to develop this platform. And I wanted to go over the agenda briefly. There are 3 sessions that are 2 hours each. They will have 4 speakers in each session with 20-minute talks, and the rest is for panel discussion. And this is where we really would like engagement from the community as we work through the different gaps and bottlenecks in developing these non-animal models. There will not be questions after each speaker, but the audience can add their questions, as we said before, into the Q&A, and that you should see again at the bottom of your screen. And then the moderators will try to incorporate those into the discussion in each panel. And we're going to take, we're actually a little bit ahead of time. We're going to take, it'll be almost a 15-minute break. But before we go, I'd really like to acknowledge the organizing committee that you see here. And that we have worked to put this together. This takes a lot of time and effort, and we really appreciate everybody's input, including Dr. Anton Simeonov, who is our scientific director of our Division of Preclinical Innovation. And I want to give a special shout out to thank Dr. Dara Zong, and she has been the workshop organizer, working with all of us, all of the logistics, contacting everybody. So we really want to thank her for all her time and energy for this really important workshop that we're having today. And we'd also like to thank Abigail Grossman for helping with the Zoom workshop logistics, and Dr. Richa Lomash and Dr. Dara Zong, who have been instrumental in carrying out the literature landscape analysis and the preliminary analysis that you saw today. So I welcome everybody. We look forward to a really engaging day for all of us to learn. And we look forward to hearing from everybody through the different sessions. And we can now take a break. And I think we're going to have an extra 5 minutes for our break. So we will meet back here at 10 o'clock. And thank you. Bye, everyone. See you at 10.
All right, so it's 10 o'clock and we'll get going so we can keep things on time. So good morning, afternoon and evening, and or night, depending on where you are. And thank you all for joining this workshop again. My name is Marc Ferrer, and I'm the director of the NCATS 3D Tissue Bioprinting Lab, and together with my colleague, Dr. Carlos Tristan, who is the acting head of the Stem Cell Translational Lab at NCATS as well, will be the moderators for this session. This is session 1, and the title is Building Experimental iPSC-derived 3D Cellular Models to Advance Translational Science for Rare Diseases. As Liz mentioned in the opening remarks, the session will last about 2 hours. So we have 4 terrific speakers who will be giving about 20-minute talks. and then they will join us for a discussion panel afterwards. Again, if you have any questions, please add them to the question and answers. We won't be taking questions in between speakers, and we'll address as many as we can during the panel discussion. The bios of the speakers are in the registration website, so we'll do brief introductions so we can keep things on time. And finally, for the speakers, please stay on your 20 minutes. Carlos and I will give you a 2-minute warning so you can start wrapping things up so we can have time for the discussion, which is the part that hopefully will get a lot of ideas. So without further delays, we'll start introducing the speakers. So the first speaker today is Professor Guo Wing-Min. He's a professor of neuroscience in the Department of Neuroscience at the Perelman School of Medicine at the University of Pennsylvania. And the title of her talk is Engineering Brain Region-Specific Organoids from Human iPSCs. So Professor Ming, please share your slides, and the floor is yours.
Thank you so much for the introduction, and also thank you for having me here. Can you see my slides now?
Yes, perfect.
Okay, great. So today I'm going to talk about our recent work, mainly in engineering brain organoids or organoids in the nervous system, which allow us to model brain development, basic sciences, and also for modeling diseases, including rare diseases in the nervous system. So as you know, starting from iPSCs, we can now generate different type of organ-like tissues, 3D culture tissues, resembling different organs. And my lab is specifically interested in the brain. And over the past few years, as shown here, we actually have developed many protocols for generating different type of brain organoids. And this is region specific. As you know, in the brain, there are different brain regions, including originally from the forebrain, midbrain, hindbrain, and also other parts of the central nervous system, spinal cord. And my lab have developed protocols for generating cortical organoids, highlighted here, hypothalamic organoids, and a subregion of hypothalamus accurate organoids, as well as hippocampal organoids and choroid plexus organoids and midbrain organoids. And we are applying this different type of brain region-specific organoids for understanding the basic biology of brain development, as well as modeling developmental disorders. Now, I would like to start with a cortical organoids. And for generating this type of organoids, it basically mimicking the developmental trajectory. For example, studying the cortical development starting from neuroepithelial cells, which are primitive neural stem cells responsible for generating the whole central nervous system. And in the cortex, the neural stem cells, which are also called radioglial cells because they are distinct morphology with a long radial process stretching from all the way from the ventricle where the stem cells localize to the marginal zone, which is linked basically to skull. And this radial process serves as a scaffold, allowing their progenies to migrate along this process and populate the whole cortex. And in the development, first, there's an expansion of neural stem cells, and specifically for our brain, including the non-human primates and humans, we have additional source of neural stem cells that are localized in the outer subventricular zone, OSVZ. And it has been hypothesized that this additional stem cell population are responsible for the expansion of our neocortex. Of course, after the stem cell expansion, the neurogenesis giving rise to different type of neurons, as well as gliogenesis giving rise to different type of glial cells in our brain that ultimately form the circuitry and responsible for our day-to-day function of the brain. And generating the forebrain specific organoids is basically following this developmental trajectory. And in the lab, when we try to develop the protocol, it is with the help of 3 high school students and together with 2 graduate students. Basically, they designed a miniaturized spinning bioreactor, as you can see here, basically attach the 12 well plates or 24 well plates, which allow us to screen for different conditions and to generate different type of brain organoids. As you can see here, this white little blob brain organoids, forebrain organoids growing in this miniaturized spinning bioreactor. And the development, as I mentioned, is basically following what happens during the nervous system development in the uterus. Starting from human iPSCs, we first direct them into neuroepithelial cells by dual SMAC inhibition, and then further divide them into forebrain specific neural stem cells. And after this, it's basically on the self-organization and intrinsic properties of these neural stem cells, which give rise to neurons and glial cells and mature over time. As shown here, brain organoids can grow relatively large in size. By 100 days, as shown here, each of these brain organoids or cortical organoids can reach ½ centimeter in size. So you can clearly see them in the culture well. And to cut the long story short, with this protocol, after screening over 100 different conditions, which allowed us to generate an organoid really mimicking the forebrain development. As shown here, we demonstrate in these organoids, we can have specific neural stem cells, as I mentioned, human-specific neural stem cells localized in the right localization outer subventricular zone, which is also shown here. Also, we can show that these neural stem cells can give rise to different cortical neurons that are expressing markers for all 6 cortical layers, and with some rudimentary separation of the cortical layers. And as shown here, different markers labeling different type of cortical neurons, they show some segregation indicating an inside-out migration pattern. We can also show that the neurons generated in these organoids are functional by electrophysiology, showing here not only the neurons can exhibit glutamatergic synaptic responses, they also exhibit GABAergic inhibitory synaptic responses. We also show there's astrocytes. As for gliogenesis, there's astrocytes, although in this first protocol, we haven't seen oligodendrocyte, another glial cell type responsible for the myelination of the neurons, of the axons. And we can profile the developmental trajectory by performing RNA sequencing and directly compare to the data sets obtained from human cortical tissue and show that by 100 days in culture, these forebrain organoids essentially resemble the cortical development towards the end of second trimester. And as I mentioned, although we show there's a lot of advantages and complex development of these organoids, we also realize quickly there are limitations in our first generation protocols, is that there's no vasculature, which is coming from different developmental origins. So I also told you that these brain organoids can grow very large in size. And that means the cells localized in the center just depend on the diffusion of the nutrients as well as oxygen. However, the diffusion limits for this solid tissue is usually only about 400 micron. So as you can imagine, when these organoids grow larger than 1 millimeter, usually the cells in the center, there's a lack of nutrient and oxygen that leads to traumatic cell deaths in the center. And if you remember the distribution of this cortical tissues is that stem cells are mainly localized near the ventricle and in the center of these organoids. That means most of the stem cells are suffered from a lack of oxygen and the nutrients. And also, of course, that leads to the death of these stem cells. That's why there's a limitation in terms of a maturation because of this to the second trimester. And since I also mentioned that the architecture, cortical layer formation, is dependent on the radial process of those neural stem cells, we don't see clear segregation of the cortical layers. It's also because of the death of these stem cells. And another one is the lack of oligodendrocytes. This is also because the oligodendrocytes are born much later during the development and mature actually postnatally. And the death of the stem cells can also explain of the lack of oligodendrocytes. Of course, we have tried many different things, including to try to avoid this diffusion problems. And in the end, former graduate student Xu Chen came up with the idea, maybe we can just simply slice the organoids into big pieces to bypass the diffusion limit. How we can do that? So here shows a diagram of an organoid. When this organoid about 1 millimeter in size, Xu Yu did a slicing by first embedding this organoid in an agarose gel. And what he did is to simply make 2 cuts, cut the top off and cut the bottom off. So which left him a very thick organoid slice, about 500 micron in thickness. The reason for choosing 500 micron is that it can maximally maintain all the stem cells, as I mentioned, in the center. And around this time, when they reach 1 millimeter in size, the stem cells, the thickness is about 200 micron. So this way, we can maximally maintain all the stem cells and allow them to further develop. As you can see, as showing here, now because of the diffusion can reach all the way to the center of the organoids, allowing further development of these organoids. However, you can imagine because of the highly proliferative nature of these stem cells, it can reach the diffusion limit very quickly. So she decided to do this repetitive slicing every 4 weeks, allowing this organoid to further development. And the result is actually quite striking, as shown here. This is 7 days in culture. You can see the complete structure of an organoid, maintaining the stem cell layers very well. And by 100 days, you can see the expansion of the iSVG, OSVG neural stem cell layer and cortical plate, showing in green here is expansion. And if you compare the void organoid without slicing, this is quite a remarkable achievement. And the end result is with this slicing technology, we call sliced neocortical organoids, we can show that these organoids can allowing us to further mature the organoids with specific cortical layer segregation. And also these neurons exhibit distinct axonal patterns, also resembling human brain development up to the 3rd trimester now. And we also show that now with this snow organoids, we do see the generation of oligodendrocytes, as well as the presence of a type of human-specific exercise that is lacking in the traditional animal models, such as rodents. And also, we can use this model system. We also identified the molecular mechanisms regulating this layer segregation, which involves the Wnt beta-catenin signaling. And also, we use this model system to study disease in psychiatric disorders using patient-specific iPSCs. I'm not going to go into details into this, but I would like to rather to show you how we can apply these model systems to study, to model rare disorders. One example is the Prader-Willi syndrome. Prader-Willi is a rare complex genetic disorder, occurs in approximately 1 of 1,500 births, is caused most of the time by deletion of part of the chromosome 15, and under most conditions, this is inherited from the father because of distinct methylation status of chromosome 15. And the most common symptoms of Prader-Willi syndrome are eating behavior problems, as well intellectual disability and obesity. This is mostly attributed to the dysfunction of the hypothalamus. And currently, there's no cure for Prader-Willi syndrome, although some of the symptoms can be treated with hormonal therapy. How can we model this Prader-Willi syndrome? So the hippocampus, unlike what I have showed you, the cortex, which is a layered structure, hypothalamus is a more ancient brain structure, meaning that the cells are organized in nucleus, a cluster of cells, rather than layered structures. Specifically, one nucleus called Accurat's nucleus or ARC is localized in the medial basal hypothalamus, and this structure contains neurons controlling the food intake and the metabolism. So we decided to try to develop a protocol for generating accurate nucleus-specific organoids. The protocol is shown here by strong inhibition of Wnt and strong activation of sonic hedgehog pathway, which allow us to generate accurate specific organoids. And as shown here, it turns out to be a round structure. It didn't show the layered structure as the cortical organoids and grow bigger in size. And I would like to summarize what we can achieve using these arco organoids. Basically, we show that using the wild-type iPSCs to generate arcos, it can recapitulate the cell type diversity as well as the molecular properties when you compare to human hypothalamic accurate nucleus. We further generated arcos from 2 patient-specific iPSCs, one with a major deletion in the chromosome 15, and one with a minor deletion within the chromosome 15. And both patients are Prader-Willi syndrome patients. And using Prader-Willi syndrome specific arcos, we show that they exhibit aberrant differentiation, reduced number of POMC neurons, which is responsible for food intake control, as well as defective leptin responses, a hormone directly involved in food intake and metabolic responses. In addition, we also show that disease-specific arcos exhibit transcriptomic dysregulation, which is indicative of cellular differentiation, the cellular phenotype exactly as what we observed, as well as metabolic deficits, and also increase the inflammatory responses, very similar to postnatal hypothalamus from the patients. Thus, with our results suggest that patient-specific ARCOs can be used as a
2 minutes, 2 minutes, Dr. Ming.
Yes, promising experimental models for investigating nucleus-specific features as well as disease-relevant mechanisms during early human accurate development. Now, extending our knowledge for growing brain-specific organoids, we have extended our toolbox by generating patient-specific brain tumor organoids. And as shown here, we have generated a glioblastoma organoid and show that we can generate this GBOs very quickly within 2 weeks directly from patient resected tumor tissue. And this GBOs recapitulate the histology, immunohistochemistry, transcriptome, and mutation panels when you compare to parental tumor tissues. And furthermore, when we transplant these GBOs into the mouse brain, we show that these GBOs exhibit rapid engraftment and infiltration. And these GBOs, we can also demonstrate as a proof of concept that the GBOs can be used to test targeted drug therapies because we can generate these organoids within 2 weeks, and perform the drug testing before giving patients any medications for treatment. Currently, we're also testing how we can use our GBOs to test the immunotherapy responses combining the CAR T treatment. Now, I want to show you just some preliminary results, how we can also develop chordoma tumor organoids. Chordoma is a very rare malignant bone tumor, mainly forms in the spine and the base of the skull. As you can see, there's no cure treatment for chordoma patients. And by modifying our growth conditions as growing glioblastoma tumor organoids, now we can maintain these organoids up to a year in culture. And we can show that these chordoma organoids exhibit histological features very much like the primary tumor tissue directly from the patients, as shown here, the bone structures within the tumor, and also immunocytochemical similarities when you compare to the original tumor. For example, the high expression level of brachyury or the T protein that is characteristic for this type of tumors. So
I'm sorry,
with that,
Professor Ming, we all need to be
yes,
closing.
this is my last slide.
Awesome.
We can show we can use this different type of brain organoid models to study human biology, but also understand disease mechanisms, including tumors and other neurological disorders. So with that, I would like to thank the people in the lab. This is a mainly collaboration with Hong Jin Sung's laboratory and the people working on the brain organoids and tumor organoid groups, especially our collaborations with neurosurgery. I'm sorry for passing the time and taking longer time. Thank you so much.
No, thank you so much, Professor Ming. This was great. Again, reminding everyone to add questions to the QA box, and now I'm going to let Carlos introduce the next speaker.
All right, next with us we have Professor Jeffrey Bickman from the University Medical Center, Utrecht. The title of his talk is Adult Stem Cell Intestinal Organoids for Cystic Fibrosis Research. Professor Bickman, please feel free to take it away.
Yes, thank you very much. And it's exciting to be here, and also thanks for the invitation. I hope people can see my screen now.
Yes.
Yes.
People can also hear me. Great, that's good. So I'll just move on with our work on adult stem cells, so mostly organoids derived from intestinal biopsies, which we receive from patients with cystic fibrosis, and how we use them for applications actually in drug development and precision medicine. So just some quick disclosures. And just to start off, so this is the Utrecht campus where I work now already. I have my research group for about 10 years, a bit longer. And we were lucky to be here next to the Utrecht Institute, actually. So I work here in the hospital. So we work with clinicians actually in my lab. We have a very translational focus, but we were very close to the Utrecht Institute, where Hans Klevers and Toshi Sato were developing these new culture protocols for adult stem cell for adult stem cell organoids. So we have been closely working together closely, and while I focus really on applications, I think Hans and his group have been really working on developing all these types of adult stem cell based organoids. And this started off actually with working on intestinal organoids, and then later on all these different tissues were also being developed. What I really enjoy and like about these types of infrastructures, these stem cell based infrastructures, that you can work towards a standardized infrastructure. And if we work on adult stem cell organoids, this is mostly all based on epithelial tissues, at least the cultures that are being developed from Klevers protocols, they focus on epithelial tissues, but you can really standardize this and you can have a workflow where you get biopsies from patients, develop these organoids, create living biobanks from these, and from these biobanks then you can do these preclinical studies, obviously, but also clinical studies. And you have different flavors if you think about clinical studies, but we mostly use organoids as a sort of living diagnostic to see whether we can look at patient-specific disease features or drug responses. But you might even also work with these types of systems as a living therapeutic. But this is another topic that I will cover in my presentation. The other thing I think, so standardization and having stem cells as infrastructure is really helpful. Another thing that which is really interesting is that these organoids are patient-derived. So if you think about a pretty standard drug development pathway here, from discovery to preclinical and clinical development, and then drug approval and market association. If you think about using these types of systems now, you can use them actually along the entire development pathway. So normally in vitro models are being used in the preclinical domain, where you work on drug selection or on target validation, target identification. But when you have these systems which you can also acquire from patients at a relatively easy manner and at a large scale, you can also really start to think about using these types of patient-derived model systems, not only for testing of drugs, but also for selection of patients. And this you can do within a drug development program, but also obviously for market available drugs, and then for other types of applications. And if you think about the workflow surrounding these schemes, so normally in these types of settings here, on a preclinical scale, you mostly focus on several patient samples, and you would like to do large scales in drug testing. But on this side of the slide, actually you focus far more on large patient numbers and fewer drugs actually for testing. So the development or one of the challenges on this side is really to standardize across so many different patient samples, because all these cells can respond a bit differently to culture conditions, for instance. So just a very brief slide, a simple slide on induced pluripotent stem cells versus these adult stem cells. And this is a bit different, but I think if you use these pluripotent stem cells, they really have, especially in the embryonic state or in the pluripotent state, you can grow these very well. And this is for adult stem cells, a bit depending on the type of tissue. Intestinal stem cells self-proliferate very rapidly and also for a very long time. But other types of adult stem cells are limited also in expansion. But I think the big differences here are obviously in potency. So pluripotent stem cells can be generated into many tissues, while these adult stem cells remain committed to the tissue biopsy from the organ where you get them from. And I think another big difference potentially is not so much in maybe access and complexity, scalability also perhaps a bit better for pluripotent stem cells, but depends a little bit on the system. But I think, and this was also shown in the already alluded by Professor Ming in the last presentation, when you can get these tissues, if you want to go towards patient outcomes, and the time from a tissue biopsy towards a patient outcome, which you can communicate, for instance, is something which is can be very rapid for these adult stem cell derived organoids, whereas this can be very lengthy for pluripotent systems. So just to sum up some of the differences and the similarities between these types of model systems. So we work with intestinal organoids. I think I alluded a little bit on this already. It's actually a starting point for many of these adult stem cell derived organoids culture media. So intestinal organoids were first described by Toshi Sato et al. In the Clavers lab. And then this really was the birthplace, I think, for a lot of different protocols. So we work really in a clinical setting with these types of systems. So we try to work with try to make disease models actually out of these organoids, and we developed a model for cystic fibrosis already pretty long time ago. We had some people also, I think what you can do also with this, especially if there's a large clinical urgent need, you can use these systems also to select patients for therapies. And we now are running a large personalized program or precision medicine program across Europe to see whether we can also get this into a more standardized clinical decision making scheme. And we're also working with our regulatory instances, so the European Medicines Agency, to see whether we can get qualification of these types of technologies for specific contexts of use in clinical domain. So I'll be just covering some of these elements in my presentation now. So I work mostly on cystic fibrosis, and I don't want to talk too long about the specifics of the disease. It's a rare monogenic disease, autosomal recessive, and it's as for all diseases actually. It's the main question also here, and the main efforts of the research is focused on this basic question on who to treat with what. This is not specifically for CF. It's for many of the diseases we just studied. So if you look at this question in more detail for CF, it means that for CF we have about 160,000 patients worldwide, and these people have, the majority of people have similar mutations actually. So the most dominant mutation is very frequent, F508del. It lacks a specific phenylalanine and a protein called CFTR, or a gene called CFTR. And the real challenge here is trying to understand the patient populations which have a very rare mutations or even ultra rare mutations. So the majority of mutations, there are more than 2,000 known. The majority of those are actually really rare to ultra rare. And we don't really understand how these mutations work, and it also leads to difficult to diagnose cases where it's difficult to diagnose disease, but also to understand therapeutic benefits. Another thing for cystic fibrosis has really been revolutionizing the treatment over the last decade is the development of mutation-specific pharmacotherapies. So this has been developed actually by Vertex Pharmaceuticals, also from the US. So they develop small molecules which can bind to pocket in the mutant CFTR protein and help to stabilize the conformation so that the protein can again get functionality again. And the difficulty actually of these types of therapies is to identify, especially in the context of these rare mutations, whether these mutations are whether these therapies can be functional or not. So if you look at the therapeutic development pathway for these new molecules, I think the first one on the market was 2012, ivacaftor, which worked on this little part of the pie chart of people with CF. Then we had a first generation of small molecules targeting this dominant mutation, which wasn't so efficacious by itself, so it could only be shown to be effective in the homozygous population. And nowadays we have a second generation in which 3 molecules together really help to stabilize the protein, and then this is so effective that it also works in people who have only a single copy. So the unmet need now is trying to identify how these drugs work in people with rare missense mutations, but also to see whether we can still help people to develop drugs also in during the preclinical stage, because there's still a need for additional drugs. So if you then think about these intestinal organoids and as a disease model system, so our questions already a long time ago was whether we can study CFTR function in these intestinal organoids, and if we can study CFTR function, and this is the basis of the disease, cystic fibrosis, is this then also predictive for clinical disease, not only for the intestinal system, but also beyond the intestinal system? So in people with CF, they suffer from a sticky mucus across many organ systems. So mostly they have troubles with the lungs, but also the pancreas and all sorts of other epithelial systems. If you have tubes in epithelial system and production of mucus, you see that there are difficulties. But also, not only to look at clinical disease, but also at therapeutic response. And we questioned also whether we could have population prediction or also even individual accuracy of these types of predictions. And apart from this clinical use, we also obviously try to develop our models in a more sophisticated way to support preclinical modulator development. So this was the test that we developed already a pretty long time ago. So these are all intestinal organoids grown from healthy controls or people with F508del. So there's only a single individual, and these are organoids from a single individual, but then also incubated with these new types of drugs. And what we observed is if you activate these organoids with forskolin, a chemical compound which activates CFTR and induces CFTR function, you get fluid secretion into the organoid lumen, which leads to rapid swelling of these organoids. So this is actually a sort of secretory diarrhea, which can be induced in intestinal organoids by adding cyclic AMP raising drugs. So this phenotypic response is actually really absent in CF organoids. So if there's a little bit of residual function here, and residual CFTR swelling, but the swelling you see is really CFTR dependent. And the phenotypic response also could be restored actually by incubating these types of CFTR modulators. So here we had a very simple system where we could look at intestinal organoids and just measure their difference in size in time. And this would be reflective for baseline CFTR function, but also for therapeutic response. So actually, and what's happening here is the swelling of these organoids is related to CFTR-dependent chloride input into the lumen, and this is followed by water and then consequently swelling of these organoids. So a very simple readout actually. It's it just needed some time-lapse microscopy, and it allows you to measure specifically CFTR function in an individual context in these types of cells. Well, I don't want to spend too much time here, but if you just look at assay design and assay quality, and you look at you can calculate the Z prime factor. This tells you something about the width or the separation between the maximal signal and the lowest signal, and also the variation between these types of signals. But this is really nice actually. If you are beyond 0.5, you really have a good assay for drug testing. In 96 well plates really well, and in 384 well plates it's also pretty okay. And this is all still without automation steps and no all manual labor. So it's a bit better nowadays, but it's already performing pretty good in these types of. It has a good assay quality. So if we now look at how we can use this assay in various clinical applications, we have a couple of conditions here. So one is if we can use it to diagnose CF. So if we just look at organoid swelling and organoid phenotypes, we actually measure 100% sensitivity and specificity in being able to separate healthy control people from people with CF. And these are pretty large studies. So our first study only looked at 60 organoids, but now a recent study from Kuijks also looked at 160 patient-derived organoids. So it's really actually a system which you can scale up and has actually tremendous sensitivity and specificity. So if you zoom in a bit more on the disease itself, so if you look at disease variation within the CF population itself, it's a bit more difficult because the changes in function are far more closely related to one another as compared to a diagnostic setting. But also here we can nicely see that organoid swelling associated with pulmonary function decline, but also pancreatic disease and also liver disease. And this is again a study where we use 170 patient-derived organoids and study how they swell and how swelling of the organoid in vitro relates to these clinical indicators of disease. And remarkably also, this is the in vitro test actually seems to outperform the classic in vivo test that we have to study CFTR function by measuring sweat chloride. And this sweat chloride test is very, very helpful for diagnostics, but for measuring disease variation within the disease population, it's not as precise and accurate. And obviously, we also looked at individual therapeutic response and how this can be predicted and associated to clinical response and organoids. And there we also see that if the differences are pretty large between genotypes, or if you study different drugs in different settings with different CFTR mutations, you see good relations. But if you look within a single genotype class for a single drug, it's actually pretty difficult to find these associations. So there's a lot of noise also in the organoid measurements. There's a bit of noise, but also in the clinical parameters, which probably also hamper the execution of these types of association studies. So I just wanted to end up with an example of how we use these actually now in a rare disease domain in Europe. So as I said in the beginning, the real challenge is now to focus on these ultra rare or on the people with CF who have these ultra rare mutations. So in Europe, these are the patient numbers in various countries which are being identified who still do not have access to these types of new CFTR modulator drugs. So all people with rare mutations, and these people might benefit from these drugs or might benefit from drugs in development, but they are very hard to organize and to cluster in clinical trials because they are so diverse and heterogeneous in their mutations, and they're so located geographically dispersed across the entire continent. So in total, we have about 2,000 patients with these very rare mutations who do not receive treatment at the moment. So we set up a program where we started to recruit organoids actually from 500 of these patients. So this included 47 CF centers, 16 countries across Europe. So we had all these centers taking biopsies actually from individual patients. They were all being shipped to Utrecht to an organization called Hub, where they performed all these. They generated organoids from all these 500 patients, and then we could follow up with drug screening. So just some numbers on the feasibility of this. So we shipped 502 biopsies. We had a 95% success rate in generating organoids from these biopsies, and then these were the reasons for failure, but it was mostly sample quality. If you don't take good biopsies, it's very difficult to make biopsies also. But it just shows how robust this model is, actually, this intestinal organoid model for growing organoids out of shipment across the continent. So if you then look at these types of model systems and the organoids we generated, we did drug screening for a lot of these organoids actually, and here what you see is each line is a specific patient-derived organoid, and we screen them against a specific type of CFTR modulator. And what you see, and what you also would expect, so these are all organoids with different types of CFTR mutations. You see a large proportion of people not being responsive, or you see the organoids not being responsive to drugs, but you also see that there's a fraction of organoids which start to respond to these types of drugs. And this is again organoid swelling. So currently we're now executing or starting up a trial actually in the high responders of this group. And then if you then look at the groups that was the group of patients that was selected here, we're now doing this phase 2 trial here. And if you just look at the average response of the organoid responders here, this also really much is within the range of the current market approved CF drugs. If we look at organoid responses for the current market-approved drugs, they are also sometimes a bit lower and sometimes a bit higher. So we really think that this type of model system helps you to select responders and try to organize clinical trials which have an increase in efficacy signal and also a reduced variation of the efficacy signal. And this helps to power up your trial and to possibly get to get better clinical data out of it. Doesn't mean that these people may not be responsive, but that can be trialed in a second clinical trial then. So
I'm sorry, Mr. Rickman. We're at 20 minutes now.
Okay, good. Yeah, so this is the actually the last slide. So this is actually how we do these types of well, we have an organoid infrastructure where you can do these types of studies, and I think this is but will also be alluded on by Dr. Daniel Paul later on. So we're now really focusing on how we can automate these types of technologies so we can get the throughput higher, but also more standardization. And we use also label-free feature extraction here. I think what's nice also is that we've been working with the EMA here, and actually it all comes down to standardization. They just want to, I think we have already pretty high and large patient cohorts, but they always want more, and they also want to see more and more standardization. So I think this is a big thing, and this is also something which automation can bring up. So just as a conclusion, I think what I've shown is that we have these organoid-based assays, which can even correlate with individual disease features and treatment response. It's feasible to execute really European scale programs here for drug development, but we also really need to standardize and automate. And with this being said, and the time being gone, I'd like to thank the people here also contributing, and I'll stop share now. Thank you very much, and apologies for passing the time.
Thank you, Professor Bigman. Next, I'll have Mark introduce the next speaker, and we're going to save the Q&A for this panel discussion.
So it is my pleasure to now introduce Professor George Struski, who is a professor of biomedical engineering at Duke University. And he's going to be telling us about a vascular microphysiological system model of Hutchinson-Gilford progeria syndrome. Professor Struski, all yours.
Great. Hopefully you can see my slides.
Yeah, we have, I think you have captions on.
I do. I probably, let's see if I can turn it off here.
Awesome. Perfect.
Okay. Oops, still there. Okay, hopefully that's okay. You can see everything.
I think it looks good. Thank
you. Great. Well, thank you very much for the invitation to speak. I will tell you a little bit about our work on our microphysiological system model for HDPS or progeria syndrome. Just a little bit about the disease. It's a rare autosomal dominant disease, produces an accelerated aging pathology. It's caused by a point mutation of the LMA gene, primarily a replacement of the T by a C at 1824, but there are a number of variations. The result is a truncated and farnesylated form of lamin A nuclear membrane protein, and that's known as progerin. Visibly within cells, you can see abnormal shaped nuclei. It's quite easily discerned that way, but there are many other functional variations, including gene expression changes, reactive oxygen species productions, double-stranded nuclear breaks. It affects mesenchymal lineages such as the skeletal system, particularly bone, as well as dermis and vascular smooth muscle cell. The primary cause of death is due to atherosclerosis. These children die between 10 and 15 years of age. And what happens is there's smooth muscle cell loss that leads to a fibrotic lesion with inflammation, some lipid accumulation, and calcification. So it has some bearings with adult atherosclerosis, but you usually don't see the lipid accumulation you see in adults. So there have been several different therapeutic approaches developed to treat the disease. The most effective so far has been to inhibit progerin farnesylation or prenylation by blocking with a farnesyl translation inhibitor has been the primary approach, but there have been others using zoledronate or pravastatin. A second approach has been to promote progerin degradation by blocking mTOR and then promoting autophagy. And these have been demonstrated in cell culture studies. Two other approaches, genetic approaches that look very promising and have, there's some nice animal data. First is using antisense oligonucleotides to suppress the protein expression. The other one is using adenine base editing developed by David Liu at the Broad Institute to correct the mutation. As I mentioned, it's a rare disease, maybe 250 to 400 children worldwide. The Progeria Research Foundation has done an enormous amount of work to identify patients with the disease and actually run several clinical trials. The most successful trial has been with lonafarnib, which showed improved weight gains, bone structure, a number of other symptoms, including increased vascular distensibility and an increased survival by just 1.6 years, but it was significant. So and that's actually now been FDA approved. A second clinical trial using a 3-drug combination unfortunately did not show additional benefits. And so that certainly presses us to look at alternative approaches to treat progeria. So what we've been doing has been to develop an engineered blood vessel model that we could apply for progeria because of the vascular symptoms. We developed this model over the past 10 years, supported by NCATS, and it consists of a vascular system shown on the right here, which can replicate the endothelial and smooth muscle cell layers, and using a collagen compression method to form the rigidity you need for the vessel. The first strength that these vessels exhibit is on the level of the venous system. We also have, in addition to measuring gene expression and histology, we also do a functional test, which we can measure continuously throughout our experiment to measure vasoactivity, a common arterial response. So we can treat the vessels with phenylephrine to cause constriction, and this just shows a healthy vessel constricting over a 4-week period. And then we can add acetylcholine to induce vasodilation through nitric oxide. And that has been very helpful to assess function. Now, to study progeria, we've been able to obtain both healthy cells and progeria iPS cells from the Progeria Research Foundation. The healthy cells are actually parents of the children who've had this disease. And using a protocol developed by Patch, we can first induce the mesoderm, and then we can then depending on the molecules we add, we can either produce these iPS-derived smooth muscle cells or endothelial cells, as shown here. And then we can use these in our iPS-derived blood vessels. We show that the iPS-derived cells express progerin, whereas the healthy cells do not, for the smooth muscle cells and for the endothelial cells. The iPS-derived cells exhibit many of the markers for vascular smooth muscle cells, both for healthy and progeria cells, including smooth muscle actin and calponin. The endothelial cells also express typical markers such as CD31 and CD144. We've looked at a number of others too. In addition, we've done some functional testing on the individual cell types of smooth muscle cells will constrict in the presence of a vasoconstrictor, although that is reduced in the progeria vessels. We looked at capillary tube formation by the endothelial cells, and both the healthy and the progeria cells will produce capillaries, although the structure is not quite as well developed in the progeria cells. So next we created our engineered blood vessels with either healthy or progeria cells, and we used primary cells consisting of mesenchymal stem cells and endothelial cells as our reference. And these are the vasoactivity results over a 4-week period. And what they show is that for vasoconstriction, first, the iPS-derived healthy cells function as well as the primary cells, but there's a deficit in, deficit in vasoconstriction in progeria. And likewise, when we do the vasodilation, we also see similarities between the healthy iPS-derived vessels and the primary cell-derived vessels. But again, there's a reduction in vasodilation. So there are functional deficits in vasoconstriction and vasodilation. If you look histologically, part of the reason for the deficit is that there's a reduction in the smooth muscle cells, as shown here by staining for alpha smooth muscle actin and the myosin heavy chain. And as expected, we see far fewer smooth muscle cells. We also see the pathological deposition of collagen 4 and fibronectin in the extracellular matrix of the progeria vessels, but not in the healthy vessels. Reduction in some of the endothelial genes, such as von Willebrand's factor, and then the expression of progerin in the progeria vessels. And we quantify these, the immunohistochemistry. We also stained by H&E, and you can see the loss of smooth muscle cells in the progeria vessels. And most interestingly, we did see calcification by staining for alizarin red and apoptosis by staining for tunnel. The endothelial cells also are pro-inflammatory in these progeria vessels, and shown here, they're expressing E-selectin. They also express VCAM1, where the healthy vessels do not. So we've got a number of markers that demonstrate the pathology of the disease. So with that, we started to look at some of the treatments. I mentioned lonafarnib a little while ago, and in cultured cells, it improves nuclear morphology. So it does suggest that we are blocking the farnesylation. And in mice, there's extended lifespan, improved skeletal properties, and improved cardiovascular phenotype. Everolimus we used as the rapamycin analog, and it does increase progerin degradation by promoting autophagy. So we looked at these individually and in combination. And first, we looked at the vasoactivity, and we did find one thing that was interesting, that and we based our doses on some clinical data that was available for both drugs. We did find a dose of 2 micromolar lonafarnib and 1.1 micromolar of everolimus was toxic to our vessels. But if we cut everything in half, we were fine. And we could see that we could improve vasoactivity, vasoconstriction with the everolimus and the combination of both, but not so much with lonafarnib alone. And then in terms of vasodilation, we found lonafarnib seemed to be more effective than everolimus. If we looked at some of the markers for the disease in the vessel, when we treated with the combination of the 2 drugs, we could restore the smooth muscle actin and the calponin and the myosin heavy chain expression. So we restored the smooth muscle cells, and their staining is shown down here. So we looked at we also looked at some of the histology is shown here, and we could, with the either lonafarnib or the combination, reduce the alizarin red staining. We could increase the number of nuclei with either lonafarnib alone or the combination, and the vessel thickness was reduced, another pathological feature of the disease, by these treatments. So we looked at a number of responses, and both in cells and in our engineered blood vessel. And what we could see is that in the cell culture experiments, we didn't see any real synergistic effect when we added both drugs. But however, in our engineered blood vessel system, we found that when both drug combinations were used together, we got a pretty significant decline in calcification and increased smooth muscle protein expression. So it does lead some hope for this combination therapy as being beneficial, at least to the vascular system. There's an ongoing clinical trial, and we're very curious to see what those results are. In addition, it does emphasize the need to look at the cells in combination in their in situ environment, because we didn't see any real benefit of the combination therapy in the individual cells. So I'll switch gears a bit and talk a little bit about the adenine base editing project, which we're doing in collaboration with David Lewis Group and Can Cal at the University of Maryland. This works by reversing the mutation by using an adenine base editor that replaces the the A here with a C, and that ends up ultimately correcting the mutation. David Liu's group published a paper in Nature a couple of years ago, which showed a significant improvement in using the adenine base editor. And the animals actually had a normal lifespan. All the functional measurements looked very good. And they found that in the aorta, there was about 23% editing. So that prompted us to ask some questions using our system, whether we could replicate these results using human cells, and what level of base editing was needed to correct the mutation. And those were the two goals. So what we did in these experiments was we made our blood vessels first with edited cells, and then constructed the vessels. So we didn't replicate the in vivo experiment. That's something that will come up next. We could show that when we edited the iPS-derived cells, we could restore the wild type allele, as shown here, with very little bystander editing and very little insertion and deletion. So it's quite impressive. There is another adenine that's about 6 nucleotides downstream from the mutated adenine, and there's very little correction of that. So then we looked first at the vasoactivity over a 3-week period, and what we could see that if we made our vessels with the adenine base edited progeria cells, we could improve both vasoconstriction and vasodilation, and that was maintained over 3 weeks, which is very promising. We also showed that we restored some of the key functions as shown here for the von Willebrand factor expression, as well as the smooth muscle cells. We saw an increase in the number of nuclei, and then just showing here myosin heavy chain synthesis. And we also reduced the deposition of the extracellular matrix as shown here. Basically restoring the key functions of the vessels. So knowing that the edited vessels behaved as well as the healthy vessels, we then did what we call a mixing experiment, where we mixed various combinations of edited and unedited cells to try to identify what combination of editing in these human cells would restore function. We first looked at 3 weeks after fabricating these vessels. And what we've shown here on the right, first is that we didn't see much change in vasoconstriction, but we did see improvements in vasodilation at the 50% and 75% editing combinations. We looked at the smooth muscle cell density and got similar results. We got improvements at 75% and 50% edited cells, but not the 25% edited cells. We then looked at over time whether if we added these edited cells, would they outcompete the progeria cells that were present, since the progeria cells would be dying off and not replicating. We perhaps thought that the healthy cells that were edited would increase in number. We saw some effect of that at the 50% editing here over a 5-week period, but not so much at the higher percent editing, and a little bit of an effect at 5 weeks with the 25%. On the right, what we plotted was the difference in the allele from the initial mixture. So we're looking for the wild type allele. And what we saw was that after 3 weeks, we definitely had an effect at the 50% editing that persisted for 5 weeks. And at 5 weeks, there's some evidence that the 25% editing was actually leading to improved function. So suggesting that in vivo experiments, we might be able to get improved vascular function with less than 100% editing. So what I hope I've been able to convey to you is that we could replicate key disease functions of progeria, such as smooth muscle loss, excess extracellular matrix deposition, increased thickness, and calcification. We did identify synergistic effects not identified with 2D cell culture, and that this adenine base editing is very promising, and it alleviates endothelial dysfunction and disease pathology. And it looks like at least 50% of the cells in short-term experiments do improve function. And there's some evidence that these edited cells are proliferating in the vessel after several weeks that could improve the efficiency of editing. So I'd like to thank all of our collaborators, both at Duke, the students who worked on this, David Liu and his group at Harvard and CanCal, as well as the funding and the Proteria Research Foundation. Thank you.
Thanks so much, Professor Trusky. Great talk. Can I let Carlos introduce the last speaker of the session?
And next we have Dr. Daniel Paul, who is just a few hours north of us at the New York Stem Cell Foundation. The title of his talk is Leveraging Automation and AI to Streamline and Scale iPSC Disease Modeling. Take it away.
Absolutely. Thank you very much. You can see my slides okay? Great, perfect. Okay, yeah, thank you very much for the invitation to speak today. We are very, very happy to be here. So, yeah, I'm from the New York Stem Cell Foundation. I'm Daniel, as was said. And for those of you that don't know us, we are a non-profit research institute here in Midtown Manhattan, been around for 17, 18 years. And our mission, founding mission, was to accelerate cures for the major diseases of our time through stem cell research. But in doing this, we've also embarked on a lot of rare disease work in this process as well. So as a nonprofit, we do have our research institute, but we do a few other things as well. So for example, we fund postdoctoral fellows and early career investigators, and we have a great community that spans neuroscientists to stem cell biologists. We do a lot of outreach, and we have our own conference coming up in a month or so's time. So if you're interested, I'll have this flyer again at the end. And then, like I said, we have our own research institute as well. And so here we have disease-focused research teams. We have a big platform I'll talk to you about today, but we really harness stem cell biology to look at disease modeling. I'm talking to you today, but I will point out that the work is part of product of an incredible team of people. This is just a few of us. We're about 120 people in total, but the faces you see represent a whole diverse, different set of backgrounds technically, and I'll come on to why that's the case in a second, but I wanted to make that very clear. So here at NICEF, we think that to get toward new treatments, we're going to need a lot of scientific convergence. And so that's going to be using things like automation and robotic infrastructure to be able to do things with the kinds of reliability and scale that we've already been touching on in some of the discussions already today. And so this has been a big focus for us for a while. But ultimately, it also needs things that can then take advantage of that as well. So things like high throughput assays that allow you to get readouts from having this kind of scale. But then ultimately as well, having the data science capabilities to be able to actually take apart that data and get to results. So as I mentioned at the start of this, we have a lot of robotics here. And so for the past 10 years or so, we've been building what we call the NICEF Global Stem Cell Array. It's a little bit of a mouthful, but we think of this as a place where science meets scale. I'll come on to what scale means for us in a second. We've touched on that word a couple of times today, but we think of it in a particular way here. And so the array as a platform is made up of really three key areas. One is hardware engineering. So we have those robotics. We have teams of programmers that program those robots. We do a lot of fabrication here as well, and obviously programming and process development. We have a fairly large and growing computer science team that spans everything from pure software development, building out limb systems and web applications, to machine learning applications using data scientists and data engineers. And then obviously biology, can't forget the biology. And so we have biologists spanning specific disease areas or scientific backgrounds, some genome editing and screening. And when you bring all those three together, that is what makes up what we think of as the array. So I'll start with an image I thought we might have seen sooner today, but just for anyone not familiar, this is the process of making a stem cell. You take a biopsy or maybe a blood sample, and it looks pretty simple. You grow out those cells, you infect them with something, maybe it's RNA, maybe it's a virus, to express some key genes to turn them back into stem cells. You isolate those stem cells, and then you can kind of grow them forever at some level. And on paper, it looks like a pretty simple process. And truth be told, it's not necessarily the most difficult. It's definitely technically challenging. But if you break it down into lots of steps, it's actually an enormous number of steps. And so the whole point of building out the automation about 10 years ago when we started this was to automate just this set. We said if we wanted to study human disease, we would need a lot of different samples to be able to do this. And one way you could do that is just to throw lots of hands at the problem and have lots of people reprogramming by hand, or you could try to bring a level of different level of scale and standardization using automation. But cell culture is innately complicated. And there's a lot of things as biologists that when we're doing our research that we just take for granted. So when we're in the hoods, we can make snap decisions on things like cell seeding numbers or the centrifuge speed we're going to use, whether we want to use a particular media without giving it really a second thought to some extent, whether we have to pre-coat our plates with things like Matrigel or other coating mechanisms. How many plates we want to make. We can just change our mind on a day-to-day basis, but doing that on a robot becomes a whole other task, because maybe some of this has to be fairly regimented, or you have to build in the capability with quite advanced programming to allow all of these things to be customized in nice graphical interfaces for the scientists within the lab. And that's basically what we've put together here over this past 10 years or so. So every single step of cellular biology that we do by our hands, we can basically do on these robots. Thawing, passaging, transfecting. It has images so that it can image overnight. You name it, freezing, thawing, yeah, all the things that we take as simple things to do by hand, we've been able to put onto these platforms here. And for us, it allows us to then operate at what we think of as a level of scale. And so scale for some people, especially in like a therapy point of view, might be billions of cells of any one given cell line. And that's great. For a therapy, you absolutely need that. But if you're studying a disease, you don't always necessarily want lots of samples from the same person. You want maybe a small amount from many, many different individuals. So you could start looking at the impact of genetics. We just heard about the great example in or we've heard a few different examples in different diseases where the genotype has such a big role in how a therapy might or might not work. Being able to culture lots of samples in parallel becomes really important. And so that's really become the focus of what we do here. So a typical day for us might look something like this. This was from a couple of weeks ago in August, but it's a pretty standard day. We had about 10,000 total wells of cells growing. It came from about 250 unique individuals. It represents about 1,700 different cell lines. And then every single well of every single plate that was growing at that point in time was imaged overnight. And so, you know, just with bright field imaging alone, that's like 12,000 different data points. And so all of a sudden, this is a giant level of scale to deal with. That's pretty tough to deal with. But we've spent a lot of time trying to figure this out. And so it's left us in this place of being able to now have quite large sample sets where we can take skin or blood samples, use our robots to make iPS lines, and then turn them into disease-relevant cell types. But as simple as I make it sound, there are still many challenges in using iPSC-based models. And so I touched on a couple of these through the duration of the talk, but I think the first one is one that starts right at the top, and that is simply access to relevant material. This is the starting point. If you can't get material that you need to be able to study a given disease, nothing else matters downstream of this. And so one of the things we've tried to do here is put together an open biorepository. So here we have clinical research teams where we can collect samples, we can collect medical information, tie that to the cell lines that we have, run QC on all the cell lines that we make, and put that into an inventory that's available to researchers around the world. And so the biobank sits at a point where we have about 300 different conditions represented, covers major ethnicity groups, and we're spending a lot of time right now trying to really expand that aspect of things. And we have some patients linked to longitudinal data. But like I said, ultimately it allows us to go out and really recruit in very interesting ways. And so if you're interested, that's the web page, nicef.org/repository. And what it's allowed us to do is a few different things. I've said we've got our own collections here with our clinical research team. So we focused on genetic and ethnic diversity and put a fair amount of effort into rare diseases as well, such as Charcot-Marie-Tooth and a disease I'll come back to later, infantile neuroaxonal dystrophy, or INAD. But we've also worked with other institutions and foundations that have been collecting samples and have now been able to come to us to say, actually, let's now turn these into iPSCs so that the community as a whole can begin to take advantage of it. And so we've done this in a big way with the Simons Foundation and Nancy Laurie Marks Foundation. This is just one example of others. And another one with the National Eye Institute, where we collected blood from around the country, brought it all here, and have been converting those to iPSCs. And so even from the very beginning, we were working with Bill Gull and the Undiagnosed Disease Program to have lines right at the beginning from that program put through the robotics as well. And we're doing similar things with CARD at the moment as well. So ultimately, though, this is where we want to get to, being able to do population scale analysis effectively, whether it's for a rare disease or a complex disease. A rare disease is not just one disease, even if it's sort of diagnosed as one. We just, like I said, heard about all the genetic variation that can exist even in one diagnosis. And so being able to capture that genetic diversity and then look at the effects of drugs or just the disease itself is really what we want to be able to do. And so one approach we've taken to do this is starting with, without even looking at iPS cells, just touch on this at the beginning, but is using techniques such as cell painting. And so this is a technique where you can just stain many different organelles within cells, take lots and lots of images, and then use things like deep learning. There are some other tools like Cell Profiler and other tools, but use things like deep learning to see if you can detect signatures that allow you to tell cells apart based on their health status and things like that. And then ultimately, could you apply a drug to reverse a phenotype? So this is what cells might look like when they're cell painted. Here we have skin cells. There's many different people represented in this well. I struggle to tell the difference. You can see different shaped cells, but there's nothing obvious that stands out. And so what we asked is, if you were to take a fairly large cohort, in this case, this is for Parkinson's disease. We went into our repository, we pulled out just under 100 samples, half of them healthy, half of them with Parkinson's diagnosis. And we ran the cell painting assay. I'm not going to go into the technical details, but we use machine learning to ultimately end up at a classifier. If I show it an image that looks like this, could it make a prediction accurately as to whether those cells came from a healthy person or someone with Parkinson's disease? And so sure enough, to skip over a whole heap of different work, you can do this fairly accurately. So even for sporadic PD, using these deep embeddings, we can get to a fairly strong ROC-AUC, so sort of measure of accuracy to some extent. And when you look at specific mutations, you can even make stronger predictions just by looking at these images, just skin cells, not even into the iPS cells at this point in time. And so we've also taken this application to rare disease. I mentioned I would come back to INAD, infantile neuroaxonal dystrophy. And so it's a pretty rare disease, one of the ultra rare conditions, and has symptoms that are sort of akin to childhood onset Parkinson's-like symptoms. Typically you see symptoms appearing between 6 months and 2 years of age, and very sadly these children don't survive into their teenage life. And so one example of where NICEF has played a role in trying to support these sort of rare diseases is to make that material available. I touched on that right at the beginning. How do you get access to this material? So we teamed up with the iNAD Foundation, a rare disease foundation for this disease, purely at the beginning to make a cohort that would be available to the research community. And so now it stands that there's 5 different families in here with trios, with iPSCs, fibroblasts, blood samples, all available to the community. And there's some some CRISPR corrections in here as well. But having this repository allowed us to then take these samples and ask questions of them as well. So we wanted to apply that same approach I just showed you in Parkinson's to this disease. And so went into our repository. Another major problem still for the rare disease field, so many rare diseases affect children, is that just having access to healthy childhood samples is actually very challenging. So we scoured repositories looking for healthy material for these age-matched controls, which machine learning is particularly sensitive to. And it is just very, very hard to find large cohorts, understandably so. You're not just going to biopsy children for no reason. But we've put them into these same kind of machine learning pipelines. We've scaled the assay, as we were sort of just hearing about as well, down into 3D-4. And we've got a model that works just fantastically well for this disease. And so to put it into sort of a visual picture, we really see these two clusters begin to appear. We can really separate the disease samples from the healthy samples. And so the question is, can I now make these disease samples move into this sort of cluster by applying drugs to make them look healthy again? And so we've been screening the FDA approved library. We're quite excited by what we're finding, but the analysis is ongoing, and hopefully we can share more of that in the near future. So that's one approach, using sort of a platform and building upon the platform that we already had here to do this kind of work. But what about iPSC models and getting into 3D? That's sort of what some of what we're here to talk about today. I'll just point out, we spent a lot of our time working in 2D though. This has been sort of the bread and butter of what we've done. So 2D flat, growing on traditional plastic, as many of us have done for a long time. And all the steps you see here, like I've said before, are automated. So from thawing on this side to passaging to saying I want to seed at very specific cell numbers inside these plates, all done on the robot, transfecting with different reagents, all automated. Really, users have to take things to and from a freezer to some extent, and help with some other bits on the robots, but basically no need to touch the cells. And again, all driven by software that we've developed in-house. And we really can sometimes spin up experiments quite quickly to go from like a hypothesis just to test something out and end up with a result toward the end. And so it allows you to build these nice plates of things like neurons. We are big fans of these transcription factor-driven differentiations, where you can overexpress just a gene or two and end up with a population of cells that you like. So in this case, neurons here. And we've used that to study human diseases such as Alzheimer's, PTSD. We've got a pretty remarkable signature here, and a lot of work in MS as well, led by Valentina Fossati here at NICEF, to look at how we can model that disease. But obviously, like I said, we want to get toward 3D, and so a big chunk of focus for us has become this sort of approach to scaling and standardizing that movement from 2D to 3D. And one of the areas we've been doing this, we've done it in a few different areas, but one has been in beta cell differentiations, and this has been led by Filippo Cipriani here at NICEF, working with Francis Collins' group at the NIH. And they've been really looking to scale up automated beta cell differentiations, which uses a combination of 2D and 3D in its differentiation. It's quite a long 30-day differentiation where you mature the cells toward these beta cells. And then here they're using rare diseases as well, so MODY, monogenic onset diabetes in this example, but going from things like having 12-well plates of cells, being able to normalize them and stamp them out into 3D 4-well plates, and forming organoids in these 3D 4 U-bottom shaped cells. I'll show you what that looks like in a second. So a lot of work has been happening behind the scenes here for this kind of project. And even further getting to the point where you could put in this particular example here is just 84, but obviously you can fill this plate up. So you could have whole plates of organoids from many individuals, 84 in this case. You get a few replicates per plate, and you see these organoids just grow up over time, and we obviously make multiple copies of these plates. The robots are very clever these days. You can do lots of randomized quadrant swapping and all sorts of things to really get away from having cells on the edge on this example. They could then be somewhere else on the plate as well. We've done this in neural organoids as well. This example's 29 different people. And again, in this case, giving more replicates and no edge effect in this plate. So again, lots of different things that we can do. One thing that we can't do, there are sort of solutions to it, and we're quite excited to see where some of the 3D printed work we saw earlier with the motorized spinners, how we can begin to automate that in these sorts of high throughput setups as well, because these aren't agitated, these are just static cultures, and we do recognize that having that agitation may be very key for 3D cells. And so how you do that at scale with many plates though is a different kettle of fish. There are shaking incubators and things to explore, but certainly some technical challenges to overcome in that realm. And then ultimately, this problem of age, we heard about it earlier, we don't get oligodendrocytes in organoids all the time unless you particularly drive them to it. Valentina spent a good amount of time here with Paul Tezar at Case Western to try and improve this, but to get to oligodendrocytes that are myelinating neurons, we're having to wait over 200 days. And that's just an awfully long time. And getting that many cells from that many different patients to grow for that long is just technically challenging. So the question becomes, how can we get to a point to speed it up So whether it's 2D, whether it's 2.5D, maybe using thicker matrices to store things in, or whether it's using 3D, I think everything still has its challenges, whether it's from just growing it in the first place to having assays that can then read out on the other end, especially if you're trying to operate at this scale to capture the complexity of human genetics. So here's one example that we're trying to deal with at the moment, where you look at typical differentiation protocols. To make a neuron might be 21 days or so. To make an astrocyte, you're looking at maybe 60 to 90 days. And oligodendrocytes, you're typically looking at 80 to 100. This is just in 2D. And so these very long protocols, which here are neurons, but it's applicable really to many different cell types. They're very long, they're very labor intensive. They often don't cryopreserve well. You can't always stop in the middle of these things, and they're very heterogeneous, where you only get maybe small fractions of the actual cells that you want at the end of these protocols. And also on top of that, for us in particular, they're not particularly automation friendly always. Getting these synced up so that you could have cold cultures. One of the things we can do on the robots is like combine things and pool things together. But timing here becomes an issue in trying to do things like that. And obviously then yield as well becomes an issue when you're trying to think about ultimately working toward a drug screen. So how do you get these things to sync up? One approach is to try and switch to the more and more to these trans differentiations. So I mentioned NGN2 earlier for making neurons. We're seeing similar things begin to appear that work reasonably well for other cell types like astrocytes and oligodendrocytes. And here you can begin to standardize things. You can standardize now the timing and the media. You can get very good yields and purities out of these. And some of the work being led by programs like the INDI program, the NIH are really helping with some of this, and ultimately get to this point of making it scalable so that you could get to things where you really can be doing myelination in a dish, whether it's in, again, 2D, 2.5D, 3D, whatever you want to, whatever your outcome might be. Along the way, we built a lot of different tools, so we've open sourced different aspects of what we've done. If this is relevant to anyone, feel free to take a look. You've got a tool that allows you to really look at images, lots of images, to work out if you've got monoclonal cell lines, important in the editing space. We deal with a lot of imaging, as I've sort of mentioned, and so knowing if things are in focus, out of focus, have imaging artifacts, we've built basically operate in near real time, a couple of seconds, to give you a feedback on whether an image is in focus or out of focus using machine learning pipelines. And then tools to, again, really quickly look at these high throughput morphology-based screens, whether we can pull out the specific features that are driving the effects that we're seeing. I probably haven't gone into enough detail for this one, but we just put that live on bioRxiv, so feel free to take a look. Everything that I've talked about today, though, has only been possible because of this lady here, Susan, starting the foundation when she did, because for all these diseases, there's always a personal connection back to them, and for her, it was type 1 diabetes. And at a time 20-ish years ago, when doing stem cell research was just nigh on impossible and there was no government funding, she really has started something that has helped the community as a whole grow, and so a true testament to everything she did. That's everything I've got for today. Some of the people I spoke about, some of the work I showed today is from partnerships with these folks. I mentioned we have the conference coming up, so feel free to take a look at that if you're interested. And there's some links on the side if you want to get in touch. Thank you.
All right, thank you. Thank you, Dr. Paul. Very nice talk.
All right, thanks all the speakers, terrific talk. Please join us for a excellent talk. We're gonna start the panel discussion with all the speakers this session. We have about half an hour left. Please, if you have any questions, if you're an attendee and you have any questions, please drop them in the question and an answer box, and then we'll try to address them as we go along in the discussion. But to get things going, I think there were a few questions during Professor Ming's talk, and it had to do with the usual question with organoids in terms of complexity, physiological complexity, the type of cells that you can get, and how do you go about assessing? I think the question for you, Professor Ming, is how you go about assessing how much complexity, physiological complexity, do you need in your models to represent diseases, and how do you in your group, in your research, go about addressing it?
So in our model, we try our best to recapitulate the maximum complexity that you can get. Obviously, if we direct the faith to the neuroectoderm, what the cells you can get is from that lineage. So other cell types, such as vasculature cells, microglia cells, they are from different lineage, and therefore they are lacking in our model systems. And there, of course, there are ways to mitigate these limitations. For example, you can add vasculature cells, you can add microglia cells, or other cell types into the brain organoids. However, there's also there's the limitation. We actually tried very beginning to add vasculature cells, but we quickly realized that you need a heart. Otherwise, there's no diffusion. That's why we went about this slicing kind of brute force method to bypass the diffusion limit. But I hopefully with the development of additional technologies, microfluidics, and other engineering approaches, we can improve and allow for further maturation as well as increased complexity in our organoid models.
If I may add, do you systematically go for each disease that you're studying? Do you systematically assess like, okay, what physiological complexity do I need to see a disease phenotype for this particular disease?
That's an interesting question, right? I think the basic is still that you generate organoids that can recapitulate maximum complexity in your model system, and then apply your model system to study disease. Ultimately, the disease happening not within a single cell types, but in potentially affecting multiple cells. So the more cell types you can have representing in the organoids, of course, the better you can recapitulate the disease potentially. That being said, there are also things you can already do, even with the organoids that's not completely recapitulate the brain, right? You can still study cell-cell interactions, environmental impacts, and things like that.
Thank you. Carlos?
Yeah, so the next question is for Professor Bickman. In your opinion, which approach do you think we should be taking when to account for heterogeneity, particularly for a specific disease? For example, for cystic fibrosis, there's about 2,000 mutations in the transmembrane receptor. And what do you think we should be thinking about when we're developing these 3D models and trying to incorporate that heterogeneity?
Yeah, it's a good one. I think at least in my lab we focus on airway models and then on intestinal models. I think it's not per se also 3D you need to have. I think what's really important is to have your systems represent your readouts also being physiologically driven. So we try to stick away from reporter systems introduced into the system. So we try to stick close to biology. And then I think CF is pretty there's a lot of knowledge base already on cystic fibrosis. So we know the majority of disease, and yeah, for me, I think if you do these clinical translations that we do towards an individual, I think we like to keep it as as complex as needed, but as simple as possible, more or less to say. So we rather go for simplicity if we can, just because of standardization issues. And we see one of the big limitations for us, at least if we try to clinically translate and to have individual outcomes predicted, that the ground truth of data is your clinical data set. And getting accurate clinical data, which really is informative for an individual, is also something which is really difficult. Sometimes if you get clinical data from trials, you only have short-term data points, which are not per se predictive for long-term disease progression or clinical response. So it's also the getting, I'd like to keep it as simple as you can. Obviously, this works for CF to some extent because it's epithelial driven. It's also then mostly in your system in epithelial organoids. But I think one of the big challenges for us is to also really get accurate readings from the clinical perspective, actually. So making sure that your clinical data, we have, if you have long-term clinical data, individual, then there's real good accuracy. But from short-term clinical trials, it's very difficult to be precise for an individual. And then you still need to bin, you do a precision approach, actually. You just, yeah. I don't know whether I answered the question, but I think.
Yeah. So I'm hearing mainly sort of not taking the models in isolation, but also incorporating the clinical data to get more out of their experiments.
Yeah, I think if it's all about predictability and trying to recapitulate in vivo biology, it's very nice if you can just directly compare to the individual data. Actually, if it's available, I think it's for CF. It's a progressive disease, and people are coming very frequently to the hospital, so there's a lot of clinical data built up. This could be very different for other types of diseases.
Very nice, thank you.
And please, if anybody has any additional comments to Carlos' questions, please feel free to, the speaker, feel free to also chime in. My next question is for Professor Trusky, and some of the question that has come up in our side is the use of patient-derived cells versus CRISPR-engineered cells. And people advise, well, if you use a CRISPR-engineered cell, it's technically easy to develop these models. So can you give us a sense in the work that you've done, your sort of thinking process, patient-derived cells versus CRISPR-edited cells, where you had to do that at some point, or by working with the foundation, you had access to patient cells and you decided to work that way?
Right. It's going to depend on the disease. With progeria, some of the mouse models have, well, they haven't necessarily used CRISPR, they've used other methods, and they have created mutant cells that will express progerin. With progeria, because it's a random mutation, you're generally affecting only 1/2 of the gene. So you're going to get a mixture of healthy and progerin expressing cells. So if you use CRISPR, at least now, I'm not an expert, but at the moment, without some targeted approach, you may just have a 100% mutation throughout, which could affect the phenotype a little bit. So you have to be careful about that. But there certainly are advantages because the patient population for progeria is very limited. And there are non-standard mutations. So the PRF currently has a small number of those with the classical mutation. So it certainly would increase your number of samples that you could work with, and you could also look at the effect of the background on your signal. So there certainly are pluses.
And one question in terms of follow-up and engineer models like the one that you show very technically sort of complex in terms of the engineering that goes into it and the throughput, right? And it's been brought up several times, the issue of throughput, not only to test compounds, but to test cells, right? And different cells, whether cells from different patients. So what sort of, with your model, how many patients or cells from different patients have you been able to test, and what sort of technical reproducibility versus biological reproducibility or patient differences do you see in your system, or have you ever to test that in your system?
Right. So far, we've only looked at 2 donors and several clones of the donors. On the plus side, we've been able to, with those two donors, replicate the same types of results. So there was some consistency. But the throughput issue is, for the system that we have, is a challenge. I mean, there are a couple of options. There's work being done by Shrike Zhang to simplify the vascular system. You have the trade-off where you can keep the cell types, but you can put it in a microfluidic system. You may not be able to measure vasoactivity, but you could potentially measure other responses. That could increase your throughput. You know, so our system is not going to be early stage. It's really when you have a finite number of compounds. There's obviously training involved to learn how to fabricate the system properly and so forth. There is some scale up, but there really are a couple of trade-offs about how many you can run and what type of system you can use. And this goes back to, I think, the point that was raised before about the level of complexity. If you want to reproduce some of the, say, a structural feature, then you have the advantage of potentially going to microfluidics that can put things on a multi-well format. There are approaches that have been developed to eliminate flow from pumping and even have unidirectional flow, which for our system would be quite critical. So that can be done. So there are ways to increase the throughput to some extent, but it is a challenge with these systems to get really high throughput.
Thank you.
My next question is for Dr. Paul. Seeing that you have, all these platforms and automated capabilities, I imagine that you're having, many groups, particularly in like rare disease foundations knocking at your door. How is it that you prioritize, which projects and cells to onboard?
Yeah, good question. Very good question. I think it is, it's everything that everyone always balances with: resources at any given time versus like active work that is going on versus sometimes the impact it might have, the stage that someone is at. If someone's already collected material, it's obviously much easier for us to just kind of take that and run with it. If it's starting from scratch where no collections have started, it's just more work. We have a team here that helps figure out which projects to take on at any given time, and so we'll do our best to try and take them on. But a lot of things go into grants, and the world of grants is its own world. So a lot of things are at the whim of those sorts of things. But yeah, it's we would love to say yes to everything. There's obviously bandwidth constraints that come into some of this. So group decisions, lots of talking. That's classic ways that decisions get made, I guess. I think, can I just touch on one question that came in on the chat, because I think it's a really good one.
Absolutely.
Which is, David asked a question about whether there's been interest in assessing the performance of models in their ability to distinguish drugs that have worked and those that have failed, and use it as predictive values of the models. I think that's a really good question. And I think this is an interesting one from, I don't know what others on the panel have found, but where I think we're seeing it shift right now. I think it's taken a while to get there, but we're really seeing a shift right now in pharmaceutical companies really beginning to embrace these models as they've got maybe more complex, but more data has come along as well to show that the reproducibility is there, or whatever they're looking for at some different time. And so I think right now we're beginning to see that these are the kinds of the things that they're asking more and more. There have been interesting programs that have tried to do this already. Like there's one in the cardiac space, a group called HESI, H-E-S-I, where they've been trying to look at the effects of drugs on arrhythmias and things like that, using IPS models, and have had involvement from pharmaceutical companies from the beginning. but I think we're on the precipice of this really like starting to shift, and it's quite exciting that that could be the case, because if you can prove out that yes, you could have stopped something either going in the reject bin or getting to a trial and failing at that point when you could have predicted a failure much early, or even the one that I like to dream about is the one where you could actually have a drug and say, hey, a trial could be run more efficaciously if you only target a population of people that have this genetic characteristic. And like, these are the people that this drug's really going to work on. Don't just do this all in one, like try and target everybody. We know that the drug's only going to work on a subset. That will be the one where I think will generate a lot of interest. So if we can prove out those things in these retrospective studies, that's where we're going to see even more shift. But it's beginning, I would say, it's beginning to happen.
Great. I have a general question, and it's about assay readouts, right? So we heard from Professor Beckman about beautiful swollen balloons for cystic fibrosis, beautiful phenotype, versus using cell painting and let machine learning tell you what is it that you're measuring, right? And it just struck me that, and then Professor Tresky talked about like vessels. So how much is the phenotype sort of really driving how good these models are at predicting relevant responses, clinical responses, right? And in your decision, especially in 3D models where some of these high content imaging are pretty challenging, right, to do cell painting and so on. So it just struck to me that having a nice, clear disease phenotype like for cystic fibrosis seems to be the way to go. And then the question is like, when you develop these models, is that what you should aim at, right? And I'm open like to anybody that is willing to give an answer to this question.
I almost put that as like one of the challenges that's there for the like just generally at the moment, because assay readout is the hardest probably. Having something that's super relevant and that your cells are recapitulating the relevancy of a phenotype. Operating at scale, like, you know, there are things like the beta cell, for example, is a good one where you can measure a phenotypic readout with something like insulin secretion. So can you detect insulin being secreted if you challenge them in the dish? And like the way that we would most commonly do that is with an ELISA. But if you're running assays with small organoids and you've got lots of them, ELISAs are maybe not always sensitive enough in their current iteration. So we've been working with like companies to test out technologies that can do effectively ELISAs at much lower liquid level sensing. But it definitely governs, maybe to the point that Carlos asked earlier, what projects you can and can't take on. Because if you don't think you can get a phenotype out of it, it's going to be a challenge from the get-go. It's a big one.
Other speakers?
Yeah, maybe I can comment also. We were a bit lucky, obviously, yeah, with this swelling phenotype. It's such a, even for me as not being an image specialist, I did confocal as a PhD and postdoc, but even I could make this quantifiable. I can't do these complex things that people nowadays do.
Jeff, a quick question. Do you see the same phenotype with liver or other organ types?
I was going that. So that was also a bit of the luck actually. So I think organoid swelling, if it's forskolin, if you add forskolin to many organoids, they swell. Sometimes in different velocities and so on. But and most of that swelling, I think, in many systems is CFTR driven. But you also see, for instance, if you do nasal airway organoids, you see that there are also alternative ion channels which get activated. And one of the beauties of this intestinal model is that it's fully that the only ion channel there leading to, you know, this fluid secretion in CFTR. So this allows you to really pinpoint CFTR, and this is more complex with other types of organoid models, for instance, which can also be interesting. You can look at different types of ion channels, but yeah. But I think it's been. And we are still, I think, a part of the solution again is automation and scale up. So if you have variation, at least you can do replicates. We haven't shown you, but I think if we do a standard typing of patients, we always do forskolin in those ranges. You do different types of drug combinations, so you do replicates over time at different culture time points. So it's just if you think about complexity and standardization, if you can scale up and you can just generate more data, at least you can do dose response curves and all those types of things, which will just help you to build confidence and get better data.
I would like
Professor
to
Ming.
add a little bit more too. In the case of our modeling using the arcuate organoids, we also show that there's a direct leptin response deficits, which correlates quite well with the patient phenotypes.
So one question I have, Professor Ming, about like the neuronal organoids. Beautiful word, beautiful histology and imaging. So what assay would you use to measure activity or disease differences or disease phenotypes in these brain organoids? Have you been able to look at neuronal activity?
Yes, there are actually many different ways to look at the neuronal activity. For example, now multi-electrode array are being used quite frequently to assay the neural network activity. Another one that commonly used in the field is the using calcium imaging to reflect the synaptic activity as well. So these are the ones that can be used. Of course, there are many things not just directly related to the network activity, but also other developmental processes, for example, affecting the neural stem cell behavior, such as brain developmental disorders related to microcephaly, macrocephaly, and other lysencephaly, for example. So there are many different phenotypes, but I think, as being said earlier, the ground truth is to directly compare to patient phenotypes. And other is discovery based. Sometimes we discover a phenotype, although that hasn't been shown or it's difficult to show in patients. Then I think making conclusions should be more careful.
Thank you, Carlos.
Yeah, so this question is, I guess, for everyone also is, given that we have sort of this limitation on being able to mature the iPSC cells when we're differentiating them, what is your guys' thought about maybe possibly just concentrating on early onset diseases where we have a closer similarity to the iPSC differentiated cell types?
Yeah, so we've done some work with some rare disease foundations looking at sort of neurodevelopmental disorders where again you can sort of begin to look at this because maybe it is a problem in NPCs or and sort of it's an architecture point of view. I think we've seen nice things from even like Paula Lotta's group looking at even things like autism and being able to see issues that stem from almost development quite early on. So in 3D models there. So yes, I think it would be naive though to only limit ourselves to those sorts of diseases. I think that definitely you can definitely go use them as models. I just think that we've got to solve the complexity of aging samples in a dish because that underpins so much more of many of the diseases that we're battling with. But there's a lot of rare diseases that stem from neurodevelopmental, so we can certainly use those, and they give us insights ultimately into these more complex diseases. Yeah, it's a little bit of both, I think, ultimately.
There's a question in the chat that it was answered by Professor Trusky, but I think we'd like to hear what others think in terms of some of these models take really long time to mature, especially some of these organoid models. And the question when you're trying to do compound testing or pharmacological testing is when do you add the compound, right? So at what point in your differentiation process you add the compound, or it's like too early, too late? How long? So how do you perhaps Jeff, you can comment on your experience and also for those George added some comments, but what are your thoughts on that aspect? when to add the compounds. Jeff, in your
Yeah.
case, when do you add the compounds in the treatments?
Yeah, so we have I think the cells how we grow them, so we have weekly replicating cells, and we just have made a standardized scheme in which we add the compounds directly after passaging the cells, because then the structures are as simple as possible, which makes the imaging again more simple, because if you wait a bit longer, they start to bud and make complex structures. So it was very pragmatic driven actually. I think one of the difficulties is also the amount of compound. So you can it's very easy to overdose in vitro, and the PK is very difficult to understand the PK in vivo. So we've seen. I think we try to stick to serum concentration plus 10 times serum minus 10-fold serum concentration or so. We try to be within those types of limits. But yeah, again, that's all. It can be difficult. And length of incubation, it's all depending on the type of. If we have these protein restoring drugs, and the protein has a turnover time of a couple of I think it's produced in 2 hours, and then it has a turnover time of 24 hours or so. So you have a pretty fast readout. While we also do similar things with primary ciliary dyskinesia, cilia diseases, in which you have very complex structural protein complexes which take a long time to assemble, and if they have once they are assembled, they're very stable. Also, if they have a mutant protein in there, you need far longer incubation. So it really depends a bit, I think, on the biology. And in the essence, I think I always go back to if you want to understand, if you can have direct clinical data related to your observations, you can just calculate how much correlation there is, and then you can just calculate the amount of causal relation, but at least you have some idea about the predictability. It's then just a simple matter of just doing math.
Thank you, George. Can you comment on your answer as well? It looked like it's sort of testing and see what you get.
Yeah, well, some of our work has been empirical in terms of making sure that we can observe the phenotype, and then we would add the compounds and see the temporal response to see an effect. But I would, at least with respect to our disease, I would add one comment and then a general comment that with progeria, it's also a concern about when you actually can diagnose the disease in an individual. So perhaps in a well-developed country like the United States or in Europe, you may catch a child at a very young age and then try the therapy. But in other parts of the world, the child may not be 9 or 10, in which case the disease is far further along. So one might want to, at some point, you have to figure that into the disease. In other words, the symptoms may have started to create problems that may or may not be treated as easily. And that needs to be looked at. The other point with our systems, while it does take a long time, if you use the animal models as the standard of reference, we're actually doing things on a much shorter time scale to create the phenotype and then test it.
Thank you. Carlos, you have any other question? I have 2 minutes to go.
Yeah, so a lot of us are still, working on a particular disease, and we're doing a lot of the biology sort of testing and then running the experiment again. But can you guys comment on what you're doing in your own labs to incorporate AI or machine learning to sort of advance the discovery and targets to treat these diseases?
Sorry, Carlos, could you just repeat that? Sorry.
Can you guys comment on how you're using artificial intelligence or machine learning to better develop discoveries in every given disease?
Yeah. Yeah, obviously we're using it in a fairly big way. We use it in a whole heap of different use cases, from, like I said, measuring focus of quality control to actual readouts. And so something like cell painting with deep learning on top is a very unbiased assay. You're not really going in with a preconceived notion of what to measure. And so from that point of view, it becomes quite helpful to you, especially if you don't know what you're looking for. But at the same time, sometimes, it's nice to be able to know that you're looking for a particular protein and just have an antibody and, go measure that specific readout. It's great. It is very challenging though. Your sample sizes have to be big enough because it's just so prone to being fooled that you can trick yourself into thinking you have something when you might not. And so things like sample sizes matter, metadata matters. We touched on that earlier, but like access to whether it's clinical data, whether it's just metadata, age collection, sex, ethnicity, all those little things can start to matter. The more you can give these models, the better they can be. There was a question that came up as well about like combining data sets. This is something we're very much involved in at the moment, looking at how you combine sequencing data sets with imaging data sets, and then genomics on top of it to build these like large multimodal models. I think they're going to help us unlock quite a lot of things as well. But they're great, they're powerful, but they can easily lead you astray. And so I think it's just they need the right expertise. Unfortunately, I have people here that do that for me, and I leave it to them to tell me the ultimate answers.
Thank you. We are past 1 minute, but I have a question for Jeff in terms of building these clinical networks in Europe, and how did you go about it? I mean, we talk a lot about the need for clinical benchmarking and clinical samples. How in your experience, how did you build this network to be able to get samples so many countries in Europe?
Yeah, I think we were lucky again. CF is a pretty a prevalent rare disease, so these networks have already been established. I think in the US there's a large clinical network. In Europe it's there. I think the challenge for us, if you work in academics, get your legal department to sign documents with 17 countries and 40 institutions. That took a year or so. But in principle, I think that the networks were there. And the excitement is also there. People see the need for people with rare variants, and people want to help to bring solutions to people. So I think there was a really good, strong vibe to move forward. And the big challenge was, at least in our, just the legal paperwork to get it organized. Because in essence, the majority of network was already established.
Thank you. All right, Carlos.
All right. I just want to thank all the panelists and everyone for attending the first session on building experimental iPSC models. We've heard some really exciting work from all of our panelists, and I think all of their work has actually opened the doors for advances in translational science, specifically in rare diseases. And we're excited and looking forward to working and continuing this work in the science, particularly in the space of rare disease. And again, thank everyone for attending. And I think we're going to take a 30-minute break. Session 2 will begin at 12:30.
Thank you very much.
Okay, bye-bye.
Hello, everyone. Welcome back for this afternoon session of the i3D-Rare workshop. So we've heard a lot of exciting examples this morning about leveraging AI for translational research efforts. And here we're going to dig in a little bit more in this session on how to build up the data resources from iPSC-derived 3D cellular models. So similar to the first session this morning, we have 2 hours and 4 speakers. So each speaker is going to be constrained to their 20 minutes. There will be no questions in between speakers. However, participants are encouraged to ask questions within the Q&A. And we will address some of those questions throughout the moderation. So without further ado, and I do want to mention as well that we have the full bios for the speakers online, and so here we'll just be doing very brief introductions so that we can get rolling. So without further ado, I'd like to introduce Professor Matthias Kretzler from the University of Michigan, and he'll talk to us about what drug for what rare diseases, with an example on kidney organoids driving targeted therapies to patients with rare renal diseases.
Hey, thanks so much for the introduction, and thanks for such an exciting morning. Really, it was incredibly powerful to see how quickly our field is advancing. So what I would like to do in the next 20 minutes to give you a case study for an overview how multi-state data integration can help us to identify targeted therapies via integration of kidney organoid signatures with tissue-level based information from patients with their kidney diseases, and then share with you a data mining ecosystem we have developed for the kidney community at large going forward. One of the key advantages we have in studying kidney diseases is that, similar to oncology, but rarely for non-neoplastic diseases, kidney disease, we do take kidney biopsies percutaneously on awake patients from the kidney for diagnostic ascertainment of the underlying histological patterns. And this allows us not only to capture the structural lesions with digital pathology for AI-driven pattern recognition and disease classification using conventional histology, but it obviously gives us also the opportunity to use all the molecular profiling technology rapidly emerging as we speak to define the underlying molecular state of the various cells of the nephron segments under attack in our diseases. In parallel, we have, as with all rare diseases, blood samples as a matrix for biomarker development. In kidney disease, we have the added advantage that we have the urine, the liquid biopsy, as the kidney is shedding proteins, metabolites, and even cells into the urines who are available for molecular profiling. And then obviously, pertinent to our talks today is that we can generate from the same individual where we have deep tissue level insight into the rare disease processes affecting their kidneys. We can generate kidney organoids ex vivo in a patient-specific framework to see which of these associations we see out of the observational cohort studies can actually be interrogated for mechanistic impacts using the organoid platform. Essential for all of the efforts here with our capture of the molecular, the structural, and the clinical framework of our patients is a framework to bring the data together to define cross-cutting disease mechanisms so that we can use that initially in the first couple of funding cycles to define on a population level what are associated good and bad outcomes, and for those with poor outcomes, what are tissue level underlying molecular profiles. And what I will be reporting out as a use case here over the next few minutes is that we now bring that forward to a patient level assessment of the disease activities. We have done that in a rare disease clinical research network funded by NCATS and i3D-Rare and the patient interest group who initiated these efforts, Nephew Kidney International. The underlying concept here is obviously well aligned with what we heard this morning, that we consider pathobiology of our rare glomerular diseases to be driven by gene-environment interactions. And then we see in our patients, even with the same genetic background, quite different disease trajectories, which are consequence of different disease drivers being activated, leading to chronic disease progression and manifestations of the disease burden in our patients. Our conventional diagnosis, usually in our case mainly histological pattern recognition of distinct structural damage features, are capturing a continuum of these gene-environment interactions and are actually even overlapping with underlying genetic and mechanistic causes. So the charge for our efforts is to identify in which of the pathway progression frameworks we can identify targetable intervention points, with the hope, as we were discussing this morning, that an intervention point might not only intervene at one specific genetic risk variant, but might capture actually progression pathways shared between many of our patients developing symptoms and signs of glomerular disease. Neptune has established a rich resource to do that from all angles with deep clinical data, genetic, molecular, and morphological phenotyping, biorepository, historical structural analysis, and also a diverse ancillary study program with many partners in the academic and private domain bring our community together to identify these cost-cutting disease points for interventions. And obviously pertinent for the discussion today, we have a rich iPSC cell line repository linked from patients where we have orthogonal data sets available as well. Over the last several years, we have taken those observations now forward to the concept which is a real challenge in our rare disease. As you might have followed, we have a heterogeneous disease, pathobiology, patients showing the same phenotype but have very different underlying molecular mechanisms present, which resulted in the fact that our trials are recruiting quite heterogeneous patient populations. And therefore, often fail despite the fact that the individual action or targeted pathway is indeed effectively reversed in a subsegment of the patients treated, but the overall signal is diluted out so that our studies have to stop due to futility because we don't see a population effect. With the Neptune match concepts, we now use a knowledge framework I have shown to you to identify the underlying disease pathways active in a given individual at a given time, communicate that after a molecular nephrology board review back to our study participants, who then select the clinical trial to engage with in the hope that they have a higher chance to see compound efficacy, and the trial needs less patients on the rare community to be studied. To do this forward, we follow a strategy which combines literature and defined molecular pathways, which experimentally define treatment responses or treatment responses already observed in early phase clinical trials. And the main research engine for the treatment response definitions are indeed the kidney organoid systems we have been discussing this morning. As we define then at tissue level pathway activity measures, we switch it over to the non-invasive biomarker surrogates, which we can also define to some extent in the organoid platform in our retrospective patient knowledge network to then predict where patients might go forward into the clinical trial. Let me give you an example. It's a study which we published a couple of months ago, where we took our gene expression signatures from patients with one rare disease type, FSGS, minimal change disease. We obtained out of the gene expression profiles comprehensive signatures, classified those into distinct patient groups using consensus clustering strategy, and replicated these findings in our European and North Sub-Saharan African sister cohorts for robustness as is essential for these machine learning-based approaches. We could show with this approach now that our mechanistically tissue-level defined clusters are distinctively different from the descriptive syndromic disease definition used to date. We could also show that our signatures are robust across the 3 continents captured with the same protocol. We then looked what are the molecular groups associated with outcome, and you can see with loss of kidney function that's cluster 3, so that the curve defined here is a group which carries a majority of the disease. The beauty with these molecular signatures, we now can ask what sets these group of patients with a unique signature in the transcript space apart from those who are doing well. And using various network level analysis, it's quite obvious that patients show an inflammatory phenotype driven by TNF. And that was old news for us. We already had attempted a trial using adalimumab to treat FSGS patients in the FRONT2 study in our research network, and the trial failed for futility because only a subgroup of patients did respond to our intervention. So the question was, can we then use our molecular insight to identify patients with TNF activation, and using a TNF activation score, developed by Sean Eddy and John Hartman and described in detail in the manuscript. We developed for each patient a downstream transcriptional fingerprint where gene expression changes are reflective of the pathway activity. And in these waterfall plots, you can see that we were able to segregate patients effectively across the 3 continents. Using the single cell data sets available from our patients, we also confirmed that 2 of the downstream readouts of TNF activity, CCL2 and TIM1, for example, were indeed activated not only in the immune cells, but also in intrinsic renal cells, arguing that we had a whole tissue response present. But the challenge as of now is that the observations shown to you are exactly just that. They are associations. So we are in need of a mechanistic platform to actually test for direct causal impact of the pathway on our activation signatures. And here we are fortunate together with Jonathan Himmelfarb and Bino Friedman, Jennifer Harder in our team, who is leading our organoid core for the Rare Disease Network, has developed a robust kidney on a chip platform, which now is utilized in the CTOC framework for ex vivo assessment of compound activities. We have carefully benchmarked our organoids, as we have discussed this morning at several occasions as well already, against single-cell data sets we have from developing and mature human kidneys, and can show that the developmental trajectories of the organoids replicate to a significant but not complete extent those we see during human renal development and human kidney disease activation. We also extended these studies beyond the transcriptome to support the human secretome, so that we have these data sets readily available to build consensus signatures to identify for specific pathways which replicate the in vivo finding, which of these biofluid compounds might be useful starting points for diagnostic target development. And in this setting, for example, here with TNF, we could show in the single cell data that TNF indeed is activating CCL2 in all 3 tissue compartments present in the organoids in a dose and time-dependent manner. And interestingly, the transcriptional changes, which we have modeled in the biopsies as well, are followed by the changes of the secreted proteins in the supramen. That allowed us now to switch over into our patient studies, where we had those biomarkers already profiled with plasma and urine proteomic studies. We could now show that the same proteins responding to the intervention in the organoids are also seen to be increased in patients with the disease in close correlation to the tissue level transcript here in the urine compartment, and thereby giving us a urine signature of the impact of TNF on kidney tissue we already have shown in the organoid compartment and in the in vivo setting in associations. And now we can use a simple urine test to identify which patients have TNF active and bring them to clinical trials. That's exactly what we did. We just completed phase 2 study as proof of concepts to see if we can bring people with high target engagement biomarker activity to an adalimumab trial and see now in this stratified manner if we see responses of our target engagement biomarker to TNF inhibition, and hopefully also responses to the proteinuria outcome. The similar strategy is not deployed against TNF, but also against other molecules active in our patients in distinct subgroups, so that we can bring the matching concept forward to bring the right patients to the right treatment in our rare disease. This was one example how I hope I could convince you bringing multiple multiscalar data sets together in vivo and ex vivo can allow us to define cross-cutting disease mechanisms effectively by communicating to a large research community. And my last few slides will be exactly on that. What are ways how we can facilitate data mining by research communities who have very diverse scientific languages, interests, and background? And here I want to give an introduction to the Kidney Precision Medicine Project we have started 6 years ago, where similar to what I have described to you for the rare disease, we build a recruitment framework to bring tissue of people with kidney disease to dense interrogation technologies for data integration and then for data share. We capture across KPMP out of biopsy tissue, following the similar strategy I've shown you for the rare disease, a multitude of data sets along the genotype, phenotype continuum, taking advantage of the latest molecular biological technology advances we all are seeing on a daily basis right now. The key question as we studied our ecosystem early on was, how can we identify what data sets and information is needed for which element of our stakeholders? We have developed for this aspect for our user-centric design of the data mining atlas user personas. You can see some of them depicted here, ranging from study participants to the expert research data miners. We mapped our data sources with a standardized knowledge environment and an ontology mapping framework towards different data search ecosystems, ranging from an atlas repository for the expert data miner to patient-level histology exploring tool for patients and their families. If you want to interrogate it further, we would invite you to explore the Atlas in the KPMP framework, where you will see that we have an extensive raw data repository for the expert data miner with carefully annotated rich metadata and open access to the vast amount of the data without need for prior authentication. We have an Atlas Explorer tool for molecular cell biologists, clinician scientists with limited bioinformatic background, but deep insight into cell types and molecular processes where those can be linked. And we have added, in response to the rapidly evolving spatial molecular concept, an Atlas Spatial Viewer system, where the 2- and 3-dimensional aspects of the molecular interactions can be effectively mapped. The tool has been quickly adopted by our community. It's out now for 3 years, and you can see that among the more than 4,000 data sets, we have more than 20,000 data downloads from our open science community in this context. We also are using the data among the network investigators to build atlases out here, for example, as publication from a couple of months ago in Nature from the teams, where we link between data from KPMP, human cell, and HubMap different cells and stages, adding to the cellular distribution of single cell states, the 2- and 3-dimensional interaction space of the available spatial technologies to define unique cellular neighborhoods present in people with rare and common kidney diseases, and then map these cell states to crosstalks among the cell niches, and taking advantage of the orthogonal data in the rare disease network and Neptune, long-term outcomes to identify niches most significantly associated with, in this instance, for example, progressive loss of kidney function, and using the organoid and biomarker data to identify non-invasive surgery. This I would like to wrap up. I hope I have given you an overview how studies linking ex vivo organoid system to in vivo disease states phenotypic captures allows us to validate organoid platforms for use in translational settings, how to bring knowledge from the organoids for potential candidate biomarkers then forward for therapeutic testing, and how to develop data sharing frameworks to enable effective interactions with diverse user communities. I have to really finish with a great acknowledgement to the rare disease research networks and our diverse group of investigators and patients who supported our journey to build the knowledge framework for our community at large. The Kidney Precision Medicine, that's a really unique effort, patient-led, for using tissue to define elements, and our patients in the long-term support. Thank you very much.
Thank you very much, Professor Kretzler, for an engaging talk and some very nice examples of leveraging many different types of data throughout the entire drug development process. So I think Dr. Zhang here is going to introduce our next speaker. Oh, I think you're still muted.
Sorry. Our second speaker is Dr. Peter Dorston, who is a professor at the University of California, San Diego. His presentation title is 3D mapping of microbial metabolites and the challenges in annotation of microbial metabolites.
I think you're muted too.
All right, thank you. That's great. That's perfect. All righty, thank you. As usual, I will change my title. Originally, I was going to talk about 3D mapping, but I think it was more important to get across how we think about database management and curation, because I think that may be useful for this community, or at least to think about it. Before I go on, I have disclosures. I'm a scientific co-founder for these companies or advisor for these particular companies. But I want to bring you to untargeted metabolomics. It's very similar to transcriptomics or metatranscriptomics or metagenomics. You collect a lot of data and then you get some information.
Now, if you did this in 2016, on average, we could annotate 1.8% of the data. In 2023, this has grown to being able to annotate 14% of the data, but only 1 to 3%, let's say you have a fecal sample, only 1 to 3% of that data could be annotated to be microbial. Now, if you were to do sequencing on that same fecal sample, you'll see that the majority of the DNA that's read out will be originating from the microbiome. So there's a discrepancy between our ability to be able to annotate microbial molecules versus what we can do with sequencing. And so if we fundamentally ever want to understand the microbiota, we need to be able to have all the puzzle pieces or get an inventory of these particular puzzle pieces. So what I'm going to try to do today is really sort of give you an example of how we discover microbial molecules experimentally, and then show how we can leverage the repository to do so, and then describe what it took to get there and key future opportunities in terms of data management. Because I realize that most people are not routinely doing untargeted metabolomics. I just want to show you what the data structure looks like. So first, when you introduce a sample into the mass spectrometer, you collect an MS1 ion. So this is an ion that represents the molecules, so protonated, sodiated, potassiated versions. Then we can isolate those ions, and then we can hit those over the head with the sledgehammer. In our case, it's helium gas, and those molecules then fall apart. And we can weigh the puzzle pieces, as you will, of those molecules, and each molecule fragments in unique ways. And so the fragmentation spectra, or the MSMS spectra, or MS2 spectra, can then be collected, and from that we can try to do data science with it. And this is really the speed with which this happens. You'll see that there's a lot of noise, there's a lot of signatures that you get, and that you have to then translate. One way is to translate it into a data table is to provide annotations and sort of quantities, but there's really 1,001 ways to use this particular data. And so I first want to carry you through experimental strategies. So here we fundamentally asked how the microbiome really impact the chemistry of the host. So what we did is we took germ-free mice and mice colonized with a normal microbial community or specific pathogen-free mice. We dissected 29 different organs into 100 different pieces and subjected them to untargeted metabolomics analysis. And so what we found was that in the gut, as much as 70% of the chemistry is different. But even distant organs such as the brain, as much as 20% of the chemistry is different. And I'm not even talking about quantitative difference. I'm talking about presence and absence. And so here we're looking at some molecules that were found in the duodenum. And what you will find is that they were related to a well-known bile acid called glycocholic acid, where we see these blue nodes that were found only in the specific pathogen-free mice, so normal microbial community. And these were related to glycocholic acid through a tool called molecular networking that we introduced in 2012. And we found essentially that glycine was substituted with phenylalanine, tyrosine, and leucine, and we were able to show that microbes are able to produce it. And that's consistent with the observation that they were only observed when the microbes are present in these germ-free mice. And so Emily became really interested in this, and she started culturing a lot of different gut microbiota with unconjugated bile acids. And all of a sudden, she started to see a lot of these amino acid conjugates, proteinogenic, in some cases non-proteinogenic, and they're phylogenetic or taxonomically clearly distributed to certain classes of organisms. So the bacilli, actinomyces, Clostridia, Fusobacteria. And so these are very common organisms that when you do sequencing of a fecal sample, you'll definitely see a lot of those organisms present. But because we built this knowledge base, and I will get into the knowledge base in a little bit, she was also able to reveal that they were disease associated. So very quickly, we were able to translate this information. And in this case, she saw many of these associated with Crohn's disease, not only in this data set, but about 7 other data sets. And so at that time, we did not know what the function was. We tested FXR interactions, and that was in the first paper. But since people have started to show that they can control germination of C. difficile, that they alter intestinal barrier, that they interact with PXR, that they interact with the aryl hydrocarbon receptor, CAR receptor. So there's a lot to be learned. And that will, as we start to discover a lot more of these microbial bile acids, I think over the next few decades, we'll really begin to understand their function But without discovering these bile acids, you would not even have the ability to think about function. And so the question that we had is, are there more? So we had about 170 that we discovered now. And the real question was, are there more, and how can we find all detected bile acids? And so if we go back to this untargeted metabolomics data complexity, I said there's 1,001 ways to analyze this data. But ideally, this data ends up in databases, and right now only a small percentage does. And then you can reuse this information. In our case, we wanted to reuse the information to discover new microbial bile acids. Now, most people think mass spectrometry is really complex data form. You can use it for proteomics, glycomics, lipidomics, metabolomics. But all of this is done by mass spectrometry. And there's only, if you actually look at the data itself, there's only 5 characteristics that make up all of mass spectrometry data. And those are the intact ions of the molecule and their isotopes. Or if you have fragmentation data, the ions of the fragment ions, or the delta masses. And if you do chromatography, you also have retention time. If you do ion mobility, you also have drift time information. And so those are all the 5 characteristics that make up all of mass spectrometry, and all of proteomics that are done by mass spectrometry, glycomics, metabolomics uses these pieces. So we decided we're going to develop a query language that allows us to ask very specific questions about a single file project or at the repository. And really the way to think about this is that this is a way to do data filtering based on MS patterns. So let's say glycolic acids. When you fragment this and hit it over the head with a sledgehammer, you lose 3 waters, and in this case, glycine as well. We lose another water. And then we also see this loss of this, whatever is attached. So if there are modifications attached to these bile acids, they will show up on the left-hand side. So we can build a query. And we actually have a translator for this query, actually into 9 different languages now. But basically, we're asking it to return all MSMS data or all fragmentation data associated with the following conditions: a peak at 337, a peak at 319, and where you take the precursor minus the unmodified version of the bile acids so that we can define and look for this particular ion. And then we search the entire GMPS repository, and some of you might not be familiar with the GMPS repository, but essentially this is a public repository and has 2,800 public projects now, 1.2 billion MSMS spectra. And so we search this repository with queries associated with these 6 different bile acid classes, so to speak. And when we do this, we had about 500,000 matches, about 20,000 unique bile acids that we were able to observe, with about 5,700 different putative modifications at this point. But because we have curated metadata that is part of Redo, where we can do controlled vocabularies of metadata, we can now begin to see where these bile acids are. And so the color coding is based on non-hydroxy to pentahydroxylated bile acids, and then the number is the number of modifications. And you can see that these bile acids are all over the place. In addition, we can readily translate to see if the data associated with animal models is also found in humans. And sure enough, we see similar type of patterns in the human data that is publicly deposited. And so the next question we want to address is, do microbes produce the bile acids? And here we use a tool called MAST. And MAST is essentially very similar to BLAST, where you can take an RNA, protein, or DNA sequence and search GenBank and find data sets that have it or analogs of those sequences. MAST, we now have this for fragmentation data. In addition, what we've started to do is we have started to create domain-specific masks where we curate, let's say, drugs or food or microbes. And so for the microbes, we got together with the community. Basically, these are cultured data from cultured microbes. We curated 60,000 LC-MS runs taxonomically. So now when you search masks, you can get taxonomic information associated with this known or even unknown molecule. And so when we search the MSMS of this phenylalanine cholic acid, which was one of the original ones that we discovered, you could clearly see that there are some classic microbiota-derived organisms that reside in the gut that are able to make these particular molecules. Now we do this for all the MSMS spectra in the in this particular project that we found with the MassQL search. Here on the bottom, you see all the delta masses that we find in these cultured microbes. And on the right, you can start to see, and on the left, some of the taxonomic information associated with these organisms that are able to produce these particular molecules with these modifications. Now, mass does not only allow you to find microbes that make them, but also data sets that are in the public domain. And one of those data sets really gave insight that they're indeed made by many of these modifications made by microbes, because this was mice treated with and without antibiotics. And when you look at the non-antibiotic treatment, you see a lot of these delta masses, whereas in the antibiotic treatment, you see most of these masses disappear, except for the 107, which are the taurine conjugates. These are the host-derived bile acids that are made in the liver. And so it really supports the notion that these are indeed made by microbes. Now, long story short, we started annotating what some of these delta masses could be. An example of the ones that we found belong to the polyamine pathway. So these polyamine molecules seem to be conjugated to these bile acids. We further can confirm, because we found with mass, we found data set matches, we found that they were associated with carnivores, but also sleep disruption and IBD and ulcerative colitis and obesity. So very quickly, without having to collect data, we're able to get this information now. And that gave us enough evidence that we should probably synthesize these molecules to see if we can confirm that these are indeed correctly annotated. And sure enough, we were able to confirm 15 of these polyamine bile acid conjugates, but there are many, many more that exist. So we've now confirmed hundreds of new microbial bile acids, thousands of new candidates, but what did it take to get here? Well, the first thing that we needed was an algorithm that can link all this MSMS data. So it's a form of spectral alignment, but we can build a large knowledge base with this. We then, to capture knowledge from the community, we built a GMPS ecosystem where people can essentially analyze their data to do molecular networking. That was the first tool that we introduced. But what we did behind the scenes is we converted all the data, including data from standards of known molecules, to a uniform format, initially as an MGF format, now MZML format, so that you can do co-analysis across the entire repository. Then we built a redo system, which really controls metadata vocabularies to each file. And then combined, this generates data tables. That's really what GMPS does. It generates data tables so that you can use it in other analysis tools. So if you want to do statistical analysis, you can use QIIME2 or Cheetah, but there are lots of other tools that we've been able to link to, about 50 plus different tools. And then you can build search engine. The first one you already heard about was MAST. However, MAST is incredibly slow. The first generation, it took for a single MSMS spectra to search about 110 million spectra, took about 20 to 40 minutes. So we needed to have a faster one, which is now about 10,000 to a million-fold faster than the original one. So that allows you to analyze entire data sets. We've started to index all of the data, and that is making it even faster. And that is really enabling MicrobeMass and MassQL, and you saw how that can be leveraged. But there are other resources that are available. And so just like the previous speaker, they're generating their own databases, and lots of people are generating their own databases. And it's actually really hard to discover these databases, and how do you interact with these databases? So two metabolomics databases that exist are Metabolon and Metabolites. Initially, what we used to do is we just downloaded data that we thought was untargeted, added to GMPS so that we can search Metabolon and Metabolites as well. But since then, we've built a dashboard that can read out directly from Metabolon and Metabolites. And now we have a beta version of a GMPS ecosystem that essentially allows you to do molecular networking and soon other analysis tools by directly leaving the data in those repositories, but still allowing co-analysis. So this is an exciting step in the right direction. But the real reason why this is so powerful is because the community really felt the need of depositing the data, curating the data. And we work by the carrot model that if people upload data, that it becomes more useful to them when they upload it. And that gives an incentive to begin contributing. Now, what are some of the gaps and opportunities? First of all, I think I'm starting to see the limitations of the MCML format. This is an open format. The reading of this format is really slow. So we need to be able to get data into a format that can be read more readily. So right now we do 1.2 billion spectra, but imagine when we're going to the 10 or the 100 billion and across repositories. That needs to be faster. Metadata is really hard. There needs to be strategies to harmonize metadata. We need build mechanisms to self-check analysis. So let's say somebody gives an annotation. If somebody else had that same data, would they give the same annotations? That's a big question. And there may be other kinds of self-check analysis that can be built into the system. We need to, in our case, make tools such as mass easier to interpret. Micromass taxonomically is one thing, but maybe disease states, tissues, sample distributions, etc. How to prioritize, not entirely sure, but I think it's very useful. And then finally, we need to be able to do effective linking across data types. So when we do micromass, we get matches to microbes, but not all microbes may be present in the particular sample. So maybe if we can link it to microbial sequencing data to filter out, now we can start to say, okay, which microbes are likely responsible for producing these kind of molecules. And this goes beyond sequencing. We need to link to activity databases, et cetera. So there's a real opportunity there. And finally, I need to acknowledge a wonderful team. I get to talk about it, but it's an amazing team I get to work with. And of course, funding from the NIH that supports a lot of this effort. And then finally, I'll take questions at the end of the session when the next two speakers are done.
Thanks. I will hand it to Abby for introduce next speaker.
Yeah. Thank you, Dr. Dorsine, for taking a deep dive, I guess, into a newer omics and what it takes to be able to link it with other omics and enhance its utility. So our next speaker is Professor Matthew Might, coming to us from the University of Alabama at Birmingham. And he's going to be talking to us about the patient scientist perspective.
All right. Thank you. I really appreciate the opportunity to speak today. And I think you'll notice sort of a somewhat of a shift in perspective from the prior two talks. And I'm doing this deliberately. I want to approach this from the lens of what it's like to approach i3D from the context of a patient advocate. And still try to answer some of the same questions on the other side. I have to start with a quick disclaimer. The disclaimer is that while I do precision medicine for my day job now, I run the Precision Medicine Institute at UAB. By training, I'm actually a computer scientist, and this may be a helpful hat to wear during the discussion later on when we talk about resources, for data or platforms or things like that. But in the context of this talk, I really want to speak from a patient perspective because, most of what I did for a long time was actually stuff like, cybersecurity and high performance computing and a lot of artificial intelligence. The interface, using language as an interface between, humans and machines. The reason I'm giving this talk, the reason I know anything about stem cells and organoids is that my son was an ultra-rare disease patient, and this prompted a number of initiatives that we'll talk about that led to ultimately the creation of stem cells, organoids, and things like that for his disorder as a partnership. So the second disclaimer here is that everything I'm talking about today, everybody else did the hard work. I was really serving as the cheerleader for all the work you're about to see today. And so I owe some big thank yous right up front to NCATS, probably most of all, to Travere Therapeutics, who was a critical partner on this project for a long time, and to the team as a whole that formed to really take this on. So the paper that I'll talk about most is this one right here. And this is the team that I want to give thanks to right up front. So to all of you cited right here, thank you for the work that you did in making this possible and giving me my education in stem cells and organoids. So there's really three major points I want to get across today. The first is, why me? And it's important to set that up right at the start for why I'm talking about this. And then through the lens of that personal experience, what does it look like to actually use this technology in practice when your goal is to find treatments for an ultra rare disorder? And from there, you use that to cover why there are such big advantages to this approach, and use all of that to then back into the fundamental theme of this session, which is, okay, if the goal is to help find treatments, and there's good rationale for doing this, how do we accelerate that part of it with the data that's coming out of this? And how do we be mindful of the kind of data that's coming out of this? And what's great is I already see some commonalities with the prior two talks about things that we want to start thinking about in terms of an ecosystem for this kind of data. So to start with, why me? Well, for me, it begins with my son, Bertrand. This is him when he was about 6 months old, and the start of his diagnostic odyssey. So by 6 months, we knew something was not quite right. And I'll fly through this part of it. We began on a place that a lot of patients call undiagnosed island. And when we were here, the major theme was uncertainty, the things we didn't know. And the arc of the next 2 minutes of this talk is a rapid increase in certainty, much of which is due to stem cells and organoids. So, when he was young, we had no idea why he was having seizures and movement disorders and severe developmental delay, a lack of tears. All of that was totally mysterious. And in fact, he was even given a life expectancy early on of just 2 to 3 years. Fortunately, he lived much longer than that in the end. Part of that was due to a series of breakthroughs, the first of which was exome analysis or exome sequencing analysis. And what it found in him was that he, in fact, had 2 mutations in a gene called NGLY1 or NGLY1. And the sort of shocking revelation that he appeared to be the very first NGLY1 deficiency patient ever discovered, which meant by extension at the moment, he was the only one. And this kickstarted a lot of efforts, spurred on by me, done by others, to sort of really understand the science of this disease. So this is not a talk per se on NGLY1 deficiency, but I'll give you some context. NGLY1 is it's a deglycosylation protein. In fact, it's responsible for deglycosylating misfolded glycoproteins after they are retrotranslocated from the endoplasmic reticulum into the cytosol. And as a result of its absence, you start seeing an accumulation of things like misfolded glycoproteins. So if you fast forward 10 years now, you will see, that was essentially the sum total of what we knew about the disorder back then. Fast forward 10 years, we know a lot. We know that it has more than just a sort of recycling component to it. It's actually important in activating certain proteins as well. Thanks to a natural history study conducted at the NIH Clinical Center, courtesy of folks like Bill Gall and Wolf, we learned a lot about the patients. And all of that eventually started to dovetail with what we were learning from stem cells and organoids as well. When that protocol ended, NCATS picked it up in the form of an RDCRN on glycosylation disorders, which allowed these patients to continue enrolling in a natural history study. So I'm grateful to NCATS and Eva Mareva on this one. So Eva is the one leading this particular RDCRN. And then another thank you to NCATS for a joint project with Travere and a nonprofit foundation that I was running at the time that allows to really search for therapeutics for NGLY1. And this is the project that ultimately enabled us to utilize these kinds of technologies in that pursuit. Just to give you a little more background, there's obviously more than my son was just discovered over time. There's dozens of patients now. And the unifying characteristic of all of them is that they appear to have sort of a total loss of function in this particular gene, one way or another. The major features are pretty consistent, severe developmental delay, they're nonverbal, real issues with movement and walking, and that lack of tears, and for a number of them, seizures as well. So that's why I'm speaking to you. What I want to show you now is the kinds of findings that started to come out of this work, in particular, when it comes to stem cells and organoids. So again, just an extra thank you to NCATS for making all of this possible, and especially to Wei for leading this team effort, because this really was a unique partnership. between NCATS and a nonprofit and a pharmaceutical company. So at a very high level, here's how the process went. We started off with patient fibroblasts. We turned those into iPSCs, those into neural stem cells, ultimately into corticoglutamatergic neurons, and then finally into midbrain organoids. At every stage downstream, we started doing multi-omics analysis like transcriptomics and proteomics, and this started producing a wealth of data, as you'll see in a second. So there were 4 lines in total taken from patient fibroblasts. And here's a sampling of the kinds of things we learned once we got to just the neuronal stage. So from these corticoglutamatergic neurons, it was clear that there was some kind of disrupted calcium signaling. And what I think is interesting about this is that, from some of the functional study that spun out of the natural history study, there was other evidence building of disrupted calcium signaling, in particular from some of the dental stem cells recovered from some of those patients. So again, a convergence between this basic science and the clinical research is a key theme over and over again. But with the derived neuronal cells here, we could start doing deeper analysis. We could look at the transcriptome and look to see, okay, well, what seemed to be driving this disruption in signaling, and really what was the nature of it? And so this became a powerful tool for gaining insight that wasn't easily obtainable using sort of directly patient collected materials. From there, of course, we did go on actually to developing some of these midbrain organoids. And here they were developed for up to 90 days and tracked using neurodevelopmental markers to watch them mature. And I just want to share here some pictures that are some of my most cherished pictures, actually. So as a member of this project, I actually got to go to NCATS and visit these cells and learned a few things about them that I, as a computer scientist, never would have known. One thing I learned is that midbrain organoids are what they called sticky. And so if you don't sort of keep them in constant motion, they can, clump together and stop functioning. And so that's what this device here was meant to do, was to keep the organoids from clumping together. Even stranger was the ability to actually hold them directly like this myself. Got to look at them, it was very strange, actually. I mean, in a really wonderful way to actually see this even at a high level. I can tell you from a patient advocate perspective, it means a lot when organizations go the distance to do things like this on behalf of patients, even before what science is coming. It's meaningful to see these artifacts exist in real life. And of course, we started learning things. So not surprisingly, what we sort of hope to see, you don't see NGLI1 protein in NGLI1 deficient organoids. So that's sort of a baseline finding. And then I think this is sort of a recurring theme, is that you can do a lot of hypothesis generation using these broad spectrum omics techniques like transcriptomics and proteomics. So you can look at things like differential gene expression between the two. And sure enough, if you look at differential expression between RNA and protein, you do find this clustering of differentially expressed genes. And if you then apply something like gene set enrichment analysis, you start to hone in on specific mechanisms that are at play. And this was really important, because there was this huge gap of understanding in between the very, very low level when it came to NGLI1 deficiency and the very high level, the clinical presentation. And I think this is the work that really started to fill in the gaps between them, in terms of understanding, well, what specific mechanisms are happening neurologically to explain what we're seeing at a very high level. And it's something that was hard to see without doing this, because if you look at cells by themselves, for many years, individual cells weren't yielding much insight. The insight really started to come with organoids. And in my experience with another hat, where I help lots of other patients do these kinds of things, that's true a lot of the time. You often need to move beyond single cells to start to see these rich mechanistic phenotypes emerge. There's really no way around it. So doing this sort of broad spectrum work is what tipped off or sort of formed specific hypotheses that enabled deeper, more targeted functional studies. So, for example, it's clear that these astrocytic markers like GFAP were reduced. And you start to know where to look based on the broad spectrum analysis, and that gave us more insight into the mechanisms at play in the diseases itself, and also gave us insights into neurotransmission, which we knew to a certain extent from very high level studies, and in the natural history study was showing signs of disruption. So here's some GABAergic disruption beginning to emerge in the organoids, which again is harder to see at the level of individual cells, and you can only see at a very coarse level, of course, in the patients themselves. And then I think one of the more actionable insights to come from this was a very specific deficit in dopaminergic activity in particular. And the reason I highlight some of these in particular is that in some model organism work going on at the time, and some of the findings from the natural history study, this dovetailed nicely on some of the predictions around what might be high priority candidates for drug repurposing, in particular drugs that very specifically modulated neurotransmission in a very targeted way. And so it even enabled us to have enough confidence to launch a little N of 1 trial under the glyco-focused RDCRN of aripiprazole. So it started off as just an N of 1 trial. So, when these findings really connect the dots, they become powerful in what they can enable in terms of meaningfully moving the clinical side of the science forward for patients. And then, of course, it's worth talking about, I think you can see why it was helpful in my son's case and for his disorder, but more broadly, we can think about why this technology is so critical. Every finding that I just showed you could be, and in some cases did become, another drug target to go after. Each finding could lead to an assay that could lead to development of, some sort of high throughput screen. And each finding could become a biomarker or shine a light on a biomarker that could become very important downstream in a clinical trial. And more generally, and again, sort of zooming back out from the NGLI-1 perspective and thinking in terms of the work I do with many patients at the Precision Medicine Institute, it's just hard to study rare diseases. We have small patient populations by the definition of rare. We have very limited availability of critical biomaterial. And so this is a way to generate that biomaterial. In particular, some of that biomaterial is just fundamentally inaccessible, and in this case, like for things like brain, we can make the inaccessible accessible and study it. And ultimately my son did succumb to this disease, but and he was the first to donate his brain and provide actual patient models of this disorder. But it's not lost on me that for several years of his life, we already knew a lot about what was going on because we had these organoid models of NGLI-1 brains, whereas previously that might have been the first time we learned anything about what was going on. Of course, we have often very complex phenotypes when it comes to rare disease, and they differ system to system. So this is an important tool for studying them in the context of the individual organ systems. And as I mentioned earlier, sometimes there is no phenotype in an individual cell, or at least an easy to find one. Or it could be that the vast majority of cell types don't exhibit some kind of readily accessible phenotype, and so this is a way of getting higher up to get to those critical phenotypes that are necessary for what I just mentioned earlier in terms of identifying drug targets, developing assays, creating biomarkers. And in my personal experience, both with NGLI-1 and with a host of other disorders, there's less loss of coherence than when you go all the way to an animal model, which was in many cases sort of the next best thing you could do without something like an organoid, where you'd have to jump to an entirely different system. And sometimes you can't make that jump. There's no reasonable ortholog available in any reasonable animal system to work with. And then to conclude, I can think a bit about now from this experience about what it would take to build better resources for data here. And it's clear, from our work that the transcriptome and the proteome are broadly useful. From prior talks, also clear that metabolomes are broadly useful. And so that leads me to believe that, we really do want to start thinking about a common repository for all of this data. And I think also the interpretation of the data too. I think, in some cases, it's going to be hard to get an apples to apples comparison. So knowing the downstream results in each case, to the extent that those can also be collected, would be very helpful. And of course, if we're going to have a common repository, having a common, analytic framework, pipelines for things like differential expressions and gene set enrichment analysis, having that all available in one spot will certainly accelerate these kinds of workflows. So with that, I thank you and look forward to questions during the discussion.
Thanks, Matt. Sorry, go ahead.
I was just going to say thank you for sharing that personal account and how we can create, I guess, this common analytical framework and also data framework for future research.
Yeah, I want to echo with Abby. It's important to create this new kind of broad application system for the not only collecting data, also, as Matt mentioned, for interpretation of those common data. It's very important if you have a large amount of the data, but people look at that, only a few people know the computer software, the knowledge you can use, that's kind of not enough. We would like to have a system many people can apply for that. Okay, our next speak is the fourth speak, the last one in our session is Dr. Monko Lek, who is a professor at the Yale University School of Medicine. His presentation title is Biomarkers and Omics in Rare Muscle Diseases.
Cool, thank you. Hopefully everyone can hear me. Are we all good? And the presentation's okay? Cool. So firstly, I'd like to thank the organizers of NCATS for giving me this opportunity to talk about a little bit about the research that we do here at Yale. And just, one disclaimer is, like a lot of the people that have given a talk, I'm directly impacted by rare disease. So I have a ultra rare muscle disease myself. So hopefully the things I talk about, although it's applied to rare muscle diseases, can be easily translated to other rare diseases, because it kind of relates some of the challenges and some of the omics data that I'll touch upon today. So first of all, I'm going to talk about the challenges and barriers in studying rare muscle diseases and the challenges in using animal models. I know I'm preaching to the choir, but I do want to touch upon this point. So one of the big threes in terms of the most common muscular dystrophies is fascicular skeletal humeral dystrophy, or just FSHD. It's a bit of a mouthful and people just say FSHD. So it's, like I said, one of the big three, one of the most common forms of muscular dystrophy, with the incidence in 1 in 8,000. It's autosomal dominant. And one of the hallmark features is it's asymmetric, the muscle wasting that we see, and affects the facial, scapula, and humeral muscles, and hence the name. And the age of onset could be is quite variable, but many is early adulthood, and that but there are some with early childhood onset. So currently there are no treatments, but there are some promising ones in development. In terms of the disease mechanism, it's caused by a transcription factor called DUX4, which is misexpressed and sporadically turned on in patients with FSHD. And so the activation is rare. So it's about 1 in 1,000, 1 in 5,000 cells. And it begins in a central nuclei, as you can see, and then it spreads across. So myotubes have multiple nuclei, and sometimes this signal can spread across. And the cellular fate of DUX4 expression is apoptosis. So what I'm showing you here is a patient cell model created in our lab, where we have a GFP reporter cell line, which has the DUX4 binding site. So you can see sporadically switching on in a population of these patient cells and then shriveling up and dying, which is the apoptosis, within 24 to 48 hours. So unlike most muscular dystrophy, it has a very good cellular phenotype we can actually study. So emphasis on unlike. So here's the challenge. The DUX4 gene is not in mouse or a lot of animals. So it's only in a subset of primates. So in order to study it in animals, we're actually studying it in artificial context. So as I said, so DUX4 or repeats of these pseudogenes found on the subtelomere of chromosome 4, and it's caused by two different genetic mechanisms, either contraction of this repeat region, allowing DUX4 to be misexpressed, or the changes of the epigenetic landscape that also allows for misexpression of DUX4. But these are some of the biomarkers when it's misexpressed that we do see in patient muscle biopsies and also cell types. So ZScan4, TRIM43, and this gene here. So this is us showing these genes being switched on in an inducible cell line where we can doxycycline induce DUX4. So here we've created new cell lines which behave similar to the one colored in blue that's currently used by the research community. The difference with the ones we've created is that it has the whole gene, so we can look at knockdown approaches also, amongst other approaches. But the problem is when you misexpress DUX4 in animal models, a different set of genetic biomarkers are actually switched on. And if you was paying particular attention on the last slide, you can see these genes are actually different. And so sometimes these are actually used in clinical trial studies, but then the question comes with these genetic biomarkers, are we actually treating a different disease altogether? Because we're looking at different genetic biomarkers. And this is just highlighting some of the challenges we have in studying some of these diseases. The genetic biomarkers are different, and it shouldn't just be an exercise of just reducing the ones that we see in a different animal model. And so these are another examples of things that I work on that are impacted when we do animal models in terms of differences in the biosynthesis pathways in humans and animals, and also creating difficult mutations such as multi-exon duplications instead of using patient cells. So sometimes creating this is just as challenging as creating a therapeutic approach itself. So that's touching upon some of the challenges that we have in the muscle disease space. So now I'm going to transition, just a food for thought for discussion. Next, I'm going to transition to some of the omics approaches that we're using in muscle disease, particularly in my lab, but this applies to other diseases too. So the first of them is, can we use CRISPR screening to identify potential genes and pathways that can be targeted? So in the case of FSHD, DUX4, the transcription factor gets turned on, and then this turns on genetic programs, many different genetic programs, but we don't know which ones are toxic, and it's actually creating the apoptosis. So one way we can actually address this is actually through CRISPR screening. So as I said, the induction of doxycycline creates cell death within 24 to 48 hours. This is a very strong phenotype we can look in this disease model. And this is a cell line created by our colleagues at the University of Washington in the Tapscott lab. So the approach we wanted to take is using CRISPR screening approach. What genes, if we knock out or activate, do we actually increase that cell survival? So we have surviving cells that we can now sequence and ask which guide RNAs underlie either the knockout or the switch on of the genes that lead to cell survival. So in terms of the knockout, so in this CRISPR screen, which we published quite a while ago, one of the top reproducible hits are actually HIF1 alpha, and this is actually HIF1 beta, and also other members of that complex underlying the hypoxia response. So I thought this was too good to be true, but I was actually analyzing this data, but it was not expected to have some of these hits actually being the same pathway. And then we can go and actually then test it. So here we're testing it by going back and knocking out the genes and asking using a caspase assay, do we still see apoptosis when we switch on DUX4? So you can see the green represents the signal when we switch it on, and you can see the reduction of the green in a lot of these knockouts that we see here, validating the hit, but just using a different guide hitting that gene. We can then ask the question, which genes, if we activate, do we actually increase cell survival? And two of the reproducible hits are EGLN1 and EGLN3. And here is us just validating it by using a different set of guides or just overexpressing it itself in these cells and decreasing the amount of apoptosis and that we actually see in the presence of doxycycline when we switch it on. So if those, for those that don't know, these are actually inhibitors of the hypoxia pathway. And so if we activate the inhibitors, if you can think about it, then that will act upon the hypoxia pathway in a similar fashion. So knowing this, then we can then ask the question, what FDA-approved drugs are out there that can actually act upon the hypoxia or the HIF1 pathway, either upstream, downstream, or somewhere along that. And we can see and then we can use these drugs and ask that question. So you would have seen in some presentations earlier today that rapamycin and everolimus seem to feature a lot, seems to be the panacea for everything, but we also saw some effects here acting upon the mTOR pathway. You can see here again, this assay, the caspase assay, looking at apoptosis. When we switch on DUX4 without treatment, you can see a lot of green cells, and you can see the reduction in all these drugs that are actually applied, and you can see this being quantitated here. So this is a neat approach we can take if we had a strong cellular phenotype in the context of rare muscle diseases. However, a lot of the rare muscle diseases do not have a strong phenotype like the switching on of DUX4. So this is actually a challenge. So what we've used, where we've used 3D tissue culture models is actually looking at contractile properties and also calcium handling properties that don't become apparent until you create these contracting muscles and you can actually measure it. But how can we actually screen it in a high throughput manner, which is actually the challenge? Because a lot of these muscle diseases, they're actually caused by structural proteins that don't lead to such a strong cellular phenotype. So this is the challenge we have in the field. So another question we want to ask is, are there other biomarkers that can be discovered? So one of the stock standard biomarkers we use in muscle disease is CK, or creatine kinase. This is leaked into the bloodstream and is a good marker of muscle damage that we continually see highly elevated in skeletal muscle disease patients. So the question we're going to ask is, can we discover other biomarkers? And what omics technology we want to use is actually Olink. So this technology uses probes. And so when the probes attach the protein target, you can see it hybridizes, and then it's actually a sequencing approach. Now it becomes just a sequencing approach, send it off for sequencing, and then identify the tags that correlate to that protein. And it's very quantitative because of the way it actually works. So we're actually using the Explore 3072 panel, which is a panel of all the panels available. And this is a way we can ask a hypothesis-free question. So this panel has been used at scale in a cohort around about just under 55,000 samples in the UK Biobank. So being also a data scientist, what I think is, well, we have a great set of controls when we produce data to compare against if we're using the same technology, normalization, and processing pipeline. So we have a great set of controls, which makes it even a good choice of sort of like method to use. And you can see here in this plot here from the bioRxiv publication, they're just showing correlation of age, sex, and BMI. So with BMI, not very surprising, leptin is one of the top hits you can see here. But this is a great data set that we want to leverage and use this to compare against healthy controls. So we want to use a Olink data set specifically for FSHD to answer one of these long-standing questions. If you remember, if DUX4 activation is rare and sporadic event that leads to eventual cell death, how does that actually cause disease? If it's only happening in 1 in 1,000, 1 in 5,000 cells, how does it actually cause disease? So there are two hypotheses out there. Is one that DUX4 is expressed at subtoxic levels and it's enough to impact gene expression profiles. So what we're seeing in cell culture and in some of our assays is the more extreme case that leads to cell death would be one hypothesis. The other is DUX4 toxic factors are actually released into the environment affecting nearby cells. So these are two hypotheses that the field thinks how a sporadic event can lead to a whole muscle or whole body event. So one thing we can test with using Olink technologies is the second hypothesis. So the idea is we can look at the inducible DUX4 inducible cell lines and also the patient cell lines and ask the question of, in culture, what do they actually secrete that we can actually measure to see, are there actually biomarkers that are actually specific to DUX4 expressing cells? And then we can use mass spec to validate it, or we can use patient serum that have been collected in many different natural history studies to actually validate, do we actually see this indeed circulating in patient blood? So this is an ongoing project, but this is just to touch upon some of the things that the muscle field is generating. Some of the challenges is that Olink is very expensive, okay? I'm an assistant professor, just let everyone know. And it's about $80,000 to $90,000 per plate of 88 samples. And there's no good way of plate sharing. Being creative, I thought about plate sharing with my colleagues at the Broad Institute. It's still not set in stone. So you have to find 88 samples and run it. Sample preparation for non-standard use is challenging when using it for discovery, but outside of just using it for blood, next thing is large sample numbers are required to get statistical significance, especially with smaller effect proteins. This wasn't a challenge in the UK Biobank because they had a lot of samples and they're interested in sometimes common disease. We're actually interested in rare diseases. Can we get enough of these samples at the same quality? And it's targeted, so it's only limited to what is actually represented in the panel. So not all the things that are on these panels may be relevant to muscle disease. They may be relevant to common diseases or more common conditions, but it may not be relevant for a particular rare disease. So as they build out these panels, it will become more and more, I think, inclusive in terms of that. So I'm going to end off with single cell omics to better utilize tissue biopsies. So when clinical trials happen in the muscle disease space, they get a muscle biopsy. So they aren't sampled that many times given the invasive nature of it and how much it hurts. I've given 2 muscle biopsies and it hurts like hell. So I wouldn't be giving too many during clinical trials. So a lot of times they're only looking at a protein biomarker. If it was, say, a gene therapy approach, they're just looking at the restoration of that particular gene and protein, and also restoration of other proteins that may be stabilized at the membrane. So single cell omics may be another way of, fully utilizing that muscle biopsy, given how invasive and how hard it actually it is to actually get. But in order to actually use it, you have to actually create a healthy reference panel. So I touched upon the UK Biobank with the Olink work that they did on that project, and we've seen advantages in the GTEx project also, looking at just bulk RNA-seq. So what we're working towards, and this is a Chan Zuckerberg initiative funded project, is the Pediatric Skeletal Muscle Cell Atlas. So how does muscle change on a single cell level as muscle develops through pediatric development? And it's important in terms of pediatric development because different muscle diseases onset at different times. And it's also a good resource, I forgot to mention, if you are going to create 3D models, to ask the question of which age group does it most look like these immature muscle, sort of like 3D models we create, what does it look like? The other thing we really want to touch upon is diversity. So diversity in not just ages, but also sex, so male, female, and also ancestry diversity. And unlike most organs, muscles all throughout the body, do we also see different patterns across different muscle areas with a focus on quadriceps. The reason why there's a focus on quadriceps is this is the one that's actually taken for diagnostic purposes. So when using this as a reference panel for disease, we want to pick the muscle that's actually used in diagnostic criteria. So the technologies we'll be applying will be single cell, single nuclei, and also ATAC-seq on each one of these samples to actually create this profile across age, sex, and also diversity. Yes, we don't have the power in terms of the number of samples we have in each group, but you have to start somewhere and then build from there. So, and we're also building tools. So two of the members of this large consortia, Doug Millay and also Ben Cosgrove, have created some of these resources for the community use. So this is single nuclei, and actually this is mouse tissue and how it changes as mice age. And while the Cosgrove lab has created this sort of metadata set where they've taken all the single cell work that's being used, that's been created by the community and aggregating it both for mouse single cell muscle work and also for human in their SC muscle 2 release. So there's some of the resources that we want to actually build also on the conclusion of this project. And so with that, I just would like to acknowledge for the FSHD work, all the colleagues involved in that and collaborators, also the colleagues and collaborators involved, the CZI pediatric work. And I'm going to put a small plug because this is a great community to talk about this. I'm actually an editor in Disease Models and Mechanisms, and we have a special series going out, and it's very related to what we're doing right here in this presentation. So if you're interested, do drop an email and submit a paper on this. So with that, thank you. And I'll take questions during the discussion.
Thank you, Professor Leck, for shedding some light on how sometimes the 2D and 3D models, our animal models, are not conducive to high throughput screening, and also for bringing up some of the single cell work. So at this point, we're going to move to our panel discussion. So a reminder to our audience to please post questions in the Q&A if they have any. And we'll kick off by one general question on what sort, so this is for all our speakers, what sort of data readouts would best serve AI and ML efforts in translational research?
Well, maybe a high-level answer first. Consistent ones and well-structured ones. So, you take any biology hat off for a second, putting just a computer scientist hat on. You're going to get far more out of almost any AI or ML technique if you're going to have highly structured data coming in. So making sure that any data we generate, the full structure is captured in easily exchangeable formats is going to give us a big lead in getting the best bang for buck out of all the data we generate.
And that's fun for scientists, right? Because we scientists love to capture all the nitty-gritty details of the metadata. But this is really a steep learning curve for scientific communities to understand that your metadata, your experimental conditions, your sample acquisition protocol are as important, or actually sometimes even more important than the data itself. And finding a good mechanism also for funding agency to incentivize that activity is absolutely critical. Because yes, we have data sharing mandates, then have metadata sharing mandates in the sense that we have to build out a culture that everybody wins. That if your data is captured from your organoids in a way that you know the differences and similarities you see between your organoids and another organoid study, and to the rich, human atlas efforts underway now across nearly every organ system and in many disease states, that you will win on all levels. Yeah? And this is a new scientific culture we will have to grow.
I'm going to jump in, and in terms of training it, what I'd like to see is a lot of exploration of the universe of possibilities or the variable space. And if it's impossible to do that, given how big it is, strategically pick it with data training in mind. So, you know, looking at, okay, these are redundant data points, and whether you can get orthogonal ones also for the training of the data itself.
Yeah, I'll chime in as well. One of the key things is to have, if you're going to do AI machine learning, if you have benchmark data sets that allow you to quickly evaluate how effective your models are working, or how well your algorithms really work to pull out the hidden data phenotypes, which is really what AI and machine learning really does, that would go a long way. And then, in addition, as was mentioned, to have a good, decent structure associated with the data, including metadata. Then you have a win sort of scenario that you can really develop solutions really quickly. If you don't have benchmarking, well, then somebody is going to have to curate that over and over and over. So it's worthwhile to really spend some time on that.
And replication cohorts. And replication cohorts is diversity. As we have heard, you know, diversity on all accounts, diversity on genetic accounts, on socioeconomics. We are very good in genotyping. We have to get a lot better on phenotyping, because the gene-environment interaction is where diseases is happening. And there are ways to capture that, comprehensive tools available. If we prospectively develop cohort studies like NCATS does so greatly, to include environmental exposure data capture instruments, is really nowadays a condition sine qua non, so that we can link our biological and genetic data sets back into this framework. And then look across. We found the compelling evidence of the signal when we replicated in Europe and in sub-Saharan Africa. And that gave us reassurances that our machine learning tools were picking up signal and worthwhile to pursue.
Thank you. I'd like to dig a little bit more into the standardization and the metadata, because I think that you see a lot more standardization that happens within certain fields or even within certain data types, maybe within a certain consortium. But when we're looking for a very broad applicability here, that is across many different types of data sets, across many different types of diseases, how do you start approaching the standardization? And for you, which are the key elements that are absolutely necessary for this to work, meaning that the data is actually useful?
Yeah, I can describe how we do it and sort of thinking about it. Within the context of metabolomics within our GMPS ecosystem, we basically created a metadata template that people can fill out, where we have controlled metadata vocabularies. There are certain fields that are required, other fields sort of open-ended, because you can't capture every metadata in a controlled fashion if nobody has created an ontology for it. And then we have a metadata validator, so that once you create a metadata table, that you then also validate the metadata itself. And in our case, we're just validating if it's compatible with our system, if the metadata entries are uniform and consistent based on the ontologies that we have. So this can work for disease ontology. There's a nice ontology for that. Organ and biofluid ontologies, they exist. In our case, mass spec ontologies exist. But as I said, there are many cases that an ontology does not exist. And so there, the validator doesn't do any checking, but we allow that metadata to take place. There are some different systems that people are trying at the moment, or different approaches. Let's say you're trying to get data from different repositories, different data infrastructures. Well, every single one will use different metadata templates. Every single one will use different nomenclatures for the same thing. And so there are some sort of language models that people are trying to use to see if they can harmonize it within the context of that study. So it wouldn't be across studies, but within those studies, that one person calls it a male or a female with capital M, little m, other study may call boy, girl, that you can use language models to harmonize that information as best as you can. And I think it will be a combination of those two. I still think metadata that's carefully curated and checked, in other words, validated, probably wins over anything that we can do with LLM that tries to integrate multiple different sort of notations for the same thing. But so it will in the end be a combination of the two that is going to be powerful.
Yeah, I'll second that. LLMs certainly have a role to play potentially in cleaning up data. That said, of course, yes, it is best to have it harmonized in advance as much as possible. And I think if we do end up moving in the direction of a common repository, the repository can help to enforce some kind of standards to say, when you check your data in, please also specify the level of metadata standards compliance for any given form of data you're checking in, so that maybe the repository even gives you a score to gently nudge you in the direction of including metadata along with your submission to the repository.
Yeah, but really think carefully how you structure your incentives and your penalties. You want to make sure that low resource communities can contribute sample and data. And if we build out excessively rigorous standards, then we will miss data from low resource community. And often those are the ones who are carrying the largest disease burden. So building out an intelligent way to balance rigor and reproducibility in your data versus comprehensiveness of your data sets. And those will be pushed in different directions. And that's where obviously language models can help to some extent, as they will allow you to pull information in which is not a priori available from some of the communities. It goes the same with scale, and having lived on both parts of the extremes here. In the kidney precision medicine projects, we are doing what we just heard, protocol research biopsies from kidney. They are risky procedures. It's a true donation of a study participant to the community at large. And therefore, we are taking great extent to capture all metadata. All protocols are up in protocol IO so that they are easily accessible for the community at large. And we try to go as deep as possible. And then, obviously, to honor the commitments of participants, you want to make sure that you cross really all these. On the other hand, we have large archives available around the world of kidney biopsy tissue in FFPE blocks. And the tools are coming available this month to actually use these archived material for multiscalar data integration. But there, obviously, extraction of relevant information will be limited and metadata will be limited. So finding the right balance and having open community dialogue, which elements of the community you would need to bring to a study, and not only the well-resourced one, in patient populations who are actively engaging with our scientists in the first place.
Thank you. We have a complementary question in the Q&A that I think, Professor Kressler, you touched on a little bit, and that's people are using different cells for controls, and sometimes it's hard to compare between different data sets, right? So here we talked about how do you submit data, but also how can you generate data to enhance globally, right, the utility of the AI for AI ML efforts. Any thoughts and comments there? So maybe I can rephrase a little bit. I think Professor Kressler, you mentioned about using standard models, for example, to generate data and such. Are there any other efforts that can be made to help standardize the data generation itself?
I think what is very helpful, and I try to quickly touch on it, if we have profiling technology who allows us to validate what the model system actually is representing. And the beauty of organoids is always the cellular complexity they reflect. It's also a challenge. If you take an organoid in a blender, then slight changes in cellular composition in your organoid will shift your analytes in one way or the other and will render your data irreproducible. So finding resolutions of your assays who are down to the individual cell differentiation and to capture that so that you compare apples to apples. And to compare your kidney filter cells at the same degree of differentiations between your different systems. The beauty is, these tools have become available with many of the Atlas programs right now, Human Cell HubMap and others, to do that out of tissue level and can be directly applied also to ex vivo organoid platforms that we define cellular subphenotypes, and then we can compare those subphenotypes. And if you see a significant shift in a cellular phenotype, then you know that that experiment is off to a certain extent and can serve you as a sanity check if you cannot a priori establish some of these standardizations. If you have a centrally activated and funded resource, then obviously sending benchmarking samples out to include a priori in your assays is what needs to be done and is done, certainly in the Kidney Precision Medicine Project. For every assay that we have a benchmarking sample, which is included together with the next batch of the human procured samples in this setting. But that requires a systematic, well-funded a priori effort. And with the other approach, you might be able to adjust a lot of experimental confounding and still find meaningful comparisons between your data sets.
So that's great. I have another question to Dr. Kressler. And there are a lot of data now available in the public database, a variety of different public database for the omics, the RNA-seq and the proteomics. But the majority of the data is from primary screening. So how do you kind of access those data? Because we know that the primary screening data, there are risks. The data is not confirmed. The change of the gene may not be confirmed. And from the informatics point of view, how do you approach it to this question?
That gets back to, I think, what Matthew mentioned earlier, that ML approaches will be very good if you have at least partial orthogonal data sets, meaning if you can take your experimental data, and we saw that also earlier in the morning session, and you can project it in a data space, for example, organoid versus in vivo disease data, or organoid versus young versus aging data sets who can serve you as a trajectory towards disease, and see to what extent you can detect similar patterns in those settings if you have only single snapshot in the data space available. And right now, this works very well if you have, like we discussed earlier, you have data sets who are reasonably standardized in their quantification approaches. But with the rapidly evolving ontological frameworks to map between these data sets, you could even project transcriptomic data into the proteome space, I think, where we are reasonably well. You can project pathway predicted transcriptional data into the phosphoproteomic space. And I think the process to do similar activities with metabolic spaces are getting very quickly to a place that at least one can do some basic sanity check and build out what I refer to as, multi-level evidence before you start to invest your, limited vet bench resources based on your in silico modeling. If you can identify a signal with two orthogonal approaches in two different cohort studies or experimental contexts, and that might raise up to a level that you start to try to validate those findings experimentally.
Thank you. I think one of the points is very important. I learned from you talked. So cross-check different data set for the same gene change, maybe provide some deep information that gene may indeed change in that particular disease or particular status. Thank you. So our next question to all speakers and in this session is how should the omic data be generated from rare disease iPS cell-derived cellular models, including 2D and organoids 3D models? So from the informatics point of view, from data analysis point of view, how do you view biologists should generate the data? So including the sample size and the disease relevant tissues in the genetic diseases, what kind of mutation, and that kind of the question to all the speakers.
I mean, I will highlight how it should be collected, and it's not always a trivial thing to do. When you have multiple data types, one of the key things is to make it efficient to track if the same sample is used. Then it's a little bit easier to track, but sometimes you have related samples. That is not easy to track yet. Actually, even the first one is hard to track. And so if you did sequencing, you did exome analysis, you did metabolomics, you did proteomics. It's a little bit easier to track it when you have the same sample. But then the question is, okay, are you going to house it all in one database internally? Do you use the standards for each of these communities? So do you use proteomic standards for the proteomics data? Do you use metabolomic standards for the metabolomics? Do you use exomes standards for the exome? Or do you create your own within your consortium? And there are strengths and weaknesses with either way that you're going to try to do. I don't know what my advice would be. I think having all the data in one place has its advantages. Having all the data not in dedicated repositories has also some weaknesses, because you won't be able to leverage the information in the dedicated repository. So if you don't put it into a proteomic repository, well, you can't take advantage of the proteomic tools. But linking becomes harder when you put it in a proteomics repository in your own consortium. So for me, if you're going to create a consortium, in this case, a lot of rare diseases are going to be studied. Study as many as you can with the same method, because then you can make comparison. It makes it easy to compare and contrast. That would be my advice.
Yeah, actually, to that point, having some centers or labs designated sort of like centers of excellence for individual modalities, so that you control some of the confounders more easily within, specific, data collection types. That could help a lot in this space, and it's why, without something like that, it will be really important to make sure that a lot of the downstream analyses are included. Because otherwise, it will be hard to get these apples to apples comparisons on, the raw data sets, especially without really good metadata.
Mm-hmm.
Thank you. Any other comments? Oh, go ahead.
Yeah, I'm going to jump in and say a comment. I think it was brought up earlier today. So if you're studying a disease with a founder mutation, such as cystic fibrosis, you do have the benefit of asking this question of how much heterogeneity do we have from saying where it was collected, so the sample site that was collected, how it was processed, and also the effects of genetic background also that's actually, contributing to heterogeneity. So I think with diseases with the benefits such as cystic fibrosis, such as a few other diseases with a founder mutation, you can actually ask some of these controlled experiments so that, it acts as internal control and then, it will strengthen the results in terms of that. So, just thinking about the genetics of it also, because a lot of these rare diseases, there's a monogenic underlying thing, we can ask that question. But as Matt said, that if you can standardize by just having one, say, center of excellence that collects it and things like that. But the problem with these monopolies, I won't name any of these centers of excellence, is they're not so great at sharing, even though it is in the NIH mandate to share. They share when it's convenient to them. I won't name any.
I think that is changing. And that's where, efforts like Human Cell Atlas, HubMap, and KPMP, who practice and live open science, open communities, are really changing the landscape. Because it's to everybody's benefit, even to the most ultra-competitive scientists, to adhere to the data generation and data sharing standards. Because then your data are linkable and you gain the insight you need. And yes, you might then, if you have the next step change with the next latest technology, you might not share that until you have your first paper out. But when we are talking of real impact for real people, that's where, 5-10 years ago, being a team scientist with systems biology, you were out there. I think a lot of the community has moved squarely into that space to expect it now. And that's why these efforts and why funding agencies incentivizing the initial establishment of a knowledge base with standards is so important. As that knowledge base with standard is present, like with the human genome, now that's not a discussion topic any longer, how you submit genetic information. Yeah? And we are moving in different spaces into the same area. So building out that critical mass where data sharing is to everyone's advantage, even to an ultra-competitive investigator, I think is one of the ways a funding agency can help us to make that step change.
Yeah, and just to emphasize, I agree. We don't want to create, a silos of excellence model. And also, I also want to say that, yes, I think, there have been great strides recently, and ensuring from the beginning that there's good data sharing built in, and that is definitely starting to happen, and more of that would be absolutely essential here.
So thank you for bringing up the data sharing, or maybe not always as shared as you've alluded to, and the question is why that is, and especially in the context here of i3D, do you see any danger in terms of privacy once the data gets linked, with other clinical data and things like that? Some of these are, technical, maybe competitive, but are there any, I guess, ethical issues that you would foresee specifically in this space here?
And one thing I really want to start off first, that it's so compelling, the stories Moncon and Matthew told us. If you have a patient or, the parent of a patient in a steering committee, that levels the playing field immediately. Nobody can argue, I have to hold back my information first before I get that paper out, when you know that the son of somebody on your committee is dying. Yeah? And that is really, really critical to ground our scientists. And we do that across all of, in our rare disease network, we were funded by patients. In KPMP, we have patient representation on our steering committee with decisive vote and guidance. And making that a standard approach to how we do science will help our scientists to be grounded and to prevent, to some extent, those activities. So this is just where I really want to thank Moncol and Matthew to bring that aspect to our discussion, which I hope we will continue to see throughout our effort in the rare disease space. And then one aspect, we do both common and rare diseases. What I always find very striking in the rare disease community, that privacy concerns are never listed amongst the five. I more often than not say, please use a picture. I showed pictures from patients. They tell us, when I asked them, can I do that? Absolutely, please do. If it helps the cause, move forward totally. And share everything you have from us if it can help somebody else. And a lot of the privacy concern with common diseases, where understandably there's a different dynamic in place, doesn't apply here. Yes, but we have to be careful and finding ways to have data, some of them gated, some of them open, when it gets to a space that they could be exploited against the interest of patient participants is critical, and it's a non-trivial exercise.
Yeah, Matthias, you bring some really good points up. So including patients on advisory committees is one way to help push for data sharing, because you're absolutely right. Patients want their data shared by and large. The irony, of course, is that a lot of these silos of excellence, I won't name names, used protecting patients as the rationale for building the silo, for building the walls and not sharing the data. Whereas if you'd asked any of the patients, literally any of the patients contributing their data to it, they would have said, no, please share it as broadly as you can. So if you have patients involved from the beginning, they'll help remedy the situation by saying, when you get consent, make sure you ask up front, can we share this as broadly as possible? And they'll say yes. And it'd be very rare to find the patient that doesn't want their data shared as broadly as possible. And then that whole rationale of, well, we have to keep it siloed goes away because the patients have said from day one, no, share this as broadly as you can.
I just want to echo that. So my cells featured in a Nature publication where they use the CRISPR approach to edit my mutation. And do I care that people know it's my mutation? I don't really care. I think most patients want more people, especially if they have an ultra rare disease, to work on their disease. You know what I mean? It's hard, because everyone wants to work on the diabetes, the congenital heart disease and things like that. When they read my abstract, oh, this affects 1 in a million, it gets chucked in the bin already, going, this is not going to make impact. And this is what rare disease patients are actually trying to fight against. That I just want to double down on what Matt said. A lot of people are using that as an excuse for not data sharing, not sharing the patient's cells. And when you tell the patients, they're absolutely disgusted to hear that at patient conferences and things like that. And I think one of the things that will advance this field is if we can sort of like get around, have ways that this data can be shared, including the cell models themselves for drug discovery.
And the exciting piece is, you have all your medical history in here. And that has changed again over the last year and a half, the landscape. There are now tools out there how patients via what in the US is the law, that we have the Data Portability Act, that every healthcare system has to give patients access to their healthcare data in a portable, standardized format. And patients with a simple app now can share their personal healthcare information with whomever they want, even if they have interacted with multiple healthcare systems. And suddenly, the corporate offices at larger healthcare center who were envisioning to use that to monetize the knowledge shortcutted that the patient is in charge and not the care provider who helped to generate that healthcare information, but it's the patient. And these are ways for even to circumvent some of the electronic healthcare record challenges when in multi-center networks we try to harmonize EHR-derived data sets, which might actually through the vector allows us to address those as well. So again, technology will help us, and seeing patient communities understanding the power they have will accelerate that first.
Thank you. We're going to switch gears a little bit now and focus a bit more on the omics. So what's the current state of integrating omics data and potentially also with other functional readouts for translational research? And what additional data needs to be generated to fill existing gaps?
I mean, I'm not, maybe the question should be addressed in a different way. So, because we have the capacity of generating large amount of multi-omics, a whole bunch of variety of different ways. It's the integration, I think, that is hard still. So, because today I may collect metabolomics. Okay, 5 years from now, somebody may do transcriptomics on that sample. 6 years from now, somebody may do metagenomics. And so keeping track of this and making sure that the data tables as the data is being processed, because algorithms change over time, that every iteration of data processing gets uploaded and managed and be available to the community. So having an infrastructure or a way to keep track of all this, what is being done to each of the samples, and then sometimes, okay, maybe it's not the exact same sample, but it was collected the same day, and it's now version 2 of a sample cohort. Keeping track of that over time, so that when somebody wants to do analysis, that they can almost on a click of the button pull it all down. Right now, creating the data table, the processing, learning which sample or which data belongs to what, is where the majority of time a data scientist will spend their time. That's where we can make a huge inroad. If we do this effective enough, I can see people off the street being able to do analysis. The data science is really the managing and finding all the information and connecting the metadata and processing. Do I have the latest processing data table? Those are some of the challenges that I think being able to keep track of that could really enhance how we do multi-omics.
Being very good steward of the sample. This is a real art to identify with a tissue repository sample bank when to invest your samples or what technology, and so that you are future compatible. It's fascinating to see that the tissue blocks we stored away 22 years ago suddenly are becoming useful in a context nobody would have imagined 2 decades. And the other piece is what is critical, remains critical, always will be critical as a phenotype. What is the lived experience of a study participant? And you never can be deep enough there. And we are seeing right now already that the insight we gain in tissue has exploded to an extent that it will start to become really comprehensive. But what is a phenotypic presentation of a patient? When is a disease causing a disease burden that we need to intervene? How much need we to push down a disease activity that the patient does not experience that disease burden for some of these common pathways we saw popping up all along all day? Those are features which are non-trivial to capture. They require patient interactions. Again, these things might become very helpful in capturing some of the phenotypic information using variables. And again, rare disease patients will lead the charge there because they will give scientists access to a lot of their information and their experiences to take us forward. And then being intelligent, what readouts do we need from patients so that we can generate similar functional readouts from the organoids so that we can do that functional mapping between the ex vivo and the in vivo state in a meaningful manner to empower our ex vivo screening capabilities to find the key switches we need to plan in humans.
I'm going to jump in and talk about in the gene discovery space. One of our challenges is, I know it sounds, hard to believe, but it's accessibility to the relevant tissues. So blood is very accessible, saliva is very accessible, other tissues are not as accessible. And when you're studying ultra rare diseases, how can you get the numbers of it? So so one thing that would help with the data integration process is to say, of things that are accessible, say blood, what things are correlated with the affected tissue also, as a way of actually increasing sample numbers, and what's also correlated to IPS models that we can then use that to generate, things from, say, patient fibroblasts, which are a little bit more accessible, and that way we can hit that gap of the affected tissues being accessible. Because it's impacting our ability to, say, integrate genome data with RNA-seq data to find the causal gene, because we're getting so good at diagnosing things through just exome sequencing alone. Not many people want to give biopsies, and now we have this issue of not having enough tissues from our ultra-rare disease patients. But, what I want to emphasize is, can we get the same readout from blood or something else that, and where we can't know what the difference is also for some of the genes and pathways of interest.
Thank you. And to dig a little bit more actually into the models, to take one of the questions for the audience, and I think this will be our last one. There's system biology models, right, where one can take into account flux balance, so these metabolic models, and one could also imagine bringing in the influences from the environment and exposures, et cetera, to inform panomic models in general. So what types of models right now do you think are useful, and why? Maybe commenting on feasibility as well.
Sounds like a very open question.
Yeah.
And did you mean like systems biological models, or are you talking about, specific tissue models, or?
I mean, the question is pretty general. I would say that, I would say there's maybe disease-specific models, tissue-specific model.
I guess from a very high level, I can say that if you want to get into specific tissues and you look at sort of a census of phenotypes across rare disease, neuronal models, brain organoid models are going to, cover somewhere between 40 to 50% of the community. So if there's a specific model area we want to have good coverage of, good platforms for, I'd argue for starting there, just given the distribution of phenotypes in rare.
Then if you're talking about, dynamic models, again, it depends very much what dynamic aspect you want to evaluate and what is your prior knowledge and is available for your algorithms to work against. And that's where, depending on the different in vivo and ex vivo dynamics you have available, you can build them out and bring that knowledge back to the experimenter as well. Obviously, metabolites are the prime example where really complex flux studies often are critical to, interpret a single time point of a measurements to see where you are in the overall flux cascade. You are interested to mechanistically potentially intervene. One aspect gets back to, again, advice to experimentalists. It is exceptionally helpful to get time sequences on some of these points. And they are sometimes challenging, these tissue procurement efforts. But in instances when you particularly have matched pair samples, your power goes up exponentially. And in our field, we have studies with 8 or 10 patients where we have paired examples before and after exposure to an intervention who show significant impact in us understanding what the intervention actually is altering, as long as we have a large background knowledge of cross-sectional data to map the basic states against. So this is again an opportunity, depending on how you set up your experimental design, having those small nested mechanistic studies embedded in your interventional trials, which I think we will discuss in a moment, and really generate significant input even with minimum sample size.
I'm going to jump in and comment on where, I see it useful. If we're talking about, say, a metabolic flux model, I think one of the difficult questions we ask in translational work is how much of this enzyme do we actually need to deliver? Because sometimes delivery to a tissue is difficult in itself. So it's like, how much do we need to deliver to have therapeutic benefits? So if it's a recessive disease, we already know that we can get down to 50% levels and still be okay. So maybe these models can be built in such a way to help address that question or better inform that question, which is quite difficult. Like, can we go at enzyme levels of 20%, 10%? Where is it that that will have therapeutic benefits? So that these models can maybe be adapted to help ask that question, so that yes, we could experimentally determine. At least it will inform also on that.
Thank you. So I think we're slightly over time, so we can close that session. Thank you to our panelists, and a reminder also that there are some questions in the Q&A if they'd like to go through and answer some of them. So I think I'll turn it over to Liz or Mark for the break.
Yeah, I think, Abby, sorry, this is Liz. I think we can just, the break is until 2:40 and we'll come back for the last session, which is on therapeutic approaches to rare diseases. So using these non-animal models. So we look forward to seeing everybody in 6 minutes. Thank you.
Okay, it's 2:42, so we should start with session 3, which is therapeutic approaches in rare diseases. And in this session, we will have, as previously, 4 speakers who will have 20 minutes to give their talks, and we will keep the questions for the end of the session. And our first speaker is going to be Professor James Hickman from University of Florida in Hesperus, and the title of his talk is Human on a Chip System as Efficacy Models of Rare Diseases. Thank you very much, Dr. Hickman, for being here for us. So take it away, please.
Can you see my screen? All right. So I'm going to talk today about efficacy in rare diseases, mostly through work that we've done at Hesperus. Hesperus is a company Mike Schuller and I started about 8 years ago. We've done a lot of work with SBIR grants. Some of it was from NCATS. We've got a 14,000 square foot facility down here in Orlando. Company's got about 48 people at present. It's a CRO only. And I'm going to talk about work that we're doing in actually filing INDs utilizing these microphysiological systems that we're creating for rare diseases. And the first one that we did was with Sanofi back in 2020, which was authorized for rare disease in 2021. So what we really focus on is this idea of clinically relevant functional readouts. So what does that mean? You go into a doctor's office, he doesn't immediately stick a needle in your arm to be able to tell you out fluids, look at biomarkers, right? He looks, how are you walking? How are you talking? Are you making sense? He listens to your heart. He checks your reflexes. What he's checking for is things like muscle contraction, electrical activity in neurons and cardiac cells, neuromuscular junction formation. He may ask you about elimination and other factors. He's basically asking how your GI tract barrier integrity is. So this allows us to look at non-invasively for acute, but more importantly for chronic monitoring of human-on-a-chip systems, up to 28 days that we've published. So what we can do, we can actually put neurons over microelectrode arrays and get their electrical activity, pattern cardiomyocytes over microelectrodes to be able to get their electrical activity and QT interval. We can put cardiac and skeletal muscle on thin pieces of silicon, so when they contract, they deflect a beam, which we can actually then sense and turn into force. We can still look at biomarkers. We can also look at TIR to be able to look at the resistance for barrier integrity. So how it works is we can do single organs, or what we're really known for is multi-organ systems, where we can actually culture out different cells on each chip separately under its own conditions, and then assemble them all to the same base, put a fluidic channel over top of it, and then that defines a fluidic channel. We use a pumpless system. We just use gravity to pump it back and forth. And everything I'm going to show you is all in serum-free media. We can then put a clear top on top, so we can actually then visualize the cells. At the same time, we can actually monitor functional measurements through electrical connections here, through the reflection of the laser through the bottom. The first system I'm going to talk about is our neuromuscular junction system. Basically, what we do is take human motor neurons, grow axons through the tunnels to be able to innervate skeletal muscle. We can put the myotubes on cantilevers, so we can motion capture here. We can then stimulate with the motor neurons with electrical activity. Just basically showing the axons going through the tunnels, making nice neuromuscular junctions. And again, getting to this idea of clinically relevant functional readouts. So I talked to somebody who was running an ALS clinical trial up in Boston, Brian Weniger. And I said, well, what are some of the tasks you have somebody do during a clinical trial? He says, one thing we have them do is something very faster and faster with their hands. He said, well, we can reproduce that. We can actually then stimulate the motor neurons faster and faster and drive it into tetanus. And what happens is, for an ALS patient, gradually they'll get spasticity, which we akin to skipping here, and they'll get loss of tetanus, basically loss of grip strength. It's a very busy figure, but just basically showing that we can actually, when we're stimulating, we get nice fidelity for the wild type. But for two mutations of SOD1, we can clearly see skipping, partial loss of tetanus, one mutation, total loss of tetanus with another mutation. And for FUS, we see a total loss of tetanus and some skipping. And then in this particular case, we had wild type on the other side. So, and that was all, the fidelity was fine, showing that we're actually getting deficits at the neuromuscular junction. And again, so these are the statistics for that. We also can measure fatigue index. Again, this is an equation directly out of a clinical trial, where we're actually looking at the ability to maintain tetanus in the system, and basically showing it goes up, okay, in the disease models. And actually, we actually looked at a holistic treatment for ALS called the Diana protocol, and showed we could actually restore function for fidelity. But even more, so we could actually restore function for the fatigue index, or basically reestablish grip strength for these systems. And this was published in Advanced Therapeutics. We've now been able to do the muscle from different mutations, ALS mutations as well. So we did a round robin of wild type, skeletal SOD1, SOD1, wild type, and an SOD1, and basically two different mutations, basically showing that the morphology of the cells is fine. This was published in Biomaterials. And I'm going to actually show you just about everything that I'm showing you is all going to be published material, because that's one way of validating the systems, that these aren't just very nice PowerPoint slides, that we actually have validation for these systems. And basically showing that the deficits in the motor neuron caused the deficits that I showed you already, but actually in the muscle, you actually had a larger deficit with one mutation, but not as much with the second mutation. But there was a recovery over a period of time, okay, in the system. And we can also then take these systems where we're looking at evaluating pegcetacoplan, a molecule produced by Apellis for ALS, and again, be able to look at a bipartite system, what I showed you before, but also adding an immune component, as well as microglia on the other side and Schwann cells. And basically showing the difference between an unactivated immune component, showing that their increasing dosage of their molecule caused a recovery, and that the you clearly see that there is a on the activated monocytes, you see a clear drop in activity, but a recovery at time. And you could also see the fatigue index was not really affected as much with the unactivated monocytes. But with the activated monocytes, you saw a recovery with the highest dosage of their compound. What's even more interesting, the neurofilament light quantification was enabled in the system because this is now being used as a biomarker for ALS by Biogen for their clinical trial, and basically show we get a decrease with their this. And this actually was used for an IND, but also for an orphan drug designation, which was authorized. We can use the NMJ system as well for looking at myasthenia gravis, which is another rare disease. What happens here is your body raises antibodies to the acetylcholine receptors in the neuromuscular junction, which either cause blocking, causes internalization, or causes complement deposition. What we're able to do is show a dose response curve, again, for the antibody, okay, for a biologic in the system. So we're not used, we don't have to look at just small molecules. And again, basically saying that with an antibody that didn't target the acetylcholine receptors, we don't see any changes. We see that it actually will affect muscle contraction at very, very high doses. And we wanted to really show is that the difference between the blocking and the internalization. So without the antibody, you see about the same amount of receptor on both. And then when we actually then treat it with the antibody at a lower concentration, you saw the loss of the receptor on the outside, but maintained inside. At very high concentrations, you see a downregulation. And this was published in Frontiers in Cell Development Biology. And this is now being utilized for, we're just finalizing an IND with a company for their molecule for myasthenia gravis. We have two other projects ongoing with two other companies for myasthenia gravis. You heard earlier about NGAL1 disease. We had partnered with NCATS. They'd asked us to look at this in terms of a functional model, where we took the stem cells from the wild type, from the knockout, and 2 patient samples, and be able to show we can actually get the expression of the markers for motor neurons. So we can actually differentiate out the motor neurons in the system. We're able again show that there was an aberrant glycosylation of the sodium potassium chloride cotransporter, okay, with the knockout and the 2, so we could actually see a biomarker. But again, what we want to do is see a functional deficit. And what we saw here is that for patch clamp electrophysiology, we did see a deficits in induced current. Okay, spontaneous action potential is a very large decrease. Some aspects in resting membrane potential and amplitude as well with patch clamp. And again, in the NMJ system, again, showing for there was deficits in NMJ number in the system, okay, and the failure rate was higher, and also showed that there was fidelity with one of the mutations, loss, okay, at 14 days, and then it was more for the other mutations for all 3 here. And then you also see that there is a loss in tetanus in the system as well. And this was published in Advanced Therapeutics. So again, you have different choices. You can use organoids, you can use functional systems, but together these can actually start looking at providing evidence that the FDA has shown that they will accept, okay, for rare diseases. We had published some of the first myelination models back a couple of years ago. This is in ACS Chemical Neuroscience, again, with human systems. Everything I'm showing you is all human data. And a company had seen that and said, well, could you help us with 2 diseases in terms of neuropathies for 2 rare diseases, multifocal motor neuropathy and CIDP. What happens is your body raises antibodies to detect the node of Ranvier, which basically allows long conduction over long distances in your axons. Basically, you see here clinically, you see a loss of the ENG signal with time, because what happens is the complement cascade gets activated and attacks the node of Ranvier. And we had shown now this is with patient sera samples. We can use both mutations, but also patient sera samples. We saw complement deposition. We showed this was abolished with the company's molecule, which was True North, was acquired by BioVerica, was acquired by Sanofi. And we took the NMJ system, turned it 90 degrees, and now ran the axons over the electrodes in the tunnels, so we can actually then follow the conduction velocity over the electrodes. And this was a key experiment that we did. We took untreated, then we took serum plus the isotype control, plus the serum plus their molecule, and basically get a range of conduction velocities. Again, the untreated, you see the range of conduction velocities, It was abolished, again, with the serum, the isotype control, but restored, okay, with their molecule. This was the only efficacy data that was included in an IND that was enabled a phase 2 clinical trial, okay? And this was published with Sanofi last year. Okay, so again, another way we can actually utilize clinically relevant functional readouts, because we're doing all this without cell death. We're not using death markers to be able to determine whether or not, you know, there's a deficit. We're looking at things that people would try to observe from patients, okay, during a clinical trial or in trying to understand their disease. We also are working with CNS models. This is actually a cartoon I made about 20 years ago. Basically, what I was hoping to do is long-term potentiation, because that's a fundamental unit of learning and memory. So we could look at information processing diseases. And so we can actually look at hippocampal neurons innervating cortical neurons. So what we want to try and do is to pattern cortical neurons over top of microelectrodes. And again, using the same patterning technology, what we can do is we can stimulate on one electrode and then record on the other electrode, okay, which is what people do doing dual patch collective physiology. And you can show the axon going between the two electrodes. And again, you see baseline activity in the system. Now, it's very, very important in trying to look at these systems, because with MEAs, it's very easy to make noise look like neuronal signals. So what you have to do is treat it with lidocaine afterwards to prove this is actually electrical activity. And this was published in Stem Cell Reports. And again, we actually were differentiating out cortical neurons from human stem cells for that one, showing at 21 days we could not maintain LTP. We could maintain it after 35 days, so we're getting mature cortical neurons. But if we actually we have at 21 days, a combination of inhibitory excitatory neurons. And if we block the inhibitory neurons with picrotoxin, we can induce LTP. Okay. And again, just confirming that we were doing LTP, you can clearly see here, then we blocked the AMPA receptor, which is involved in synaptic activity. We lost the ability to do LTP. And actually then not looking at blocking the NMDA receptor as well, we lost the ability to do LTP. We're able to use this to be able to look at for an Alzheimer's disease, which is not a rare disease, but I'll get to the reason why, where we can actually then for the control, we can actually then not see the loss, maintain LTP, but have it lost with both A beta and tau oligomers. And this is published in Alzheimer's and dementia. But then we actually then use looking at A beta scrambled and A beta 42 with patch clamp, and then an approved AD drug, donepezil, and showed that we can actually get loss of activity, but then restoration, and so we get a one-to-one correspondence with the MEA data, okay, for the oligomers and restoration of function. And we've now done that with 4 drugs that are being used to treat AD, donepezil, ceritinib, memantine, and roloperam. And where it actually applies for rare diseases, we're now applying this for frontal temporal dementia, okay, which is a rare disease where there is basically not even any treatments available at this point. But we believe this shows we can develop an assay for it, for that. So that kind of went through a number of the efficacy models we have, but to show you we can also look at the both efficacy and off-target tox in the same system. Michael Schuler had built a multi-organ system by using the equations for PBK modeling to design the first multi-organ system back in 1995, was published. We're now using this as a multi-organ system, look at efficacy and off-target tox for the HEAL initiative for opioid overdose and recovery with naloxone. And again, we go back, we use this pumpless system. Okay, we get rid of all the tubing, get rid of all the pumps, but most importantly, make it a low volume system. So we can create PK/PD models of these systems where we can actually look at the absorption of the molecule into the housing materials, which happens with all housing materials, look at metabolism in the system, and then be able to get the PK for that, and also be able to look at the cardiac pharmacodynamics from the outside of the cell, and then for the inside of the cell for a terafenadine, fexofenadine couple for both parent and metabolite. We're able to use that data with AstraZeneca to be able to predict in vivo data for guinea pig, dog, and non-human primate. That's really important because what we can do is we can take these in vitro models, determine the data, and then be able to correlate it to in vivo to be able to start looking at first dosing okay, for a rare disease. Again, for repurposing of drugs and for different applications. This was published in Nature Scientific Reports. We've been able to create a innate immune system in one of these models, where we can actually then get the cells to be non-active in the system until we activate them selectively. In this case, we're able to show both the inflammatory phenotype of the M1, but also the restorative phenotype of the M2. This was published in Advanced Science with Roche. And then lastly, Mike was one of the, did a lot of the earlier barrier tissues. We work with L'Oreal early on too. We have a skin model and a multi-organ system. We can look at kidney as a tox target. We can also add in human blood-brain barrier and GI tract systems as well. And again, Mike had shown that there's really good correlation, and we can actually have a model. I don't have time to go through it right now. We have GI tract, BBB, CNS, where we're looking at transport for oral dosing versus IV dosing versus intrathecal. And so with that, I'd like to thank for being invited and be able to share our systems with you, and look forward to the discussion.
Thank you so much, Dr. Hickman. Next, we're going to introduce Dr. Jason Eckert. If you could please share your slides. And Dr. Eckert's going to give us a talk titled Utilization of Translational In Vitro Models in Drug Discovery for Rare Diseases.
Great. Can you see my, yeah, you can see my full slides?
I don't see them yet.
Working just a second ago. Let's give it a sec. Raise it up. Yeah, so this should hopefully, you see it now?
Yes, it's not in presenter mode yet, but your PowerPoint is up. There we go.
Okay.
Take it away.
Great, thank you. Yeah, thank you for the organisers for coming today to talk about the utilisation of translational models in drug discovery for rare diseases. So yeah, I'm going to touch on a number of different models that we've been working on. But before going into the different models, I thought it would be good to give an overview of some of the opportunities and challenges in using translational models. Also, how accessing stem cells and patient cells as well plays into how we can utilize rare models in drug discovery. So how we can go about improving the clinical success and also reducing clinical attrition. So I see this is a common theme in complex in vitro models, but also in rare diseases is initially we're having the right target. So can we have the right target engagement and pathway modulation? So it's key to have the right cells that we can then engage the target. Also the right PK/PD. So Jim mentioned about some of the models, more complex models and multi-organ models, that these are important when looking at PK/PD. Also the right patient population. So getting access to the right cells, and also especially in the rare disease space, different mutational statuses of the disease. And then looking at the therapeutic response also, so understanding if there's any safety concerns as well compared to your therapeutic response. Some of the challenges that I see, so from a scientific point of view, sometimes there's a lack of established in vitro and in vivo animal models. And then the ability to determine translational relevance with limited pathobiological understanding of rare diseases. So sometimes it's not always clear what's the main driver of the rare disease. And then balancing the complexity and translational relevance and when building the models. Then from an operational viewpoint, and I've heard there's been a few comments already today about accessing disease cells, getting access also to primary or stem cells. One of the challenges in early drug discovery is the length of time in developing the validating, qualifying the models can be, that can also really impact the using them as a decision-making tool. And then also, this is also the length of time where they're using them for a field service or research collaboration versus in-house implementation. So the opportunities I'm going to discuss today is the first one is going to be the mechanistic one in more in late development from using a model, and then the second example will be in early discovery. So then this will also touch on some of the challenges using models in the earlier discovery phase. So you heard a little bit from Jim about neurological disorders. So I'm actually going to touch on two of the areas that we've been working on at UCB. One is in the myasthenia gravis space, so where there's a antibody response, so acetylcholine receptor or created autoantibodies, and I'll go into a little bit more detail about that. And then the second one is in the MMN or GBS, where there's the effect of autoantibodies at the node of Ranvier. So for looking at the, for myasthenia gravis at the neuromuscular junction, you can see here on the left the normal healthy state of a neuromuscular model junction, where you don't have any blocking of the receptors, but in the muscle weakness in a disease patient for myasthenia gravis is blocking of the acetylcholine receptors, a number of different mechanisms. Either there can be internalization of the receptor, blocking of the receptor. Also, the MAC can impact the acetylcholine receptor. So in the middle here is where using cellular, which is a inhibitor for C5, and we can use that to allow for recovery of the acetylcholine receptor signaling. So we hopefully these work for the. Yep. So we created a neuromuscular junction model. This is where we had human iPSC-derived motor neurons, and then we used including muscles. We had these where we could then cause the release of the acetylcholine receptor as they'll activate from glutamate stimulation. And then we're looking at neurotransmission, looking at the calcium flux. So you can see, for example, here the controls, we're getting nice the neurotransmission, but then using the alpha bungletoxin, we're not getting any neurotransmission or calcium showing as calcium flux. So go to the next slide. So then the development of the neuromuscular model, we worked with a NANDA device to create the model. You can see here on the left figure is that we have the neurons, and then they're extending out into where the muscle fibers are. And then we get really nice extension of the axons within the muscle fiber. And so there was a number of optimization studies that were used for creating the co-culture system. And then using as I mentioned, the calcium flux as a readout. We weren't looking, there's another number of other ways of understanding functionality, but we use this as it was a fairly consistent way. So we here on the left, we're looking at controls, volunteers, or we had 5 different patients. So from MG patient sera, so we're adding the patient sera, and then we were coming in with zilucoplan so we could block the patient sera. And so you can see that there's without any zilucoplan, you see a reduction in the calcium transmission. But then when we add zilucoplan, it comes back to baseline. On the right here, you can see there's MAC deposition, which is C5b9, which when you add zolpidem, you don't see that. And then we also were looking at split products. This was induced by the MG patient serum. And when we add on the left here, C5a or soluble C5b9, where we have a similar response. So you see increases in the split products. But then when we add zolpidem, we inhibit those split products. We also have extended this work. We're looking at, so that was where we were adding zolpidem at the same time as the patient serum. We also have looked at where we wait either 24 or 48 hours after the treating. Also, we add the patient serum and then we treat either 24 or 48 hours. And then you can see how if we treat for 48 hours, we can inhibit any further neural damage, but then at 48 hours we continue to get the neuromuscular transmission damage. So this is to mimic what we see in a patient setting. So now moving to another model. So this was where this is in early discovery, where we're looking to leverage in vitro peripheral neuropathy models. And this is looking at MMN or GBS, which is they're both fairly rare diseases. And so the effect is where you have these autoantibodies, either anti-GM1 for MMN or GD1B for the GBS. And then we were looking to see effects of these autoantibodies on the functional impact on the neurons. And some of the readouts we were looking at was neuronal or skeletal damage, conduction block, demyelination, and phagocytosis. And this is a comment here where some of the different potential mechanisms of how there's peripheral neuropathy occurs is still under investigation. So we were looking at some of these different readouts to better understand what would happen to the neurons. So here we were using motor neurons derived from, and then we were either adding anti-GM1 antibody to mimic MMN, or then we were coming in with a inhibitor, upstream C1, C4 inhibitor, zolpidem, or an anti-C1q. And what you can see is we can block C3b deposition for C1q and also the C14 inhibitor, but not the zolpidem. And then over on the right, we were able to quantify this. So looking at the amount of C3b and also cell surface map. We also wanted to see, was there any functional effects on the neurons? And so on the left here, we're looking at neurite degeneration, and the blue is the DMSO control. So you see a decrease in neurite length, while all the inhibitors for complement have stopped the effect of neurite degeneration. We also were looking at neurofilament light chain, which is also a way of understanding the effect on neurites or neurons. And where we were adding DMSO, you see an increase in neurofilament light, but a reduction in when we're adding the different complement inhibitors. So we had that information, and we wanted to see if we could then look at more functional effects. And what we have ran into some really challenges in being able to have higher throughput models for myelination of motor neurons. Dr. Jim Hickman mentioned about some of the myelinated models that he worked with. We were trying to recreate these in-house, and what we found into, and we ran into some of the challenges, is from a discovery point of view, is some of the cultures are really long time to take. There was some world-to-world variability, and really we had a real challenge in the showing evidence of myelination of the nodes of Grandia. So what we did is we had to then move to using a model of where we were taking actually dissected T's nerve fibers. So a scientist in the group just came up with this idea, and it actually worked really well, where we were doing ex vivo models. So sometimes where we have to step back and use more translational models using in vivo tissue. So here, where I want to call out is we were able to show so that Casper is a marker for where the node of Ranvier is. We saw where for using anti-GT1B, we get the deposition of C3B, also C5B9. While when we added the inhibitors, we see a reduction in the diffusion effect of when adding the GT1B. So we're currently looking to investigate this more and using machine learning to be able to quantitate the effects of the node of Ranvier structure. So some of the lessons learned and conclusions. So this, we found really sometimes the challenges is getting limited tools, reagents. So for example, getting monoclonal antibodies. We also run into issues of getting patient sera. So for example, it's been quite challenging to, for example, in the MMN or GBS to get access to the patient sera. We found that, so sometimes with rare disease, it's not always well understood and also from the clinical results, it can sometimes be misleading. We're trying to then elucidate the pathophysiology and also pathways. We also found limited reference tool compounds. So compared to more well-established diseases, there's so you have less reference patient samples. So then to be able to validate the model. And then, as I mentioned about the long cell culture times and also differentiating iPSCs and creating these models is sometimes at odds with wanting to make quick decisions in this early discovery. And then from the conclusions from the two studies that I presented today, so we were able to, which was fantastic, we were able to provide mechanistic rationale for the clinical response for the acetylcholine receptor positive and myasthenia gravis patients where they're treated with the C5 inhibitors. And then secondly, for the peripheral neuropathy studies, the data showed evidence of blocking complement would inhibit human motor neuron derived from iPSC damage. And we also showed some evidence of the preservation of the node of Ranvier structures in rat-sided nerves. So I'd just like to acknowledge that this obviously wouldn't happen without dedicated scientists in the lab. So with that, I'm finished and look forward to the discussion and roundtable discussions later.
Thank you so much, Dr. Eckert. So we will move to our third talk, and this will be a talk given by Professor Clive Svetson from Cedars-Sinai. And the title of the talk is Using iPSCs and organ chips to develop a gene therapy clinical for MCT8 deficiencies in children. Thank you very much, Dr. Svetson, for being here today with us.
Hey, thanks very much. Can you see my screen okay? Can you see my screen? Okay, good. I broadened the talk title out a little bit, but I'm going to finish with MCT8 and the modeling that we've been doing. And how we're taking that to the clinic. But I thought I'd start with more of a high-level discussion of some of the other work we've done. Just my disclosures, Cedars-Sinai has a stock interest in Emulate. I took out the goals of this meeting and summarized them here. And what I'd like to start with is this part and the importance of recording, having the right kind of cells to do these studies that we just heard some beautiful work from previously in diseases and in diseases where you've actually got the patient tissue rather than having to add drugs or toxins. And that's really been the focus of a lot of the work we've done over the years. Of course, you've seen the concepts and we've discussed a lot at this meeting, and I think we shouldn't lose the value of two-dimensional culture technology as it's very high throughput. And as we go down this scale here, down towards the chimeric animals, which is maybe the ultimate model, we get to more and more complexity, but and also increase the physiological relevance. But as we just heard from Jason, the downside is the more complex we make these models, the more unreliable they are in some senses and more difficult to standardize. As we just heard, getting Schwann cells to remyelinate in vitro, actually quite a challenge. So there's yins and yangs in all these models. Now, we kind of reviewed this field recently in this paper. I'll take you through a few of the ideas for where we can get these cells as we go through the Cedars Biomanufacturing Center. We have a wide range of iPS cells. I'll mention that as we go along. And we've been basically storing cells from models that we've developed. And I'll give you a very high level view of some of the work that we've done. We've heard a lot from Jim and Jason on motor neuron models. We actually started in this field when I first was in the lab next to Jamie Thompson and iPS were discovered back in 2008, 2009. Jamie kind of said, well, what disease do you think we should model first in humans? And I said, well, it's easy. It's going to be SMA, spinal muscular atrophy, because it's a degeneration of motor neurons early on in life. And sure enough, in that particular model, we generated iPS cells from one mother and one child who had the disease. The mother was a carrier. And we saw beautiful motor neuron death over time in this paper. So that was kind of an easy hit for a modeling. We then turned to more complex diseases, late onset, like Huntington's, through numerous consortiums and show that we do get phenotypes slightly more difficult to see. We also did this for a disease I'm going to come back to in a little bit, rare disease in children, which has a T3 transporter deficit. This is the MCT8 transporter. And that would actually lead us to the blood-brain barrier as a main route of targeting this disease. And it was, I think, a very nice story of going from basic science into the clinic. I'll mention and touch on iPS cells from patients with this mutation as well, where we just published a paper showing some of the omics that we've done on those cells to understand biology. We've been in the Parkinson's space with this paper. And I think one of the themes that I've found in iPS modeling is the earlier the onset of the disease, the more likely you are to see a phenotype in the dish. So in this case, we took early onset Parkinson's patients, no mutations in synuclein or anything else. And we found significant phenotypes in synuclein expression, which we're now moving forward into other tests. And very finally, this is a very recent paper that just came out where we kind of scaled this, and I'm going to talk about that a little bit more now, where we actually have generated iPS lines from a large number of patients to do more of a kind of GWAS differentiation. We differentiated 500 iPS lines into motor neurons. The results of this paper are very interesting. We didn't see a dramatic difference between ALS and controls, or in sporadic cases at least. But we did see a major male-female difference. And I think for the modeling world, we have to, even motor neurons grown in culture, the male-female motor neurons are very different. So I think that's kind of a lesson. We have to maybe separate male and female studies because the biology is very different between the two. And all of these lines, a lot of them are available, again, through Cedars-Sinai. I think that's important because it links to the clinical data. And in the case of Answer ALS, which is the next part I'll talk about, we've actually looked in detail at this disease, ALS. As many of you know, and we've heard a discussion about it from Jim before. Motor neurons die, they're both the upper and lower motor neurons. There's really no drugs, so the ones that are available just extend lifespan by about 3 to 4 months. 10% of cases are familial, and I'll talk about that, but 90%, 85% we just don't know what causes it. So from that point of view, we desperately need to understand and try and find new subtypes. And that really led to Answer ALS, which was this very large global project focusing on, a national project in the US rather, focusing on collecting iPS cells from patients around the country. Again, we just published this, so you can go see this paper, and that represents the amount of collaborators that we had. And really, we took, in this case, 1,000 patients with ALS. We drew blood from a number of clinics around the country, ALS clinics. We actually got the blood to Cedars-Sinai, and the blood also went to New York Genome Center for whole genome sequencing. So once the sequencing was done at Cedars-Sinai, in a remarkably similar building, but green, in our Cedars-Sinai iPS core, we actually did the motor neuron differentiation that you just heard about. And then finally, we collected from these patients 24/7 clinical data. So a lot of wearable information, etc., was available as well. And the idea was, just hearing all the discussion about big data, having this resource is quite valuable. The idea is to try and find a signature for ALS in all of this omics. We're in the middle of trying to find that. Collaborators like Google and IBM at the beginning. And if you could find a subtype, then we could try to create drugs for the input patients based on this signature, so that when a new patient comes in and we've generated the iPS motor neurons, we'd be able to use that. You can go look at this online. This is the data portal. That's at answerals.org. And when you go in, you can actually screen through all of these cases, look for differences, and look at the RNA-seq expression. As time goes on, we're going to add proteomics and ATAC-seq to this data set. But I think the bottom line is it's a good resource for obtaining IPS lines from these well-documented cases. We can use the data and link it to the portal. So if you can kind of order 10 IPS lines, you'll have all of this associated data with it. I think the point of this model is, and being at an eye meeting right now in DC, we could do the same for retinitis pigmentosa and other diseases of the eye, for example. And in fact, any disease, we're thinking about making prostate organoids for prostate cancer, developing these large data sets. I think it's going to be very powerful to convince or to get that into the hands of everybody out there who needs it to do the model. The next part, the few minutes, and I'm giving an overview so we can all discuss this in the discussion after. But of course, we've been doing a lot of organ chip technology, and we do think the future is in these complex models. And the system we've been using is actually a vacuum-fed system developed initially out of Don Ingber's lab at the WIS Institute, that then created the company Emulate. I've been working with Don and the WIS from the very beginning of Emulate, and again, been partnering with them in the systems that they've developed. And I think let's go through this quickly, going back to spinal motor neurons, which has been a favorite topic of the last two speakers. Of course, we make the iPS-derived motor neurons. We've also made BMECs from the same isogenic line, from the same patient, so that we've now got the BMECs and the motor neurons. And published recently, Sam Sands, a graduate student in the lab, actually showed that when you do this together in the organ chip, the blood vessels at the bottom here, the endothelial cells, BMECs, wrap around and make an artificial vessel. And the motor neurons there are on the top with some little sticking out, some end feet, astrocytes through these 7-micron holes. You do create a very nice model of motor neurons in the dish that are electrophysiologically active, as you can see here. And I won't show you all the, you can add tetrodotoxin and block this. You can do a lot of beautiful things, as we've heard about in the previous talks. I think what I got most excited about when we started developing these models, though, was devising a blood-brain barrier. There's a lot of challenges for the field. It's getting these into the clinic. The most fun part of this project was when we actually got to get whole human blood, run it through the blood side of the chip, and ask the question, how much does it maintain the barrier? So when we have IBMCs here, the blood-brain barrier and neural cells on top, and no TNF-alpha or stimulant, it maintains the blood-brain barrier very nicely. This is the blood channel, and you can see there's no leakage into the other channel, the two ends here, the brain side. If you take away the bone marrow, the brain endothelial cells, of course, you get leakage. You can see the blood start to leak. Just likewise, if you stimulate TNF-alpha and you break the blood-brain barrier, you also get leakage through. Very importantly, given the drugs that we know get into the brain or don't get into the brain, such as retigamine, which we know does, they actually passed into the blood-brain barrier compartment just like they do in patients, which is very, very nice to reproduce. Things like colchicine that don't get through so well didn't either. So we kind of validated this blood-brain model. I think overall, in my experience with going from 2D cultures into these physiological cultures, and I have a lot of other data, I don't have time to tell you, this adding flow to in vitro is more physiological. Just sitting like a pond in a 2D culture is, to me, not physiological. It provides nutrients and removes waste, which is very similar to the actual physiology of the body. If you can have like an artificial CSF flowing over the top, and you have the blood at the bottom, and you can then do constant sampling. And that really led to a nice collaboration with Bill Seeley. And I'll tell you one project, this is funded through NCATS. This is the clinical trial on trip part of the NCATS offerings that they had. And so we applied, very happy we got this grant. We're like 2.5 years in. And here we actually picked ALS again. This is ALS. It's caused by a very specific mutation in C9orf. C9orf, if it has repeats, is a very interesting and horrific disease. On one hand, you can get ALS with C9 repeats, and on the other hand, you can get frontotemporal dementia. And we don't really understand why some patients with C9 get FTD and other patients get ALS. That was a very important question for the field. So we're kind of interested in exploring this more and focusing more on the FTD side initially, picking patients who did get FTD. Just quickly on the mutation, this is a repeat disorder between 30 and 1,000 or even 2,000 repeats. You get disease, very complicated biology that there's been lots of papers written on in terms of how the disease happens, but essentially it's a complex disorder where you get loss of function. So the normal C9 protein is reduced by about 50%. So we have a haploinsufficiency. But at the same time, these repeats spit out like a machine gun, these dipeptide toxic fragments and also RAN fragments, RAN peptides, which build up inside the cell. We don't quite know what they do, but we assume they may be toxic. There is also some evidence that they actually accumulate to get rid of their toxicity. So it's kind of a complex mix. But these two pathologies go on, and the question is, can we model it? So we took 8 control subjects, 8 C9orf that had ALS and 8 that had FTD. And the questions were, can we see pathology that represents these patients? Is it possible to do a clinical trial on a chip? And finally, does the pathology correlate with pathology in the patient? And the reason we could do this is the patients that we took, actually Bill Sheely had pathology on them already. We took patients who already died, had pathology. So these patients I mentioned here already died, Bill has pathology. So we can now compare what we see in the dish with the pathology of the brain, which is very exciting. We made the cortical microglia and brain endothelial cortical cells. So we did a cortical chip, put it on top. We get all the phenotypes you'd expect, lots of nice neurons, astrocytes, and C-tip positive cortical neurons in 2 dimensions. We can both do it from the C9orf and the control. And if you look at the top of these chips and do confocal imaging, they make quite a thick block with flow. You get a mini cortex on the top of this chip, and on the bottom you get the endothelial cells. What's also nice and satisfying is these microglial cells that we seed in will remain at about the right density, shown in pink here with IBA1 staining. So you now have your full microglial complement within the organ chip on top, and you have your vascular system underneath. Very quickly on the correlation with the postmortem data, this is one patient that Bill had postmortem data on, looking here at TDP43, which we know mislocalizes in this disease. And we're now starting to look in vitro to see if there's mislocalization as well. And we're in the middle of these studies, but it's very nice that we can actually see what mislocalization was in the brain and then see if our model could have predicted it. And likewise, we can look at antisense foci in the dish on the right, and this is actually the same patient brain tissue on the left, and we're starting to see some evidence that there's correlation, although again, this is early times, but it'd be very nice to have an in vitro pathology report before the patient dies for many, many reasons. There'll be more coming soon. And finally, as a biomarker, these DPRs, as I mentioned, are spat out. These are symptomatic carriers looking at poly-G expression in the brain. They don't get too easily into the CSF and other tissues. You do see them, but mainly in the brain itself. And this is controls here looking at polypeptide expression. And you can see nice distribution. The ALS patients are having expression, and that expression is variable between patients, and we can now correlate that with the patient data. So just to summarize, a lot of work that's going on, and Deepti Lal in my lab has been working on this for a while, and she's now got this whole kind of output, biomarker output. And the first time we have live analysis, which includes the imaging, just measuring LDH, neurofilament release. So this is our kind of biomarker panel. We could do metabolomics. We're doing less of that, but more on the lactate dehydrogenase and ELISA. And then we can do post-analysis after death, if you like, when we kill the chip, look at a lot of the other biology. And we're working with companies, Denali, we're just finishing off the contracting, Curastem, Mitochondrial Motion, and Ionis, who is a big ASO company working with Biogen. And we're actually testing a lot of their compounds and seeing if we can give them information back for this clinical trial on a chip that would inform them in their clinical trial and perhaps be used as part of an IND-enabling study in the future. I feel my feeling on this is it's going to be integrated into their packages. I know Jim has got more success than we have, but I think the initial stages is going to be integrating this kind of technology to enhance current INDs rather than, on their own standalone being able to support an IND. And I think that's the way we'll infiltrate. And then eventually, of course, the goal is to take over from animal studies in the future. But right now, I think if we can integrate these technologies, it'll be great. So we can summarize biomarkers. We might be able to predict drug response. And again, I think the future of this technology is using the patient's own cells. So in 10 years' time, every patient will probably have their iPS line made before they can enter a trial. And we'll have segregated them into whether they were a responder or non-responder prior to entering the trial. This is going to have a massive impact on the success of trials and precision health. So that's kind of where we're at. The last few minutes, I'll finish up with this disease, which I've been working on for quite a while. With Dr. Gad Vatin, who was a postdoc in my lab and now is back in Israel and has his own lab. And this is a unique disease where you lose there's a specific deficit in the MCT8 transporter. And MCT8 transports thyroid hormone into the brain. And paradoxically, because you don't have a transporter, it builds up, it can't get into tissue, so it builds up in the blood, thyroid hormone. You get lack of speech, delayed myelination, feeding difficulties. It's involved in development, and we know it's caused by this mutation in this MCT8 gene. It's a monocarboxylate transporter. And TH analogs can help the periphery, but has no effect on the neurological symptoms, so we're desperate for something that affects the brain. And so we published some time ago that we made iPS cells from both the patient with MCT8 transporter. In fact, we did 3 patients. We took an MCT8 patient and CRISPR edited it back. And we also took an iPS line and gave it the mutation. So we did it in both directions. And very interestingly, we were expecting to have all sorts of deficits in the neurons. Well, they showed absolutely no deficit. And Gad worked for 2 years with no deficits. And I just kept saying, this is science and this is fine. There's no deficit. It means that these cells can somehow compensate for the lack of the transporter. So the neurons and the glia seemed fine. However, then we thought, well, let's look at the blood-brain barrier. So together with Eric Schuster and some of my other colleagues in Wisconsin, we developed the blood-brain barrier model in inserts. And we actually showed that when you use this functional assay, healthy human obviously blocks T3, but we had massive effect on T3 transport using this model in the MCT8 transporter deficient line. So we really summarize this. We can get normal cells, differentiation work nicely. When we made these brain endothelial cells, they had a very severe deficit in the transporter. And now we've got this model to test drugs that can get T3 into the brain. And we've been utilizing that. In addition to the model for getting drugs into the brain, it gives us a platform to think about therapeutics. As we all know, MCT8 gene therapy is coming. There's lots of trials going on. Of course, SMA is the archetypical trial where there's now been over 1,000 kids dosed by Novartis, the Zolgensma. However, this vector can also be used to deliver other things. We've shown now quite nicely that it can correct a mouse model. There is a mouse model of MCT8 deficiency. We can reverse that in the mouse model. And in fact, due to our data a little bit, other groups have been focusing on AAVs, which actually target the blood-brain barrier cells. And those AAVs may be even more effective because we don't need to get the vector into the brain, it turns out from our data. It's enough to get the endothelial cells, the vascular tissues of the brain, expressing this transporter, and then the transporter should get through and it should help the kids. And how we envisage using this right now, we're applying to the FDA. We've got a pre-IND approved already with a partner company called Prison. And we're really at this early stage of preclinical development, proof of concept in the mouse. We're trying to decide on route of delivery, but recently, thanks to the organ chip studies, really, we decided that systemic will be okay because systemic should label the blood vessels and that should get it into the brain. So we don't need to go from the CSF angle. So we're really following this route of systemic delivery thanks to the data that we've gotten from the organ chip systems. And whether we can include that in our filing is something that we're going to discuss as we push this forward into a clinical trial. And we're applying to the state California CERM for this particular gene therapy. And then finally, I'll finish up on the other use that we've used it for, and just to show you this data that we showed that we could take blood, put it through the chip, and measure T3. This was done by Gad when he was in my lab, and it was in one of the papers. And if you measure T3 with mass spec coming through the other side of the blood-brain barrier, we can see that in MCT8 you get a massive reduction in T3 transport. And if you correct it with isogenic lines and you correct the mutation, the transport comes back. So again, a good model of getting T3 through. This is all Gad's work. The last two slides, he shared these with me recently. And he's saying, well, could we actually do the clinical trial on the chip and give the AAV to the chip and see if it gets in? Because AAV is pretty bad at infecting cells in culture. You get about 10% transfection, and that's always been an issue. AAV9 doesn't work very well in 2D. So he's developed the same emulate chip system, runs it through he runs the AAV through either the ICV mimic, which is putting it on the brain side, or he can IV mimic and put it on the blood side, and then looks at the chip and does imaging and facts. Very early days, guys. This is unpublished, all from Gad's group, and he shared this with me. But he is seeing increase when you have a static. You don't get many GFP positive cells, which are in this case just using AAV GFP as a marker. But when you put flow on, you get a massive increase in the number of GFP positive cells. And likewise, this is from the endothelial side. And then when you look at the neural side, it's even more impressive. You go from 20% up to 35% just by adding flow. So this is helping, we think, dosing maybe the virus a little higher, but also helping penetrate the tissue. So it's kind of a model that we can use for assessing AAV delivery, either CSF or through the vascular system. So to end, just on the first half, we've generated these IPS for developing models, and now we're using these advanced 3D technologies. And I think the future clinical trial on a chip with some of the technologies that Jim mentioned and Jason and what I just went through, we want to try and integrate this more into the IND packages as they're developed and try and push this into the clinic. For the most important part, thank you to all the people who did this work in my lab over the years, and I've probably missed people out. And in particular for the MCT8 story, I'd really like to thank Gad as one of my fantastic postdocs in the lab who's doing remarkable things in Israel now. And I'll thank everybody else on this slide as well. And I'll stop there and look forward to the discussion.
Thank you so much, Dr. Spendenson. That was a great talk. Now we're going to go to our last but not least of our talks for the afternoon before we go into the discussion. So this is Dr. Klee Kim from Regenix Bio. And the title of the talk is In Vitro Pharmacology Study of Gene Therapy Using Retina Organoids and Retina on a Chip of CLN2 Patient-Derived Induced Pluripotent Stem Cells. Dr. Kim?
Do you see the slides?
Yes, it's not in presenter mode yet. There we go.
Okay. Well, thank you for the invitation and the opportunity to share this story using retinal organoids and retina on a chip for CLN2. The data was generated to support the IND and CTA submission, and it was included for European CTA submission. So before I go into the study itself, let me just introduce the CLN2 disease. CLN2 is one of the subset of lysosomal storage disorder called Batten disease. CLN2 patients have onset of symptoms at about 2 to 4 years of their age. Symptoms include recurrent seizures, loss of motor skills, loss of cognitive ability, and as also have visual impairment. Currently, there's enzyme replacement therapy, which is injected ventricularly into the brain directly. But there's still unmet needs even with the enzyme replacement. That even though the ERT slows down the progress of the disease, it doesn't really stop the vision loss in this patient. So we were focusing on how we can rescue the eyesight of this patient. As you can see on the right side, on the right side, you can see the difference of OCT and RP and photoreceptor layers in these pictures. in the normal, you can see a very nicely defined retina structure, but in CLN2, you see the hallmark of the macular bullseye macula in these CLN2 patients, and the retina structures are in disarray, and eventually they lose the eyesight. So what's the limitation of the current models? So before we go into the iPSC, the model using iPSC, we had a study with the non-human primates where we injected subretinally to express AAV9 HCLN2 genes. The vectors were delivered subretinally, and the vector delivery was confirmed by qPCR. As you can see on these two graphs, after delivery of those vectors, TPP1 expression was greatly increased over the untreated NHPIs, both in aqueous humor and vitreous humors. But the problem is this is a wild type NHP, and somehow we need to show the disease pharmacology in the CLN2 background. There's mouse model that's been around for a long time. The mouse models are really great model to show the CNS phenotypes such as seizures, impairment of motor skills, and eventually the premature death, but they do not show ocular phenotypes, unfortunately. When we started this iPS-derived retina organoid studies around 2019, there was a dog model available, but it was a little bit hard to get to the model. But the study showed that repeated injection of the recombinant enzyme could attenuate the progression of the retinal disease. Recently, there was another animal model published, which is porcine model, where they show similar CNS symptoms as different animal models, as well the ocular phenotypes, but the porcine model was not available when we started this. So with the limitation of the animal models, we were trying to find the alternative ways to do pharmacological studies with our vectors. And we had a CLN2 iPSCs already generated by the time. And instead of going into 2D model like RPEs or photoreceptors, we decided to go with the retinal organoids. There were several publications were available with the retinal organoid generation with the iPSCs, where they show the photoreceptor layers and other retinal cell types within the retinal organoids. About the time when we started, the group at University of Tübingen came out with this publication where they separated out the RP, retina pigmented epithelial cells, from the retinal organoids to put it at the bottom of the chip, and then embed the retina organoids without the RP on top of it with the hydrogel, so that you can make a more complex and complete retina system in a chip. So when the paper was about to publish, we decided to collaborate with this group and see if we can model CLN2 ocular disease within the chip as well as retinal organoids. So the study design is we already had made a CLN2 patient iPSCs at the time, and first of all, we want to. Before I go into that, one of the reasons that we wanted to do 3D, another reason that we wanted to do 3D is that not only they're complex, we did not have the understanding of which cell types are really affected in the CLN2 ocular tissues. It could be all the cells, or it could be RP, it could be photoreceptors, or it could be any other cell types within the retina. And if we choose one cell types, we might miss the opportunity to look at other cell types that could be more detrimental in the CLN2 patients. So with our iPSCs, we first phase was to generate RPs, retinal organoids, and then retina on a chip, and see if we can see the TPP expression in the control and absence of CLN2 lines. And if there's a difference, which cell types are showing the difference. And also, another question that we wanted to see was, can we look at the disease phenotype in this retina organoids or retina on a chip? Here we use the mitochondrial ATPase subunit C, or SCMAS, as the disease marker that was used in many other in vivo studies as well. Once that's established, we were going to characterize Rs, and once the Rs and RCs are characterized, this will be treated with our AAV vector and see if we can see if we can restore the TPP1 expression. And if we see the SCMAs in the CLN2, can we decrease or slow down the SCMAs accumulation? Also, we were going to do TPP1 protein quantification and then biodistribution as well. So here's the outline where all these different cell types and the differentiation. So we got 2 patient fibroblasts from the Korean Institute and reprogrammed into iPSCs. The mutation was confirmed by sequencing, and they were differentiated by the published protocols. And I put example dates where the samples were collected, but later we did different time points as well. iPSCs were differentiated in the retinal organoids or neuroretinal field with RPs, the black dots here, and they were separated out into monolayer. RPs were separated out as a monolayer, and the retinal organoids were mature further. RPs were characterized first. So ZO1 is the tight junction formation, and the LAMP2 is the lysosomal proteins. And as you can see, the TPP1 was present in the control RPs, but it was missing or very minimal in the patient lines. So this shows that our RP could be one of the target cell types that we can use for cell AAV target. What's more interesting is with the retinal organoids. So we collected retinal organoids at the time of the RP separation and mature further. Around the time when we got the RPs out, you can see the TPP1 expression was very minimal, both in control cells and the CLN2 patient cells. Recovering is the photoreceptor marker, and they were very minimal in both cases, and SCMAS was absent. When they mature further, TPP1 expression is shown in the control arrows, but not in the CLN2. recovering is there in all four of them, showing that differentiation is going nicely even for the CLN2 cell types. SCMAS started to be accumulated in the CLN2 ROs. So this shows that TPP1, one more thing here is that TPP1 is more localized into the 4 receptor region, and the SCMAS accumulation is also more concentrated in the 4 receptor region. So this really shows something that we never shown in vivo, that the TPP1 is expressed temporally, differentially expressed in time and in location. It's same figures, but subunit C or SCMAS was quantified by imaging intensities, and you can see that at later time points, the accumulation of SCMAS in the patient lines. What happens in the retina on a chip? It was very similar that TPP1 expression was very minimal in the patient lines compared to control, and SCMAS was higher. So now we wanted to treat this retina organoids or retina on a chip with the our vector now that we characterized. Once we transduce the cell retina organoids, we could restore the TPP1, and then with the expression of TPP1, we could lower the SCMAS accumulations at early time points. Here we did 3 different time points. At early, when the SCMAS accumulation just started, I'll jump into the mature part, where it's almost 1 year old, where the SCMAS decrease is minimal, and then TPP1 expression is a little bit lower than what we saw in the early time points. With the lower SCMAS decrease, we were wondering if this is if TPP1 can reverse the SCMAS accumulation, or is it just preventing SCMAS accumulation? So that's why we included another time points in says intermediate, where there's a very SCMAS accumulation just started to show. That's when we treated the cells with the vector, and you can see that as the accumulation went down almost to the control level at this point. So this indicates that treating at earlier time points is more beneficial for the patients. Same story for the retina on a chip. Here we have 2 time points. TPP1 expression was dose-dependently increased, just like the retina organoids, and SCMAS was accumulation was decreased. Vector delivery and TPP1 quantification. So previous analysis was done by image analysis. Here we added actual quantification. So the first column shows the DNA delivery into the retina organoids or retina on a chip. The TPP1 was measured in the media as a secreted protein, or TPP1 in the retina organoids, within the retina organoids or in a chip. So this all show there's a slight difference between RO and RC. That's due to the environment, but we see the dose-dependent increase of the protein production. So in summary, we looked at 3 different time points: early, intermediate, mature, and this is what we are hypothesizing, that At early intervention, we can prevent the storage material accumulation. When we intervene where the storage materials start to accumulate, we can prevent further accumulation as well as reverse those storage materials. At the mature stage where a lot of months of accumulation, it's going to be hard to completely reverse the storage materials, but we can at least maintain the level of the SCMAs. So throughout this study, there's two things that we show. That one is that something that we never seen in vivo, which cell types are affected. And it looks like the photoreceptors are more affected by in CLN2. If we started with the 2D model such as RP, we might have missed that window. And by having complex model, we can just see more than one scenario at a time. And two is this will be probably same for many other diseases. Early intervention will be more effective than later stage of the disease. With that, I want to thank everyone who was involved in this project, especially Serena and Kevin, who was working at TVN. They work really tirelessly to generate this organoids and organ on a chip and do all the data analysis. And our lab member, Ryan and Chris, who did all these TPP1 assays and DNA qPCR assays as well. With that, I'll finish and take the questions.
Thank you so much, Dr. Kim, and thank you to all of our panelists this afternoon, in particular for staying on time so that we have plenty of time for our discussion here. To those in the audience, please feel free to enter any questions that you may have in the Q&A so we can address the questions. While we're waiting for any questions from the audience, I wanted to start off really with kind of the big question. I heard this afternoon different uses of these 3D models for either discovery or the development pipeline. And, so these models are being used at different steps. I would love to hear from the group here, where do you think that these types of models can have the most immediate impact for rare disease therapeutic development?
Well, I think it's basically by providing a phenotypic model, right? Because we don't have to have a target, because we can just simply get observe clinically what the deficits are, okay, for that disease, rare disease, and we can identify what the cells are that are involved, and then differentiate them out. And as long as we have a functional model, we can create a phenotypic system, right? I mean, before molecular biology, all drug discovery was done for phenotypic models, right? It was either in humans or it was in animals. And so we just go back to doing that, which is you have a disease model, you have the wild type, and then if your drug makes it better, if you can, after you've created the disease model, if it shows a phenotype, FDA is accepting that data because it always has. It always has accepted phenotypic data that if you have a disease and it gets better, they will allow you to put it in patients as long as you have safety data.
Yeah, I can add to that as well. I agree with Jim. Also, I didn't show you today. I think there's some also interesting results using muscle organ on a chip models. So where you can actually look at, and this I think gets to the comments from Jim and what I was also showing from a functionality, showing clinically relevant endpoints, so to show improvement in function. So that's what we were showing for the zilucoplan. Then also there's some interesting muscle models where they've shown in say DM1 or that you can rescue and improve the muscle contractility. So I think you could use it in that pharmacology space, but also you can start to use it in the PD space, where if you don't have in vivo models, you can use that to better understand your pharmacodynamics.
Well, I agree. I think what's critically missing for all of us is validation in a human trial. So what I'd like to see, I think will really move the field forward, is if you had 3 drugs, 2 of which didn't work in a patient or 1 did, could you have predicted that from your model? And I think, Emulate had a paper recently on liver chip and predicting toxicity, which seemed to do a better job than animals. We need to do something kind of similar, I think, something that would really prove that our model had validity in the clinic, because we can talk about endpoints in vitro, but if it doesn't have clinical relevance, then none of it's going to matter, right? So I think that would be a goal that we should think about. What could we really test? Is there a drug that, the problem with neurological diseases, we don't have many drugs that really work very well. And so we're often limited in ALS, for example. Oh, let's put the drug that works on. Well, there is no drug that works. So we don't have a positive control to really validate if our model would work in patients. Whereas in some neuromuscular disorders that was mentioned, there are some drugs which work. And I think the acetylcholine receptor is a good target. And, did that predict something, Jason, that, you could validate in the clinic? You could maybe work backwards from data you already have to prove that.
I mean, well, we probably by the end of the year will have 15 INDs filed, and we already have a number that are in clinical trials. Wait and see what happens with those clinical trials. I mean, if we start getting drugs into patients, I mean, right now we're doing a clinical trial of one rare disease, a rare variant of CMT. And it's such a rare variant, it's only one, but we hope to be able to have drug in that patient within a year after starting, which will be in about 3 or 4 months. Assuming that everything keeps going the way what we see. But that's another thing that we can start looking at too, which is clinical trials of one. I mean, we're already doing it. That people are already, collecting the samples, creating the models for it, and then, showing the disease phenotype and already, treating it now. And as soon as we start getting some of those done, because those go a lot faster than a recruitment trial, right? So that's another aspect.
I think it's great, a great idea. What probably will happen, as always happens, say your model, you predicted your drug was going to work beautifully, and that's how it got through the IND and it's reversed it. What never happens is we never hear the trial failed. Does that mean your model is useless? And do we ever report that back? Because nobody wants to say the model didn't work, right? So I think we have to have a feedback somehow from these trials that would be useful. And it's very difficult to get that information post-trial because it all gets closed up. I mean, it's just a practical issue. I'm not saying it's not interesting, but worth discussing.
I mean, stuff always fails, but generally, if it's a public company, it's telling its investors that they've got this clinical trial going. Just follow their stock price, right? You'll know pretty quickly whether or not for a public company whether it worked or it didn't, or if you just listen into their quarterly stockholder meetings or whatever. So we're finding better ways of getting that information, and there are ways to get it. It's just not what we would call scientifically palatable in many ways.
Yeah, and I think one of the challenges is while you may hear the negative result just from, as you were saying, if it's a publicly traded company, from some shareholder meeting, but it doesn't necessarily answer the question, what part of the model failed? And so how can we iterate? Was it really the model that failed? Was it something about the individual that was recruited that, or the outcome measures that were selected that didn't actually capture the improvement that, the primary endpoint was the wrong one.
Actually, that's a really interesting point, because we're starting to take our models and do some retrospective analysis with some companies and starting to show them they should have stratified their model. They should have stratified their clinical trial, because we can show them the data that shows what they should have done. And it also gets to a point too, in that what you were saying, Dominique, we just had it was an IND, it was accepted, they rare orphan drug designation, but it was a strategic decision by the company not to continue pursuing it. So the model worked, the FDA accepted it, but it strategically was something they decided they didn't want to do. So, and that's one of the other problems with rare diseases too, is that, sometimes it's a hard sell for a drug company that's trying to make a lot of money to keep going with something. If it's not a real slam dunk for them, or if it looks like the space is getting too crowded, they may just decide not to pursue it either. So that's another aspect that we have to, I think it's one of the reasons that the more we use these human on a chip functional models, we'll start decreasing the cost, I hope. Because the one we did with Sanofi was done at a fraction of the cost it would normally take to repurpose a drug. We'll hopefully start making it more attractive for companies, especially smaller companies, to stay with it, longer, because their upfront costs haven't been as high. And that's one of the things we're trying very hard to do.
And that really leads me to a question I was going to ask a little bit later, but since we're naturally there in this discussion, what then is the role of some intramural, extramural collaborations versus the role of drug companies, which certainly has its benefits and challenges, and the role of patient foundations? I mean, these are all important stakeholders who each have their own priorities that may not necessarily align with the goal of the patient. So kind of what are thoughts on how NCATS versus drug companies versus patient foundations can be involved in advancing this science so that we can get more treatments to more people faster?
Well, I've been talking too much, so I'll let you guys go.
So the data we generated, it was collaboration with the academic lab, which was, for me, it was really great that, they have really cutting edge technology. We had the idea and then we had the drug. And when we combine them together, it really works synergistically. That in the beginning, it started as a smaller collaboration, but when we saw the opportunity, we worked together. And trust me, when you involve postdocs, it's really faster than scientists within the company. For me, it was really great experience, and I would go for that again if I have the chance to do it.
So I got the reverse. So we're not a company, we're an academic. Cedars-Sinai is a nonprofit academic. First, NCATS has been fantastic, right? These chip grants introduced me to the whole field, and met Jim and all the chippers, and it's been great learning. And then we reached out to Denali and Ionis and asked them if they wanted to try some of their drugs, in the chip system, see if we could add something. And it took a few lawyers and a few MTAs and work to get it all set with a contract and everything. But if you do it right, and I think we know we're both going to benefit from the interaction of learning about their drug pipeline. One of the things they're very sensitive about is drugs that, you might find a toxic effect of a drug that already is on the market. So all they want to do is change it by one molecule so that you're using something very similar and you can test it out, but it's still not the real thing. And so it's not going to have an impact on anything that they've already done. And so those are the kind of the tricks you can have to understand. Again, I think working with the companies, like I said before, for me, validation is everything. And they're finding maybe a drug that failed. Could you have predicted it? Because in their vaults, believe me, drug companies have a lot of failed drugs. I mean, they're very, and if you just let us have them and see if we could have told, like Jim just, if we could have told you that would have failed, it would have saved you a phase 3 clinical trial. You got me, right? So I think these kind of interactions are really critical for the field. If NIH can support any of that and get off this nervousness of drug development, I would lock into all the failed drugs. I think we can learn more from failed drugs than we can of successful ones. Obviously it's nice to have a positive control, but anyway.
Actually, I was looking into that before I was working on the retinal organoids. I was looking for more on tox screen with the in vitro model. And I was trying to find, which drugs are really failed in the. So you see all these drugs that was approved for the IND studies, and then they closed, and you never know why they closed the studies. Is there safety issues or the efficacy was not there? That's never reported. And if there is a database where all these what the target of these drugs are, I think this can be done at the NIH level, where there's a database and then showing that what's the target, was why the drug was stopped developed. Then we can go back with a new platform that can we predict better with the neural system. And that information is really missing in the market right now.
Well, actually, as a CRO, we get their failed compounds all the time. They want to see that as their first validation. Okay, either a known compound, like the stuff with AstraZeneca, they wanted to see terafenadine, fexofenadine, and we showed it. We predicted the in vivo outcomes for it for both PK and PD, but they weren't going to let us look at anything they were really interested in until we ran those validations. So as a CRO, we do get to see that. But getting back to your original point, without NIH money, we wouldn't be able to do a lot of rare disease work, because for whatever reason, the rare disease foundations won't even look at us, because we're in Orlando, we're not a big name, we're not associated with a big university. They just don't even look at us. We've given up trying to go to the rare disease foundations and everything else like that. We're either getting money through the NIH for it, or it's from a company that's trying to develop a molecule for a rare disease. But we get no foundation money, and no matter, it hasn't been for lack of trying either. Zero.
Yeah, Dominic, maybe a slightly different take as well. I think you mentioned, patient advocacy groups, or actually where we've found some challenges from a industry point of view is getting access to patient sera or patient cells, and then being actually to use that for drug discovery purposes. So a lot of the times the patient consent isn't allowable for drug testing, or when the clinical trials, you can't use those for, depending on what the context was, you can't use them in an early drug discovery setting. So so sometimes it's actually we're inhibitory, where you need to think ahead for in the clinical trials that can the samples be used to go back and validate these models. But then there's also the challenge of creating additional data that could be impactful on when going to for a final decision.
And that's a great point. It's something that was brought up in the prior session about a lot of the hurdles with using patient samples or patient data, that often it's not the patients themselves that don't want it to be used, but rather maybe the lawyers or the powers that be that have put language into place or not put language into place, not realizing that they would have needed to make it a very broad consent in some way, shape, or form to allow the use. But it could certainly be. I hear you, Jim, that you've had difficulty getting kind of patient foundations to engage in a manner that where you're ready to engage and they're not reciprocating. But so maybe it's a combination of figuring out how the patient foundations can bring patient samples to those that can use them. And like you're saying, a CRO may not be the name or, a familiarity that they have. So how can that process improve, as well as when others are collecting samples, whether it's an academic lab or a company doing, a preliminary study, making sure that those consents have language.
Well, I guess it's more, it's just, we haven't had the foundations engage with us to use our systems, to do studies. We don't have any problem getting patient samples because you can buy them. And you also have networks. We have networks of folks, I've been around, Mike and I have been in academia for a long time. We know folks, so getting the patient serum samples and getting the induced pluripotent stem cells, there are banks and things like that where you can get them from, or you can buy them. It's just more of a, we know that there's certain rare diseases we can, have models for that we could evaluate their systems, but no company has come to us to contract with us for the services, even though we know we could help that rare disease, but since the Rare Disease Foundation isn't going to give us any money to do it, and a company isn't going to come to us to fund their study, there's nothing we can do.
So I just wanted to remind the presenters, if you see any technical questions, it's always better to answer it in question and answer section. And we have one more general question that's, are the models also proprietary, or can others have access to the validated to validate them and determine which models are better to model disease? So I guess this is a question to all of you.
We're happy to collaborate. We don't, and so yeah, that's all I can say. Give me an email and we'll help you set up the model. And I love validation. Anytime somebody can just repeat something we do, I'd be super excited. I think we all need to repeat each other's work. So we'd be happy to chat to anybody who wants to try these models.
So our collaborator has the ownership of the technology. So I can't really say about the if it's appropriate. What I'm sure they can collaborate. They are open for collaboration all the time.
Yeah, I can comment. For the neuromuscular junction model, we'll probably hopefully be having a paper coming out late this year, early next year. But also, as I mentioned, we did collaborate with the NAND device, so you could reach out to them as well.
Yeah, and I also saw in the questionnaire about the sharing. We publish everything and present everything, unless we're bound by contract not to, and that's generally a very compound specific for a specific company. But no, Mike and I have gone out of our way to publish everything we do. And somebody lets us know, if somebody reaches out and says, could you give us a webinar on your system? We do that all the time. So we're happy to share any information that we have in our systems.
Thank you for the answer. So I have also question relevant to you all have these wonderful complex models that are very good for mechanistic studies and for things that you mentioned, like efficacy and everything. But when you think about drug discovery, and you can argue this, how simple models need to be in order to be able to go to the more high throughput. So considering your models, do you think there's some level of reduction you could do relatively easy that you could then adapt them to a more high throughput? The second question is, and I like to think about this because it's something I see these technologies also as a tool to be able to a little bit more democratize drug discovery and development as well. So we don't always have to go to big pharma to be able to complete this. So do you have efforts that would go in this direction?
Well, we're not compatible with high throughput. Because we're high-end models, and that's, our model is we custom build what people want, and generally they have their 3 or 4 compounds, but they get the high end we try to give them the information before they have to scale up to go into an animal model for safety or something like that, because the go, no go decision point for most of these things is going from milligrams to kilograms. Okay. Once they make that decision, they're stuck. That's the molecule. So I think it was Clive or somebody else earlier was saying that, just trying to increase the success rate of once you go into an animal model for safety testing from 10% to 20%, cut the cost of drug development in half. So we can inform, say, if you've got a tool compound, where you're looking at different modifications of it, we can look at all the different variations of it and better inform before you go into the really high cost aspects of scale up and animal safety testing. Okay. So that's where the value added for what we do is. Now we also can do safety testing. We can also work with clinical. We can also work with repurposing. I really like the talk that was earlier, and I don't remember, where they're looking at trying to draw correlations between drugs that already exist to try and, repurpose them by getting some aspects of that drug. So where you could have maybe 5 to 10 drugs that you've looked at the available drugs and said, these might work. That's where you use what we're trying to do. Okay, because then you say, what's the rare disease? We build the model for that rare disease. And then we look at the 5 or 10 that they've come up with by exploring the existing database that already has the safety data, right? That's when it really starts becoming, a big deal in really addressing rare diseases. If that project works and they can start with just all about repurposing drugs for rare diseases, us and others, we can build that can be able to, and again, we've already shown we can get those phenotypic models into a phase 2.
So that's your opinion. This is the area that will bring the most impact.
I believe so.
Okay.
I'll just add that, and I agree with Jim's assessment. We're not high throughput. We never will be high throughput in the case of 96 well plates, even 384. Forget that. The only thing we could do a little better is, I emphasize this to my lab because of the cost of the chip model, is treat the chip like a patient. If we could get biomarker responses, so if you could run 20 drugs through the bottom blood channel and measure a response in the brain side, so keep the chip alive. Now our challenge there is keeping them alive for more than 28 days on PDMS has been a bit tricky, and I think we have to get better at our substrates and longevity. But if we could get a chip that could live for 6 months, it's a constant assay for you. You can keep running drugs through the bottom channel. It's like your patient. I mean, theoretically, maybe, but that's still not what I call high throughput. But at least Jim gets a little further towards the idea of being able to do multiple drugs on, and then you could have like 6 Parkinson's patients and 6 controls, and just run your drug study on the chip. And we'd love to get to that, and we're trying desperately to, because A, will save us a ton of money, so we don't have to keep setting chips up every 28 days, which is expensive. But B, I think it gets more to the real power of this technology for NCATS is clinical trial on the chip, is having that chip be your patient and having 10 patients in the incubator, 5 control, 5 ALS, or whatever you want, neuromuscular, and being able to run drugs through that day 24/7 automated. Yeah, not really high throughput, but it's not bad if you don't kill them. Now, if you kill one, you come in the morning and they're all dead, you found a drug that was toxic. You have to start again.
Yeah, Clive, yeah, I was going to say I agree with the whole biosensor piece, and it's interesting you bring that up. So just recently involved, I'll put a plug in for the NC3Rs. There's a biosensor on a chip cracker challenge that's involved in that. We see that as actually a pretty important step of using these models where you can get more information from the models and maintain them longer in culture. And then one additional piece, just from a high throughput point of view, yeah, we've found it. I suppose we would like to have them higher throughput, but then it's that balance between complexity and throughput. So where can we use more singles culture systems that I showed the motor neuron work, then going into a more complex system?
Yeah, I was just going to say, so working with the organoids and organ on a chip, probably if we work with the organoids in 96 well plate, not high throughput, but medium throughput, where we can screen the conditions. And moving on to the chip, by doing that, we can get more sophisticated platform for the study. And as Clive said, I consider one organoid or organ on a chip as one mouse or one animal. So if I have 3 chips, it's equivalent of 3 animals. So it's not really a high throughput, but it's more like, it looks more like a biological replicate in my mind.
Well, I was more interested, like in the process of building this complex, did you come to the point where you say, okay, I'm going to build it more complex, but this is the minimal number of conditions that I can see that it's physiologically relevant to disease, and at that stage, this can be used for a more high throughput.
Mm-hmm.
Yeah. I wish we knew when that was. The problem is you don't know, right? You just sometimes just keep going. Oh, should we add microglia? Yeah. Should we add this? Parasites? Should we just keep going? And if we don't know, you might have done it with just a 2D, like I said. If we don't have a validation in a trial, it's kind of a complex question. And I don't think we really know. It's a lot of fun building these complex models, but we don't know how far you need to go to validate a drug is what we all need to know. So I think we'll get there through these iterative studies. We're really on the cutting edge of all this, and it's exciting to deal with these questions and try and figure out how far do you have to be physiologically relevant in a human.
So the other thing that I like that you, Clive, brought is that when you were studying ALS, you were addressing multiple paths of pathogenesis because it's a complex disease. Now, the other way these chips can be useful, that you once you develop a technology for one disease that affects motor neurons or something, you can then develop the similar model for multiple diseases that have a similar pathological effect, and then shorten the time, potentially discover similar treatment that come across diseases. So one example, ALS would be one, but another one would be like neurological diseases have usually groups of like dystonia, spinocerebellar ataxias, and everything. So do you see that there is a power building a larger consortia where the technology developed will be then shared, and the same tools apply, the specific cells applied, but the same tools then be used to check similar treatments across multiple diseases.
Yeah, and I'll give you one example, which is I gave was the FTD-ALS, which is the same mutation C9 gives you either FTD or ALS. So the dream scenario is we'll see a patient who had ALS, and we make the iPS cells, we put them on a motor cortex chip, a motor chip for the motor neurons, and we see a phenotype, but we don't see that in the cortical chip. And vice versa, if you have an FTD patient, we see it the other way around, and that would validate with the same model 2 different spectrums of a disease, and you predicted it. And then the really cool thing with that is there are C9 carriers walking around today with C9, just like Huntington's disease, where you can be a carrier that repeats and not have the disease because we've identified them. It's the most frightening thing to be walking around with this horrible death sentence, essentially, of this disease, just like Huntington's repeats. So we're trying to think about ways that you can take those patients who haven't converted yet and use these models to predict when they might convert, because that's really important about, when they start to get aggressive on potential interventions, etc. So I think for rare diseases like this, if we can do a predictor model and there's a phenotype, and just finally, you're not likely to get the cell death phenotype, but I think what I love about modeling is you're seeing the very, very early stages of the disease. And I think the molecular hiccup that happens early on that then leads to death in 5 to 10 years later, that's what we want to try and prevent and reverse. So we're not expecting, like in the SMA study, death, but I think these phenotypes, if they're molecular and reproducible, could be very valuable. And then you can try and reverse them before the disease even starts for these. A lot of these rare diseases are inherited, especially in kids. So I think we have the potential is this, having these biopsies and predictors of disease is fantastic, very exciting. We did it for BRCA1. Actually, BRCA1 mutation gives you either breast cancer or ovarian, and we showed women who haven't even got the disease yet, we can make the fallopian tubes and they actually form the tumors before the woman even gets the disease. And that's kind of a modeling in the dish of these rare diseases is going to be very powerful. Anyway.
To kind of expand on this a little bit in terms of, while the ALS FTD spectrum was a great way to potentially look at 2 different diseases with one, model. Is there a way to use these 3D models in a way that allows you to look at multiple diseases. Can you use, make it more of a platform where you build something and you might have to modify based on, a particular genetic mutation, but really to make these applicable to more than just one disease at a time.
Well, I think we're already doing that with the NMJ platform. We're doing it over ALS. We're doing it over myasthenia gravis, Charcot-Marie Tooth disease, NGAL1. We're using it for 4 different diseases, as well as a couple others. And so we're also our LTP assay. We're using that for Alzheimer's, FTD, stress models, as well. So we're already developing that, in terms of the assay for a particular function can be used across many diseases.
Yeah, I mean, the blood-brain barrier can work for lots of different diseases. And, if you've got Parkinson's iPS cells or spinal muscular atrophy or Huntington's disease, you can load them on in that model. And, putting the microglia in and getting the BMECs is standard, straightforward. And then it depends what part of the brain you're trying to mimic: cortical, motor neurons, striatal, mesencephalon, etc. But yeah, I think this technology is pretty shared across neurological diseases. And then everybody else is trying to get their own. Like Jim's doing great work, and just these multi-organ chips will deal with lots of different diseases. And metabolizing the drug through the liver on the chip prior to getting to the brain is really crucial as well for some of the work. And it's very exciting. And I think these are very adaptable and flexible systems.
So I have one additional question that more goes about the collaboration and the needs of laboratories like yours to advance science. So I'm particularly interested in the role NCATS can play besides funding. What else could NCATS do? And I don't know whether everyone was here at the beginning when Liz and Mark were talking about the different capabilities NCATS intramural laboratories have. But when you think about your research and think about the possible roadblocks, is there anything that you would benefit from closer collaboration with NCATS when it comes to, you already said that your systems are difficult to miniaturize. We do have experience with development of assays and miniaturization, but is there anything else? Is there a need to really, at least some of you, is the really making a large amount of cells that are produced in a non-biased manner by removing the human component as much in an automated way? Is that something? Is there anything else, or drug screening platform using our capabilities to drug screen? What else do you think? Where else there's a role for NCATS that can play in helping you remove some of the roadblocks that you may be experiencing?
Yeah, I can make first comment. The probably at the getting a quicker probably access to patient iPSC cells from diseased patients, and I would also want to call out being involved from when I was at GSK and also UCB. The tissue chip consortia through NCATS has been phenomenal. So the work that the different groups have done, and also understanding what the next platforms are coming down from academia have been super helpful in understanding at what stage would these be ready for prime time in drug discovery.
I'm just jealous at everything that Ilias set up, with the automated system. And we'd love to just say, right, we'll give you 10 lines and we'd like, 100 organoids next week, please, of astrocytes, and with your new technology. It'd be great. Save me a ton of money. And I think if we had a centralized way of making cells, and this could be combined, right? Because the resource, I think if NCATS could provide a constant, reliable resource in cells, that would help all of us. And we talked about this at the NCATS, the chip meetings a lot. But On the other hand, you don't want to centralize too much because you lose a lot of the learning things that we're having. Someone might come up with a better way to make IPS and differentiate them, and we don't want to homogenize everything to the point where we lose innovation on differentiation. So kind of there's a yin and yang to that. Maybe a core set of lines would be great that, you know, Jim and I and Jason could all agree on. If we use that in our neuromuscular model, we get an output that's a certain way. We tried to do that a little bit with the CHIP consortium. I think there are some lines we've shared that happen to be from CEDARS, but they could be from anywhere.
The call center sounds really good. If we need a new model, if there's a place where we can consult within NCATS, you know, and if there's some advice that you can provide, you know, where we can get the new models, new advance within the chip field, that would be really nice. Right now, you know, I have to go through a lot of literature and then a lot of meeting notes to find, you know, where which lab or which company is the best option for our purpose. You know, if we can cut short on the time, that would be really advantageous for us as well.
So in addition to small molecules, are there any other modalities, therapeutic modalities that you think these models are suitable? I mean, there's no reason that others are not, but I'm just like, you know, just trying to find out your opinion, which kind of you would prioritize.
Well, I'm biased for the gene therapy.
Yeah, I mean, gene therapy is we know small molecules work. We know biologics work. We can test antibodies. ASOs, I think there's enough data out there showing that we can work with ASOs. You know we can work with the gene therapy too, but that really needs to be shown, you know, and done, because I think there's things that the microphysiological systems can do with gene therapy that you just can't get any other way.
Yeah, I agreed. So when I was showing that so zilucoplan actually is a macrocyclic peptide, so it's not a antibody but a small peptide. So there's and then I think also small molecule, there's opportunities large molecule, but the other area definitely in the rare disease could be gene therapy as well.
I want to go back to a regulatory, you know, how these models can be used for regulatory submissions. You know, certainly we know with rare diseases, there's multiple challenges, and the regulatory landscape is certainly one of the areas where there's significant challenges. And Jimmy talked about how you've submitted INDs using this data as efficacy data.
It's efficacy data. We're working on safety, but it wouldn't be for the INDs we submitted. It was efficacy data only with our systems. Okay, so there wasn't a lot of other supporting. It was basically efficacy data, and that was allowed to proceed, you know, to the phase 2. So that's what we try and do, is that. But you know, it's the functional aspect, I think, is what something they can, you know, we can show a one-to-one correspondence with what they see clinically, like for the rare diseases, for the peripheral neuropathy, it was the conduction velocity. Well, that's what they measure in a patient. There was a one-to-one correspondence. You know, when we show for the ALS stuff, when we can show basically spasticity and loss of grip strength and show a one-to-one correspondence with what they're measuring, I think that's why I believe that the ones we put in so far that have gotten into clinical trials have been successful. You know, it's because that one-to-one correspondence for what they're seeing functionally in a patient.
And do you think that it was sufficient because of that one-to-one correlation that you saw? And was there no other model that could be used in these early studies, in these efficacy studies?
I mean, I don't know. I mean, there probably was some animal models that they probably could have used. I mean, they could have humanized a mouse. I mean, but they chose not to request an animal model.
Yeah.
You know, and so, you know, kudos to them, right? So too much we get, you know, yeah, that's really nice, but we still want to see the animal data. You know, we see that quite a bit.
Right.
You know, and so, but again, I mean, you made a really good point of it. There's over 10,000 rare diseases. There's only 500 active research programs because there are no animal models. So this is a huge opportunity for rare diseases, is to make advantage of these microphysiological systems. It's just a huge opportunity.
Well, that's why we use the retina organizer and retina on a chip, because we didn't have accessible animal models. I mean, there was animal models in the literature, but there was no accessible animal models that we can use. And even the dog model, there was huge immunogenicity. So when there is a safety issue, we don't know if it's related to the test articles or it's just the species difference. So it's hard to tease out that safety issue. So with this in vitro human models, you are just straight to the question and answer. Is this drug really effective in the human model? And you can answer with this in vitro model. So there's a huge advantage with this. With the rare disorders, one of the diseases that I'm working on, I cannot find the iPSCs right now. So we have to make it in that case. So there's a availability of animal models, and then how replicable for the human disease in this animal model. So there's two issues that we can tackle with iPSC-driven in vitro models, I think.
Yeah, I think that retinal model you showed is fantastic because you had all the pieces. You gave the AAV. You could even do dosing toxicity studies separate from the immunological problems of adding AAV to patients. So you can get a lot done. And I think, like I said, adding that to an IND package is just going to strengthen it, I imagine, and help get that through. And that's where we're going. Now,
so when we went to CTA, European CTA, we didn't have real in vivo pharmacology data. This was the pharmacology data for the CTA submission. We had a tox studies with the animal models, but this was the only pharmacology study.
And one thing I want to add is, this came out of the meeting I'm at here, actually, this retinal meeting I'm at, is, the idea of having a JAX Labs kind of set of IPS lines from rare diseases where you can just go order them and not have to bother trying to find the patient, and, even have CRISPR editing on some of the key ones. So if we come up with a list of the top 100 rare diseases that we could model, if NCATS or NIH could coordinate, just having the IPS lines made and maybe CRISPR edited, and someone like JAX would be great because they're used to doing things at scale and they want to get away from animal models. They've made their whole foundation on animal models, but I know Mike Ward now has his whole system there. This is the iNeurons, that you can directly put any genetic mutation into a cell and get a line out from it. And JAX are starting to do that at scale. I think there's a validity to that. Also, just collecting the actual patient lines gives you the background genetics that's a complementary to the Michael Ward technique. So I think it's something that would be incredibly useful to all of us modelers, is to have access to and annotate it, right? So have clinical history, and even a whole genome sequencing would be great. A little bit like Answer ALS that I was discussing, and that would push the field forward enormously, because then all we have to deal with is putting them in our chips, not trying to source the cells, which to me is big roadblock for these rares.
All right, I think we're ready to close the day. It's been a long day, and I would like to thank all the speakers. It's been terrific. I've learned a lot. I think, like Jay said, the potential is huge, and I think as the next steps, as Liz mentioned in the morning, I'm gonna make this closing remarks short because it's late. But I think as the next steps, I think we're gonna gather like the highlights of the different sessions, what ideas. I think it's loud and clear that we need cells, patient cells. I think it's loud and clear that we need validation and standardization. I think it's loud and clear that we need clinical benchmarking. But the great news is that some of these models, like Jay is saying, are already producing data that it's enabling IND filings. It's being used at different stages of drug discovery, which is amazing. Some of the, I like the word communities. There's all these communities coming up, whether it's cystic fibrosis, ALS, whether it's kidney diseases, that are doing a lot of work, getting tissue samples from patients, getting omics in there, using machine learning to come up with ways to stratify patients, getting responses from 3D models, the responders versus non-responders. Very, very interesting work. And I think going on in the future is how we sort of get all these communities to change notes, perhaps how we leverage what they've learned, how we leverage what how they did it, and really make it a more systematic approach to help all the different rare diseases more efficiently. So thank you, the speakers. Thank you, all the attendees. Thank you, the moderators. And as I said, we're gonna gather the highlights. We're gonna try to put a paper together, and we'll reach out to the speakers. Just 1 second. Oh, sorry. I've been told that you couldn't hear me very well, but I hope you got most of it. So I think that's gonna be it. And thanks again, and have a good day, have a good night, and thank you.
All right, good to see you all. Take care. Thank you much.
Thank you.
Thank
Bye.
you.